to act as rapporteurs to note the proceedings of the session. In this session, we have 5 lead talks. One from Dr. M.S. Prasad sir, another one Dr. A.K. Padma Kumari madam, and Dr. T.R. Sharma sir. And then we have a talk from Dr. R.M. Sundaram sir, and the last one will be from Dr. Amresh Chandal sir. With this, now I hand over the session to chair of the session.
Very good morning to all. Distinguished participants, ladies and gentlemen, I would like to welcome you all for this very interesting session on advances in molecular breeding and tools for multiple pest and disease resistance breeding in crops. And today we are dealing with this in this session on rice. You know, rice is one of the most important food security crops, and this year we have become #1 in rice production, producing 150 million tons of rice. But still, you know, we have to overcome many problems which are associated with rice cultivation, and in that context, this is one of the most important sessions which I consider. And also, being working in rice for the past more than 35 years, I also considered it a privilege to be here this morning to chair this session and also to deliver a talk. I would also like to welcome my co-chair, Dr. Viresh Gowda, Head R&D Bayer Bioscience, Hyderabad, and also would like to welcome our former DDG Horticulture, Dr. N. Krishnan Kumar, Dr. Sundaram, Director, Indian Institute of Rice Research, and our very senior colleague, Dr. B.M. Khadi, Dr. Tikku, and all of you to this particular session, and I'm sure you will enjoy the deliberations and discussions during this session. So with these words now, I would like to first invite Dr. M.S. Prasad, Principal Scientist and Senior Plant Pathologist at Indian Institute of Rice Research, Hyderabad, to deliver a talk on disease scenario in rice. In fact, Dr. Prasad was also one of my old collaborator and colleagues with whom we did a lot of work on PI54 and blast resistance during my initial days at IRI. And thank you, Dr. Prasad. You are given 25 minutes to complete your talk. Okay?
Thank
Please
you, sir.
continue.
Thank you, sir. Respected Dr. T.R. Sharma ji, and then Dr. Gowda sahab, and then respected Kumar sir, and then Director Sundaram, and my dear colleagues. The task which I have been given to today's program is that I have to give you a scenario of rice diseases in India. So I'll stick to my presentation to the scenario only. The molecular part and other things Dr. Sundaram will take care of from the rice. So I'll give a brief outline of all the diseases that where they occur, and then let us. Can you pass it on the slides? You pass it. I'll say next. Just now, our sir told that T.R. Sharma sir that India already surpassed China in production. It is the first in the world that we have achieved 142 million tons, and then it is produced in different agro ecosystems. So that all we know very well. Then go to the next slide. Just this slide gives a wonderful how the disease progression took place in rice. Actually, I made 4 breakups like pre-60 and then 65 above, and 65 to 85, 85 to 2005, 2005 onwards. how the agriculture, actually a traditional period to green revolution to intensification in agriculture and then climate change and hybrid era. So how the disease scenario has taken place. In 60s, pre-60s, the disease pressure was low, the two brown spot and blast. And 60 and above is 85, it is a high increase of disease pressure, and then the blast and BLB again dominated. 85, 2005, high and complex scenarios occurred, and then here the sheath blight has dominated, and then Tungro has taken the toll of rice crop. And then above, after 2005, the very high intensity and the shift in the cultivation, like hybrid race has come into picture, and then there the blast and false smut has taken over, and then Buckany. Because of this, these two diseases, this is the scenario, the changing scenario over the period of time in rice diseases. This is what I wanted to. In 1945, we know only one disease, that's the brown spot disease, that's a famine, that everybody knows, those who read agriculture. So after that blast, now as on today, that around 13 major diseases are there that are threatening the cultivation of rice. This is the slide that very nicely depicted how the progression took place. Here, what are the reasons that disease scenario is changing or shifting? Here, the main factor is a climatic factor. That as we are all aware that there is a change in the temperature, rise in the night temperature, and also increased humidity due to erratic rainfall. Rainfalls are very erratic, and frequent floods and droughts, and extended cropping windows, like the cropping window is extended, and then that favors the survival of the pathogen. These are some of the factors from the another factors are changing in cropping systems. Like now we are advocating the farmers that we go for the DSR mode of cultivation. There the disease scenario is differs, that brown spot again it is coming up very high. So in some places the irrigation actually very high intensive cultivation they are doing, like Telangana I took as a model, that in recent times in Telangana the rice cultivation has gone up very high. So there the pathogen system also, disease also very high intensity throughout the year. Like continuous cropping, like year by season by season we are going only rice. These are some of the factors that contributes in the changing the disease scenario. And as we very well know that the host genetic factors, like only few popular varieties that are specific to some regions, like in Andhra we go only BPT, in some Tamil Nadu Ponni, like there several varieties are there. So those farmers they prefer those varieties only because of their quality and all, but they are susceptible to the pathogens. So that breakdown of the resistance is another problem. Now at the pathogen front also there are changes, pathotypes, new pathotypes and new races are coming, and then adaptability, and then virulence, increased virulence, and survival during off season also increased. Then another one more factor we have to take into consideration. Now use of the N factor is very high, but people are not bothering about how K is to be utilized, and then micronutrient management also people are not bothering. That's why some of the diseases like blast and BLB, they are very high incidence had been happened in recent times. Now this is the actually picture what we generally see in rice, that in regarding fungal diseases there are around 13 diseases are there. And then bacterial diseases, generally two are made dominant, one is BLB. In viral diseases, we know only Tungro, but now recent times, one disease, the southern rice black streak dwarf virus are known that in Punjab and Haryana, how it is threatened cultivation of basmati. You take the example of Buckany also. Buckany also recent times, the increase has been very high in Punjab and these places, Haryana, where the basmati rice is being cultivated. False smut, as you all are very well aware that it was once a minor disease and then it is a Lakshmi disease we call, but now a stage has come without managing the false disease in hybrids. I think that cultivation is unsuccessful and they are getting a lot of losses. The other diseases like blast, every year it occurs. It takes, even still it is a major disease, and then brown spot now it is aggravating year by year, and then because as I told earlier also that the DSR shifting of the cultivation, and then people are taking in upperlands, and then where the nutrient management is not proper, this disease is very much aggravated. Leaf scald, this western belt of India, the leaf scald is very much Konkan region and all. Leaf scald is also very severely occurring, and the sheath blight, once it is known to occur only in the coastal belts of West Bengal to down to Tamil Nadu, but now it has gone into other parts of the country, like in Gangetic plains and then Punjab, Haryana, even in North Eastern also you will be seeing in coming slides. Udamata disease, earlier known only in Karnataka. Now in few pockets, this disease is also being observed. And then stag-born, and then bunt, narrow brown spot. These are all now, they are the minor ones, but we don't know what a scenario will take place tomorrow because of these outbreaks. Actually, the strength of our IARR is we have around 48 funded centers. There our group is State Agriculture University people are there. And then we generally work on host plant resistance. That against 6 diseases, every year we screen them, and then we take them a disease-resistant lines, and then we give it to the breeders. And then this is our general program. And then virulence also we will monitor, like 2 pathogens, blast and BLB. And then management aspects, we take care of the management aspect of all these fungal diseases, like using fungicides. Nowadays, only fungicides we are not, site fungicides we are not using, we are using the mixed fungicides. And then now one more, here in this context, like disease scenario, POS only plays a major, because this is also one aspect we take care of at IARR, this production-oriented survey. Our team, that plant pathology team, along with other groups like breeders and then agronomists, they go out for the survey during the crop season. In the entire 48 centers, like it spread across the country, as you have seen. Then they survey it, what are the diseases are there in that season. 2 to 3 times they go for survey, and then they analyze the data, and then they'll send to us what is the now which is the disease happening more in which place. Like now this happens in this disease scenario aspect, production oriented survey I take as a background, and then we'll discuss about each disease, how the change has happened. So our team actually comprises of all agriculture department, line department people, and the State Department of Agriculture, and also our university staff, and also our ICR employees. And then we surveyed almost all the 125 districts every year. And then we put forward that these are the 5 diseases like blast, BLB, sheath blight, false smut, and brown spot. They are the major 5 dominant diseases that are threatening to the cultivation of rice. Now blast, as you know very well, that is a leaf and then node and neck blast. How the leaf blast is also very tricky. Earlier it happens to occur at few places, because now it has gone to several interior places where earlier it was not reported also, because of change in the climate or because of wind. The wind what we are using it that for crop period, that is also very less now. As I told that this is a production oriented survey. Every year we have a data of a repository for the last 30 years. So we have analyzed the data of the 30 years data for all the 5 diseases, and then we come out a map, that the decadal map of each disease, and then that map shows that how disease has progression has happened. Now I will present for each disease. Oh, this is not clear. Then one, I think you missed. It's not clear. Okay, I'll explain that. You just go back. Oh, it is not coming. You just, yeah. Wait. Oh, it is. I think something happened, sir. I will proceed further. Yeah, I have. मैं आगे जाता हूँ. Sir, leaf blast, it is also in the 3 decades we have taken the maps of leaf blast. It has shifted from 10 years to 10 years. Now it has gone to the deeper into the different places. Now this is a case of neck blast. Don't do it anything. I'll do from here. You just tap that window also. Yeah. Okay. This is a neck blast scenario. Earlier neck blast was not that much prominent. Now these increased rain during the flowering and ripening stage, mild night temperature increase, and then high use of N application, and then only one variety, that's what I told, the preferential variety that is available on the ground, that favors the enhancement of this disease, very severe levels. In 81 to 90, this was very sparse in the country, you see the dots, and then in 91 to 2000, it was a little bit aggravated. Now you see the present scenario, it has gone to the, wherever rice is grown, I think now neck blast is also a major problem. Actually, at DRR, we have regarding this topic is actually related to genes and then molecular level. So I wanted to show some of the work what we did at IARR in collaboration with Dr. T.R. Sharma sir. Sir has told that we have several common projects were there. So in BLAST, we have taken the first work at IARR in collaboration with IARI. Dr. Sir was the main PA for the time. And then we didn't have that monogenic lines background, in the background of different genes what we have, like PA1, PA2, PA9 and PA54. These are the 4 major genes available. So we didn't have that monogenic lines. For BLB we have monogenic lines, but here we didn't have. So our idea was to develop monogenic lines, a project we were closely associated, and then we developed monogenic lines in the 2 backgrounds, 2 popular varieties like Swarna and then BPT. So different lines, why these lines we developed, I will tell you, again it will come. So in those 12 lines what we have developed in the both the backgrounds, 2 out yielded the other lines that were, so the 2 were released as a dearer than 51 in the background of which is having a PA2 in the background of Swarna, and then dearer than 62 it is having both BLB and blast resistance. So those monogenic lines, why we developed, because we wanted to give as a donor to the directly used for breeders, but in that we have put those, as suggested by Sharma sir during that time as a DDG, we took these aspects, we put them in the 48 locations, how these genes are performing. So we are given a gene map that in zone 1 what are the genes that are really working well for blast, and zone 2 and zone 3, zone 4 up to zone 7. These are the different combinations and the different gene combinations we have given. Out of all, the PA54 was the best one in most of the zones. And now we discuss with sheath blight. Actually this sheath blight disease is easy to work but difficult to manage, because it grows very well in the petri plate, but very difficult to manage this disease. Once it comes into the main field, I think years together it will be there. The sclerotial body survive, I think 6 to 7 years. So here, this is a picture in the global level how this disease is there. And then one of my scientists, Dr. Prakasham, is working on this disease along with Dr. Sundaram. And then here also we made some maps. Earlier this disease was known to occur only in the coastal belts. But now you see the recent map, sheath blight, it has gone to very high level in the northeastern and then Gangetic plains, and also in Punjab and Ludhiana. Very high intensity, because here the challenging task is there, we don't have any source of resistance. There in blast we have different source of resistance, but here we don't have a source of resistance. At IARR, we have identified some source of resistance like some QTLs in the setup, and then in the background in setup, and also Vajupek. I think Sundaram will tell about in detail about those genes and then how they work in sheath blight. Bacterial blight. This is the actually disease. Earlier it was very minor, very mildly it is appearing, but in recent times, I think it is appearing in a very severe form. Wherever you go, I think it is threatening the rice cultivation. Dr. Laha, one of my colleagues, is working on the disease, and then with the help of Sir Sundaram, both together they work. This is a bacterial blight, bacterial leaf streak, and panicle blight. These 3 are present, as rest 2 are, I reported, they are not in India. Again, some problem has come. Okay, this is also the map which he missed actually in the how it time series it changed the area to that area to, how intense this disease was. This is international. This is a global picture of BB, mainly in Asia and some parts of Western Africa, and also USA and then South America. These are existing in those places. In India, it is endemic, mostly in Punjab, Haryana, Uttar Pradesh, parts of Uttarakhand, Bihar, Jharkhand, Chhattisgarh, West Bengal. Most of the rice growing places it is there, as you see in the map. Because this is a major, now it has become a major production constraint. Here we can't manage this disease by chemicals, because fungal disease there are chemicals available. At least you can manage if you take rightly application right time. But here this disease is not having it. This is a bacterial disease. We don't have chemicals for this disease. We have to go through certainly through host plant resistance. Very great work has been taken up at IARR with Dr. Sundaram and team, and then in that Dr. Laha is one of the person, and we all took forward this work. This is a map, decadal map of this bacterial blight. This is also very severe form. In southern India, it increased a lot, like Kerala, Tamil Nadu, Andhra, Telangana. This incidence nowadays is hampering the cultivation. Even in Punjab, Ludhiana also this disease is very severe. These are the things that in some pockets it has gone down, in some pockets it has increased. Like Punjab and then Haryana, that belt, it has come down a little bit over a period of time, little bit. But here in southern belt, it's aggravated like anything, like Andhra, Telangana, Karnataka, Kerala, Tamil Nadu, Pondicherry. This you see the red map here, it is the most, I think, very severely it is occurring year by year. This is the intensity you can see, bacterial blight, how it is there in East and West Godavari districts of Andhra Pradesh. This is in Telangana. You can see acres and acres it has gone like that only. Here the main, actually the intervention came from IARR, the first variety, that is a gene pyramidal line developed by Dr. Sundaram and our team, and there is improved Sama Masuri. It has taken a solace and then it has gone into deeper into the farmers. Now that cultivation has picked up. Again, that also a little bit toned down, and then again we identified a gene 38 and then actually 38, and that has been incorporated. That is taken still further strength. Still some more gene, one more gene they identified. They are putting it in the background. This is dearer than 53. It is a wonderful variety. And then you see the incidence of BBT on BBT and all dearer than 53 in Telangana. These are some of the lines which are having bacterial leaf blight resistant genes. These are the nutshell about bacterial blight. And then one of our colleague, the Basavaraj, is working on brown spot disease. I told you that this disease is picking up very fast, and then because of changing in the cultivation pattern like DSR and all the methods, we know only two famines in the Bengal famine, and then 1918 is this Krishna and Godavari Delta famine. But now, this is the scenario of this disease. This disease is there in mostly Madhya Pradesh, Uttar Pradesh, Bihar, Chhattisgarh, Odisha, Assam, Jharkhand, Punjab and Karnataka, AP, Telangana, West Bengal. In a very severe form nowadays, this disease is there. You can see in these maps, in decadal maps, how it is progressed to after 10 years to 10 years. In some areas, what are the real, in case of Chhattisgarh and Jharkhand, he has gone further and then identified the pockets of the places where it is really affecting the cultivation. This is, see the severity in the survey you have collected such a severe symptoms are there. Here the one work I would like to stress here, this is actually very sparse growth, but maintaining one culture itself is very difficult, brown spot. But here at IIRR, we have developed a wonderful, that Basavaraj, and then we have developed a wonderful technique to sporulate it. Actually, sporulation is a major problem here. So he has developed one technique with rapid food agar medium. He is going to patent it, and then using this technique in art, because we need to screen a number of lines in a short period of time. This is a facility we have created. When Dr. Sir was the DDG, T.R. Sharma, he has inaugurated this facility at IIRR. And then these are some of the work has gone further to identify some of the resistant lines in the brown spot, and then identified few resistant lines, and then they are being now put it into the breeding program. Now another disease, this is one of the false mud disease. Lata Lakshmi is working on this. Since she joined, we asked her to work on this disease only, because that time the etiology was not known. Even fungus also, it was not even culturable properly. Now we have gone to a level that we have a repository of, I think, 200 to 300 lines we have, cultures we have across the country. And also this disease artificially we have inoculated. Artificially we have created the disease. In this is maps I wanted to show this severity. Here also the false mud, after the advent of hybridizers, it has gone up like anything in the 2013, 23 years. You see the change in the disease pattern. And now at IARR, we have developed a facility called artificial screening in a large scale germplasm of rice. This is a wonderful technique, and it has been applied for patent. In field evaluation also we have gone. This is a field evaluation technique. With hypodermic system they inject, and then typical type of the symptoms it will produce there also. It's a wonderful technique, and then it has been, patent has been applied. And the strength of our institute is actually molecular breeding people are sitting here. And then if you want to be a successful molecular breeder or molecular person, you need to have thorough phenotyping background work like pathologist. So we can clearly say that we have for blast wonderful screening facility UBN, we have false smut screening facility at IARR, and also sheath blight, bacterial blight, brown spot. Now we have gone for sheath rot. There's one Jesudas who has joined and he has developed wonderful technique for sheath rot, and also rice tungro. I think tungro, nowhere that screening facility is available, I think except IARR, I feel. So it's the only place where we are getting it screened. With this, I think with 1 or 2 slides, I'll project this management of rice diseases. You know very well that is a host plant resistance is a major what we are looking at IARR, and then several lines have been developed. And then now one scientist joined on biocontrol aspect also, few work, very good work he has conducted. Anyway, the fungicides, anyway, fungal diseases without fungicides, when it is in severe form, we have to go for resort to the fungicide supply. Maybe now mixtures of fungicides we are going. These are some of the lines developed at DRR using different, these are with the different genes, gene pyramid lines. With is a collaborative work of breeding, crop protection, and also biotechnology. This I told you that one person giant is a Kannan. He has identified 3. One is Trichoderma aspilarum, tyke, and then bike, Bacillus species, and also one more is there, Pseudomonas species. These 3 he has now gone to another level of delivery system, and then he has about to patent the mechanism of delivery and everything. It's a wonderful work, and these are all maintained against fungal diseases. Their utility was there. Slide has gone. One more slide was there. It was IPM slide, sir. Without IPM, I think we have wonderful 7 zones are there, rice zone. At each zone, specific IPM module has been developed. So with this, I thank all of you for patiently listening. Our chairman sir and my director. Without their help, I think it will be my colleagues at IARRI, their help. Otherwise, it is not possible to do the work, sir. Thank you, sir.
Thank you, Dr. Prasad. Excellent presentation, and you have given us an overall scenario of all these diseases, how these are becoming prevalent now, which have become obscure in previous years. So that's really very good, and I don't know whether we can take questions. We have time for questions, 1 or 2 questions. Yeah. He is asking about
I got his point. Rice tungro. Tungro actually in recent times, our survey says that its incidence is very minimal. That too, it is in small pockets in Tamil Nadu and then in West Bengal. It is there, very small area, and the intensity is also very low. That's why the scientists are also not coming forward to screen it. Yes. That's why we had only, because since it is a rice institute, we cannot ignore that screening of the disease. We are doing it at IARR level only. But as per the intensity level, it is very less. One disease I told you that southern, that is there now, but it is also very erratic. 1 year it was very high on basmati, and next year it disappeared. Again, last year a little bit was there.
One more, sir.
Yeah.
I seen their screening protocol, their neck blast screening protocol not there. So you are working on
because I don't want to, because neck blast protocol is the protocol we have developed. That is also one of the, because neck blast screening is very difficult. Leaf blast, okay. Yeah. It is standardized protocols. But neck blast, we have developed it IARR. That I cut the slides because of time, paucity of the time. It was there still last night.
Maybe you can discuss later.
Yes, you can discuss. Because a wonderful protocol is there for that.
Yes. Thank you, sir.
Thank you.
Good morning, sir. Just I have to know clarity. When we see the severity of leaf blast and neck blast, but in the field there will be less severity. Otherwise, no disease in the leaf blast at early stage. But same field, when we see at the panicle stage, we will get very severe of neck blast. What would be the reason?
Actually, I told that blast appears because that 3 critical growth stages are there for blast. One is leaf, another is node, another is panicle. So at three critical stages. Why I am telling critical stages? On these critical stages, the management aspect depends. So when in these critical stages, the climate, because blast is very much prone to the climatic factors. That conducive climate is available on the and then a susceptible variety available on the ground, it will knock. Suppose it may be the escape at leaf blast, and at neck blast, these conducive conditions are prevailed. That's why the disease has been very rampant. Nowadays, the season is little bit progressed. When the neck blast, in November, earlier the season was a little bit earlier. Now the season little bit more. That it extended to November. That panicle emergence. That time either humidity or wind splash of some splash rain, they are there. That is favoring the disease development. That is the reason why.
And one more, sir. How about the PI2 and PI54? Is it working all over, uniform working?
PI54 is the, I told you also, because SAR is here, I am not telling, but the PI54 is very strong, because it had the capacity to knock down almost all the pathotypes.
All over
the country. PI54 had that, even in hilly area, plains, everywhere. PI2, PI1, they had limited this thing.
Maybe last question from. नहीं आपका हो गया.
Okay.
Is it that PI54 is means resistant for neck blast or what?
Yes, sir. Yes, sir. It is there.
Yeah.
It's there.
Is it?
Okay, thank you,
Thank
Dr.
you, sir.
Prasad. We will stop this discussion now. Thank you for excellent presentation.
Sir, let them mail the questions, whatever that they have. In case they still have questions, they can bring it to him or you.
Okay.
Then that,
Okay, but we are getting suggestion from the organizer that those who have not able to ask question, they can send by mail all those questions to Dr. Prasad or to Dr. Meenakshi Sundaram or to me, because then we can reply. Yeah. Thank you very much. For next presenter, I will request my co-chair to invite him.
Thank you. Now I would like to invite Dr. A.P. Padma Kumari, Head of Entomology, IIR Hyderabad, to talk on current insect pest scenario in rice. It will be online.
Unfortunately, the screening protocol was not available earlier, but now they have to. Yeah, very. Have you seen the? We should go and The Kani is coming in the Bagani and Javan.
She is speaking at Parliamentary meeting. Hello? No, we can't hear you, madam. It should be okay now.
Am I audible?
Yeah, now we can hear you, madam. Please go ahead.
Oh. You are sharing the slides? Are you sharing the slides?
Yeah, our team is doing that one, madam. Just give us a minute.
Yeah. Okay. Or I can share from my end also if you permit me.
Yeah, that would be good, Dr. Padma Kumari. Please go ahead. Yeah.
If the host can permit me to share the slides, I can share. It says host is not allowing sharing for this meeting. So host has to allow me to share the slides. No, meeting ID is 875-9478-7484. Meeting ID. Are you able to see me? This is, the meeting ID is 875-9478-7484. And the passcode is 58005.
This meeting is being recorded.
875-9478.
We should have.
I think slides.
Yeah, we can see your slides, madam. Please go ahead.
Voice audible to you all?
Yeah.
Okay, thank you. So, very good morning to you all. I'll start my presentation. Respected chair, co-chair, dignitaries on the dais and off the dais, faculty and students. Today, I'll be speaking to you on the current status of insect pest scenario on rice. And we can see that rice is grown in many ecosystems, like it is grown in many ecosystems, like direct seeded rice, hilly rice, Kuttanad area, irrigated rice and deep water rices. And all these cultivation systems influence the pest scenario. And if you go into, I'll be briefly talking about the pests of rice. And there are 100 insect pests that inflict rice, but 7 are important. And there are some regional pests confined to few regions, of which 5 are important. And we can see that 5 are. And here in emerging pests, there are whiteflies, rice mealybug, termites, and panicle mites that are of growing concern. And if you see the importance of the rice pest stages and the insects that inflict the crop, most of them are vegetative feeders. And in the early stages, if we take chemical control appropriately, the damage is compensated in the vegetative phase. But there are some insects where the tiller damage is like gall midge or stem borer and BPH, which affect from right from sowing till harvest. And here lots of compensation is not possible, and invariably we have to look for other mechanisms of tolerance. And most of the, if you look at the stem borers, gall midge, leaf folder, plant hoppers and gandhibug, these are the major pests of national significance. And in stem borer complex, there are many species, of which if you see I can say that yellow stem borer is the most dominant yellow stem borer is the most dominant pest among the all species of stem borers, that is the Syrpophaga insectivus. But there is white stem borer in the Himachal area and in the Kuttanad area. which is majorly a vegetative pest in Kutanad area, but white stem borer in Himachal, it causes both vegetative and reproductive stage damage. But of late, with the change in cultivation practices, you can see that it is gaining pink stem borer and dark-headed borers are gaining prominence. And here, if you look at the damage, everything is dead heart or white ear. And here we can see that across India, it is almost above ETL. And white ear damage, it is spread across India, it is again above ETL. This is the average of 5 years data from our ECRIP reports. It is spread widely across India. But when you look at the species composition, you can see some are yellow stem borer, which goes deep into the root system and it survives in the stubbles. Pink stem borer is gregarious and it causes lots of tunneling. And dark-headed borer, it causes both white ear and dead hearts, and it is gregarious in nature. So the damage they inflict is much more as compared to yellow stem borer because there will be extensive tunneling in the tillers. But we only worked ETLs and 10% dead heart is taken as the criteria, and we have 6 pheromones only for yellow stem borer control. Coming to the species composition, in one study in Srikakulam area and West Godavari and Nellore of Andhra Pradesh, in rice-rice pest cropping system, we could see only yellow stem borer. But wherever there was a rice-maize cropping system, we could see in West Godavari, pink stem borer followed by yellow stem borer. But when there were other cropping systems adjacent to rice, like sugarcane, ragi, and other millets, we could see 3 species that are occurring in the rice crop. It is mostly dark-headed striped borer, pink stem borer, and yellow stem borer. So the cropping systems also do matter in the occurrence of the stem borer, but mostly all the symptoms are either dead heart or white ear. And in deep water rices, it's exclusively by yellow stem borer where it can survive and flourish. And coming to the galmage, here you can see in the coastal...
The recording has stopped.
All through you can see. Am I audible, please?
Yes.
Am I audible?
Yes, madam.
Hello?
We can hear you, madam. Please go ahead.
Am I audible? Can someone tell me whether... Am I audible?
Yes. We can hear you, madam. Please go ahead.
Thank you. Because recording has stopped, then I wanted to confirm. So here you can see that in the galmage here, it is along the coastal belt and up to interior Sakoli we can see, and it is always above the ETL. And here you can see that the tillers are damaged, and if the variety is having more tillers, there will be a bit of compensation, otherwise the losses are huge. And even the stubbles, they serve as a source of inoculum, and ratoon rices, they are a big source of inoculum. Here you can see that the feeding stage is very less of the maggot, and people go in for sprays. As the feeding stage has a low duration, spraying is not effective. We advocate granular insecticides, and of late not many granular insecticides for this general purpose are available. And this PPH you can see, it is a dangerous pest and it keeps sucking the sap from the tillers and it debilitates the plant, leading to hopper burns. And here you can see the whole tillers, they get chappy, and then it travels from plant to plant and becomes it loses its vigor. But coupled with unseasonal rains and lots of infestation due to migratory population, most of the lodging occurs at this particular stage. And this is always above ETL in most of the areas, and most of the insecticides are directed either to BPH or to yellow stem borer. And here, due to unseasonal rains, we can see that lots of damage is occurring and people are not able to harvest the crop because of lodging. And coming to whiteback plant hopper, most of the time BPH and WBPH coexist, But here, WBPH actually colonizes the crop, and after that, BPH takes over due to its high multiplication rate. But since 2022, we could see that some of the states, this southern rice blackstreak dwarf virus, also known as Fiji virus, is gaining prominence. And what we could see that due to the spread of this virus, the infected plants develop narrow upright leaves, weak roots. And here you can see that PR131 is the highly susceptible variety. And farmers are uprooting the plants, and the cost involved is around 10,000 to 15,000 per acre on resowing, and about 15,000 per acre for insecticides. And not many farmers are willing to take up rice because of this particular virus, and close monitoring is essential in the nurseries to prevent the spread of the virus. And the next one is the GLH, that is the green leaf hopper, where this year, after a gap of I think 5 to 6 years, we could see tumgro infestation in the West Bengal area of Chinsura, and it's a xylem feeler, and nearly more than 50 acres has been damaged by this particular pest, causing tumgro-like symptoms. And here we can see that virulence monitoring is a constant endeavor in our studies, and we could see that we monitor virulence for brown plant hopper and galmich through a set of host plant differentials. And we could see that our Indian populations vary in their reaction to certain genotypes. So we should have our own donors in conferring resistance to this particular BPH or to galmich. And it is a vicious cycle. Once you deploy a resistant cultivar, there would be virulence. And once virulence is there, again we look for more genotypes. And we are going for pyramiding of genes. I think GM1 and GM4, or GM4 plus GM8 and GM1 plus GM8 are more effective. Coming to the sources of resistance, land races and breeding lines are being exploited. Once we don't find any significance in these significant donors in these lines, we look for wild races and mutagenesis. In such one endeavor, we could find that through EMS mutagenesis, we could identify few sources of resistance for stem borer. It can be called as tolerance at vegetative phase. And out of the, for BPH, 51 genes identified across world, only 4 to 5 sources are working very well for Indian populations. WBPH genes have to be exploited. Galmich genes, 11 genes we are deploying, out of which only 3 genes are very effective, and we are going for gene combinations. We have identified leaf folder QTLs and YSB QTLs. This knowledge is very much useful for this symposium, where we are looking for molecular breeding tools. We have donors for both the insect pests, multiple insect pests, and donors conferring resistance to both galmich and BLB, and heat tolerance and WBPH in N22 mutants. Leaf folder, this is also a major pest, and you can see it is spreading across India, and it is always above ETL. And there is a complex in this, but of this, Nephilocrosis is the most dominant species. And here you can see that in the early stages, just after transplanting, if there is a leaf folder incidence, there will be less possibility of recovery. So insecticide spray is a must. But losses will occur again in the flag, if the flag leaf is affected, because chlorophyll in damage will be, chlorophyll will be depleted and there is reduced turgor. And this causes losses in the yield. Definitely it will have loss in the yield. And this species composition is important because when we are deploying pheromones which are biorational approaches, we should know which species we are addressing to. So more detailed studies on the species composition at very location specific is important. And most of the insecticides are directed towards leaf folder because after a certain stage in the crop, they start moving in large numbers to another green crop. And farmer always thinks that these are migrating and they cause lots of damage, and they tend to spray many insecticides. Earhead bug or gandhi bug, it causes pecky rice, and it also affects the quality of the grain. And here also species is very much important, but all of them cause specky rice. Thrips. Thrips are gaining prominence because in the nurseries where there is a drought for 10 to short spell of drought for 10 to 15 days, you can see the tips of the leaves being rolled out, and this causes drying. There is resistance to this, but appropriate chemical control will address the problem. And you can see in some parts this is a common thing. Insect pests of regional significance, you have termites, white grubs, swarming caterpillars, grasshoppers, caseworm, cutworm, hispa, and mealybug. And if you have hispa here, it is more in the hilly tracts of Himachal and Assam. It scrapes both grub and adult, they cause damage. It scrapes the chlorophyll. And if the vegetative phase it occurs, small seedlings, if it affects the small seedlings, recovery would be difficult. But in the mid-tillering phase, if there is any infestation, definitely it would be recovered. And insecticides is the best possible control measure. And here in caseworm, in Monkampu, Brahmawad, Karaikal and Rajnagar, this has become a serious problem because if there is drainage problems, lack of drainage is increasing this pest, and of late this is gaining prominence and it is creating vegetative phase damage, and again insecticide or pulling a rope will be the alternates for this control. And grasshoppers are a perennial problem in Kudwani area of Jammu Kashmir, and it is also observed in Adutara area, and white grubs are a problem in Kumaon region of Uttaranchal. Emerging insect pests are mites, which will be leaf mite, which is always there, but panicle mite is gaining prominence. Blackbug, blue beetles, and whitefly in rice. Here we could see that blackbug outbreak was reported in 14 when there was an unseasonal rain. In light trap catches it was there up to 23,190 in 2 days. These unseasonal incidents of rains, they create a favorable environment for this pest to come onto the rice crop. The symptoms of damage are they suck like a BPH and debilitate the tillers, but they also cause white ears, so it is all the more dangerous. But it is restricted to Aduthurai area of Tamil Nadu. And here we could see that appropriate monitoring and insecticide measures will take care of this particular insect. Hall maggot is gaining prominence, and you can see all along the coastline it is almost previously it was confined to seedling stage, but now panicle initiation stage also we are seeing this insect. There are few doubts about the species that is causing incidents in the panicle initiation stage because there are losses are high. We are yet to confirm whether it is Hydrelia species or any other dipteran pest, and the work is underway. In rice-based cropping systems, wherever it is rice, we could see that stubbles, they serve as a source of inoculum for both yellow stem borer larvae or galmage or BPH eggs. And winter nurseries, the source of inoculum will be the infected nurseries. I think last year nearly 1.5 lakh hectares of area was infected in the Tungabhadra command area by galmage, and the source of inoculum was spread through infected nurseries. And harvesting through mechanized harvesting, lots of stubbles are left over, and these are the source of inoculum. And weeds are the main sources of inoculum for all these insect pests, and we have to take care of them. And armyworms and cutworms, we can see they just are sporadic in nature. They come, as the name indicates, they come in huge masses and they devour the plants. So only chemical control is the best option. And we have to be vigilant about how the things are going about. And it was found in Mandya district, Chhattisgarh, Odisha, Telangana. And in Maitimna also we could see in Anantnag and Kulgam districts some time back. And it is also reported from Jagdalpur, Patambi and Warangal devouring the whole crop. overnight. And a new pest was reported from Undi area of West Godavari, and this was attributed to the spraying of bifenthrin in rice. Frequent application of synthetic pyrethroid has increased the incidence of Bemisia formosana, and which had led to yellowing, sooty mole formation, chaffiness, and there was finally drying and twisting of the panicles. This information was shared by the in charge from KVK Undi, Dr. Mallikarjuna. And paddy leaf mite, yellow mite is always there, and wherever there is a short spell of drought, we can see that, and one rain would take care of it. But panicle mite and sheath mite is taking that all these days. Here we could see that there will be black lesions on the midrib. People ignore it. If they take a prophylactic measure, that would take care of it. But here people just ignore it, and once there is a rain and the panicle starts emerging out, we could see that most of the grains are sterile. And it is always confused with the dirty panicle. And we could see that most of the farmers think that it is a disease and they go for fungicides. So still people are not aware about or not clear about the dirty panicle that is caused by mites or due to fungus, and there should be an awareness campaign for this. Bloodworms are picking up now. Since 3 to 4 years, these are reported from Punjab, Gujarat, and this year from Chhattisgarh and Nellore also. And in direct seeded rice areas, you can see these red color worms. They are also called as red worms. And there are few studies which say that they are feeding on the fresh foods. So a further etiology has to be studied. Now SRA, system of rice cultivation, then DSR versus pest incidence. Vital spacing adopted in SRA cultivation may favor increased hispa but reduce garbage. In the tillering stage, vigorous plant growth with clusters of tillers may attract defoliators such as cutworm, ear-cutting caterpillars, and leaf folder. However, significant increase in number of tillers and leaves should be able to sufficiently compensate the loss due to defoliation in case of SRA. And SRA cultivation may reduce BPH incidence due to increased aeration. But in some areas where DSR is practiced, when the spacing is not taken into account by the labor who do close-up planting, there is an increase of BPH incidence that we have to take into account. Now, nematodes are gaining prominence, and with the change in system of intensification, system of cultivation, DSR, SRA, and aerobic systems, there is an outbreak of Maladogen graminicola. Many years ago it was there. It was in the nursery. Now it is coming into the main field also. And this year also in Mandya area, we could get the reports that Maladogen is there. So it is everywhere now. White-tip nematode is confined, is seed-borne and has quarantine significance, and it is prevalent in humid areas. Ufra nematode is common in deep water rice. Rice root nematode occurs in irrigated and semi-deep water rice. Root lesion pratylincus are serious problem in upland and aerobic rice. Now, effect of organic manure, it is more effective when we apply organic manure, that is neem cake and poultry manure, which reduces stem borer as well as rice root nematode populations. Vermicompost application can also result in low nematode populations. But these organic packages are more adopted in basmati rice, but can we really cater to the need? Can we really control if the pest is beyond epidemic proportions? And that is a big question mark. Now coming to the toxicity of the
Dr. Padma Kumari.
Insecticides, what we found is with the ban of insecticides, not many granular formulations are available.
Yeah.
And
Dr. Padma Kumari, it would be nice if you can close the presentation in next 2 minutes.
Sure. 2 minutes, sir.
Yeah.
So dealers invariably use and they control the market. State policy and availability of the insecticides matter. Biopesticides 2 through 3 sprays are effective. And finally, with the change in pest scenario, we have 3 to almost 21, 23 pests now. and each has their own role in causing damage. So a basket of options are available for rice apium, but we can address DPH, WBP, and Stembora for host plant resistance. And we have a few mobile clinical labs where the knowledge will be shared in various languages to the farmers, and future researchable issues are pyramiding of insect-resistant genes in allied background, location-specific IPM models, which is a must, exploitation of susceptibility genes, secondary metabolites and metabolic pathways, VOCs, HIPV and HAMP for pest surveillance, habitat management, utilization of food cues for conservation biology, and exploring gene editing as tool. Finally, my word is know your pest, that is KYP, and know your ecosystem, so that pest surveillance and monitoring will add to it, and we can identify the dynamics of the pest. And emphasis on nutrient management, behavior, monitoring of virulence is a must. Location-specific deployment of varieties with a durable broad-spectrum resistance with good quality traits is the need of our. We can look into induced resistance and caramones, and sensitization of location-specific IPM to the agricultural offices. Thank you. I thank our director and the organizers for facilitating this Zoom meeting so that I can share my thoughts with you. My colleagues, Dr. Chitrashankar, Padmavati, Sridhar, Raghavendra, Somsekar, and Chavan, who have given their inputs. Thank you one and all for your patient hearing.
Yeah, thank you so much, Dr. Padma Kumari, for giving an excellent overview on insect pest scenario in India, and also highlighted some of the new pest, emerging pests with a different cropping system, and also bringing some available donors also for the improvement. So thank you for your patience, and since we don't have time, we can go to the next presentation. Please reach out to Dr. Padma Kumari for any questions. So it's my honor now to invite next presenter, Dr. T.R. Sharma, our today's chairman, and also national professor and a former DDG. He comes with 30 years of experience, 30 plus years of experience in the rice. So he is going to present on genomic revolution, discovery and deployment of broad spectrum blotch resistance genes in rice. So over to you, sir.
Okay, good morning to all. Very good morning. I would like to welcome you all for this presentation, specifically Honorable Vice Chancellor Dr. Patil, who spared some time to join here. Can you show my slides? So it's a very interesting symposium, and I would like to thank the organizer, particularly Dr. Sundaram and also Dr. N. Krishnan Kumar, our former DDG, for inviting me here to deliver this talk. Actually, this is a work on various aspects of plant genomics and blast resistance, on which I'm working for the past more than 35 years. And I would like to tell you how we can approach a particular problem and then try to solve that problem systematically over the years. This will also give you an idea. If you spend your resources vertically, then you are bound to get publications, products, patents, and partnership. So I'm sure that they are going to make efforts to show my slides, but by that time I would like to tell the students that it's very important nowadays to select a particular researchable issue which has national and international significance, and then go on chasing your dreams by applying all latest approaches. Thank you very much. We can see now. It's there. Can you go to the first slide? So maybe I will start without the slides. My first part of presentation is on genomic revolution, in which I'm going to tell you how we started decoding of different genomes of eukaryotic species in India. And in that context, most of the students might be aware that the real molecular biology was started with the decoding of structure of DNA by Watson and Crick in 1953 at Cold Spring Harbor Laboratory, which laid the foundation of today's molecular biology. So that's what I was interested to tell you. And how can I move slides? It's not going. It's okay. So this is what I was interested to tell, and it will cover on genomic sequencing efforts, rice blast, its identification of gene, map-based cloning, and then mining orthologs and how we can deploy these genes. Can you go to the next? So in 1953, DNA's double helical structure was decoded by Watson and Crick. And the first sequencing efforts of DNA was made in 1976. That was RNA. And in 1977 by Fred Sangers. And you know that he was the scientist who got 2 Nobel Prizes. One on developing the techniques to decode double helical structure of DNA and also protein sequencing. And if you see the down below here, the first crop plant genome was sequenced in 2005, and in 2001, human genome was sequenced. All these genomes now available in public domain. Next slide, please. And this is how we can sequence a particular genome. Till 2005, we were using a typical Sanger's dideoxy method of sequencing, but after 2005, pyrosequencing was developed, and now we have Illumina, PacBio, and Nanopore-like platform where you can sequence the genomes. And I would like to tell you here, in 1994, when I manually sequenced a genome, within 7 weeks' time, we could get only 100 nucleotides. Now, in 7 hours, you can get full genome. Next slide, please. This is one of the most important efforts in which India was participating in international efforts to sequence rice genome, and we were sequencing chromosome number 11 of rice. And that's how we sequenced Nippon Bear variety of rice with 388 genome size and predicted 37,500 genes in this particular variety. Next slide, please. Subsequent to that, another international genome sequencing effort was there in which India participated and sequenced chromosome number 5 of tomato. And here we sequenced cultivar Heinz 1706 with 900 million base pair genome size and predicted around 34,000 genes in tomato. Next slide, please. Then India also sequenced independently pigeon pea genome, which was published in 2012, and here we used only next generation sequencing approach to decode the complete genome of pigeon pea variety Asha. Next slide. After that, if you see here, these are our efforts in which we sequenced mango, jute cultivar, wheat cultivar C306, lathyrus, wild rices, black rice of Assam, guar, and tea. And all these reports are published, and in these all genomes, we identified many agriculturally important genes and also genes related to disease resistance. Next slide. Besides that, many plant pathogen genomes were also decoded by our group, like Puccinia genome of all 3 Puccinia species, and then Magnaporthe and Fusarium oxysporum. Here, we identified gene for pathogenicity, A virulence, and different effector proteins. Next slide, please. The most important slide which I would like to show here, in 2000 for sequencing human genome, we need around $100 million US, but now in 2022 and 23, within 100 US dollar you can sequence complete human genome. Till 2000, we were having few genome sequences available in public domain. If you see in plant and animal species, there were only 2 species were available in public domain, but now more than 30,000 species genomes are available in public domain. Next slide. So utilizing genome sequence information, if we see here, after the decoding of genomes of different species, most of the disease resistance genes were decoded in Arabidopsis. Before 2002, when genome was not available in public domain, only 14 genes for disease resistance were cloned. But after 2000, more than 31 genes have already been cloned. Similarly, in rice, only 4 genes were cloned before 2002, and 24 genes were cloned after 2002. So that shows how genomic information is important for cloning and characterization of different genes. Next slide, please. And using genomic resources and genomic sequences, these are the genes which we decoded in our lab, starting from PI54, PI54RH, PI54OF, and then many promoters, and also genes related for programmed cell death, and also an avirulence genes for PI54. Next slide, please. So I would like to concentrate here more on telling you the story of cloning and characterization of PI54 genes, how we started this work. And you know, Dr. Prasad just now presented how this disease is important nationally and internationally. Next slide, please. If you see internationally, this disease is present everywhere where rice is grown, and in India, even all parts of the country, you can have this disease already presented by Dr. Prasad. And it's responsible for approximately 30% rice production losses, which can feed 60 million people every year. We estimated that for the management of rice blast, around 68 crore rupees is being spent in basmati growing areas of India. And the best management option is host resistance, for which not only you need sources of resistance, but also need gene for resistance. But by using conventional method, which is very time consuming, laborious, and amenable to linkage drag, we have to concentrate on a specific gene. Next slide, please. So our first question in 1996 was, is there any rice line resistant to rice blast in India, which was present? How many genes control that resistance? And then third question was how to discover that gene. Next slide, please. So for doing that particular experiment, we exposed 13 rice lines which were known as differential host to 119 strains of Magnoporthia fungus, which was collected from J&K, Himachal Pradesh, and Uttarakhand. And we identified a line TTIP. You can see this particular red arrow, which I showed here. It was completely resistant to 119 strains of Magnoporthia oryzae. And this is the phenotype of TTIP leaf. And to identify a gene in this particular variety, which contains 36,000 genes, you have to develop a mapping population. For doing that, you have to cross this with a susceptible line. So we crossed TTIP with HP2216 and developed a mapping population. Next slide, please. So we developed a linkage map here, and you can see from this green line here, PI54 was mapped with 2 markers, RM207 and RM224, with 0.7 and 10.7 centimorgan distance. And when we looked at the literature, there was no DNA markers available with us. So we found the sequence of rice genome from the databases and designed 40 new SSR markers in the Nippon bear genome, and used those 40 markers for genotyping of our mapping population 208 plants, and developed, next slide please, and developed a typical fine map of this particular gene here. You can see the fine map which we developed by using 40 new SSR marker. Out of those 40s, only 2 markers, that is TRS26 and TRS33, was present at 0.7 and 0.5 centimorgan distance. So in terms of centimorgan, this is a very short distance, but in terms of physical distance, it comes around 142 kilobases. So to identify one gene in 142 kilobases, again, it's a big task. To do that, we used various computational method and identified 18 genes within these 142 kilobases, and all those 18 genes were annotated, and finally we found that there was one gene which we named as a candidate gene, which was showing similarity to resistance-like genes, so we named it as a candidate genes. So starting from a complete genome of rice in TATUP, with all genetic and genomic approaches, we narrowed down to a small fragment of 1.5 kV as a candidate gene. Next slide, please. So to clone this particular gene from resistant line TATUP, we amplified this from both resistant and susceptible line. You can see P1 and P2. Again, sequenced this particular fragment, and we were happy to see that we found a typical gene structure which has open reading frame, promoter sequences, and poly A tails. When we compared TATUP sequence with the Nippon ware, which is susceptible to rice blast, in the susceptible allele of PI54, there were 3 insertions. One was in open reading frame, another was in promoter region, and third one was in poly A tail. And when this typical protein structure was designed for PI54, we found a horseshoe-shaped structure. You can see here a protein structure here, which is red in color, which is called leucine-rich repeat region, LRR. And this is the region which is very, very important to receive signal from the pathogen side. Next slide, please. So to prove the function of this particular gene, this gene was cloned in along with its native promoter in a plant transformation vector. So if you want to prove the function of a gene, you have to put this gene back in a susceptible line. For doing that, this gene was transferred in PI54 along with PI54 promoter regions and transferred to the susceptible line of rice type I309, and using all these transgenic methods, when we analyzed those plants with PCR, we found that all those plants were PCR positive, and our southern analysis tells us this, that most of these plants contain single copy insertions. When we did expression analysis of all these transgenic lines, we found that the gene is expressing similar to that of its native allele, which is present in TATUP. And our final test came when we inoculated those lines with virulent strains of Magnaporthia oryzae. And you can see that all the transgenic plants are resistant to that particular strain. The next step for validating this particular function was using RNAi approach, which we did, and we knocked it down. And we also found that the gene which we cloned was showing resistant to the strains, at least 4 strains collected from North India, East India, and also South India, which tells us that PI54 is a broad spectrum resistance. Next slide. So next question was, our question was how this single gene is controlling resistance phenotype in rice. And to do that, we performed global gene expression analysis. Next slide. And we showed here with microarray analysis where in RNA of our transgenic line was hybridized with gene chip, which contains 56,000 genes. And our analysis shows that all these red color indicates on right-hand side is that all those genes related to response to stimulus, signal transduction, transcription regulators, program cell death, etc., they are expressing in our plant, which is resistant to Magnaporthia oryzae. Next slide. And our differential gene expression analysis tells us very clearly that in transgenic lines, 107 genes exclusively upregulated, and those genes were mostly related to transcription factors, programmed cell death, and signal transduction. These genes were not only regulated or expressed, but also producing enzymes like peroxidase, polyphenol oxidase, and beta-glucanase and chitinases, which are considered as defense response-related genes, and these were present in transgenic lines. Next slide. We then developed a typical molecular model how this gene is working in a typical plant cell here. You can see here the gene is shown here. Unfortunately, this pointer is not working. But you can see this is a typical rice cell which is infected by Magnaporthia spores. And when plant infects rice cell, it releases certain proteins which are called effector molecules. These effector molecules are recognized by resistant gene PI54 at leucine repeat region, and then all these dotted lines which I showed here, it shows that it regulates the various defense response related genes within a plant cell. And then finally we concluded that PI54 regulate the regulators of defense response against rice blast pathogen. Next slide. And then we also showed in our subsequent publications that PI54 was the smallest gene cloned from literature, and it contains a typical zinc finger domain which was not present in the rest of the genes cloned from different plant species, that makes it a unique genes cloned from rice. Next level. And our proteome analysis also tells us that by eye-track based proteome revealed that PI54 regulates secondary metabolism, hormone signaling, and pathogens-related proteins. And that's how you can say that this particular gene is working within a rice cell. Next slide. So next question was, since gene we have cloned here is a broad spectrum genes, then our question was how its alleles are working in land races and wild species of rice. Next slide. We collected 92 land races from different parts of India and then looked at various features of this particular alleles, particularly nucleotide binding sites and leucine repeat region, and we developed even allele-specific markers based on two conserved region of 163 base pair and 144 base pair, and developed allele-specific DNA markers. And then finally, with this analysis, we concluded that PI54 allele in rice land races may offer broad spectrum and durable resistance, making them invaluable for breeding programs. Next slide, please. To mine its orthologue in different wild species, rice has 24 wild species, and one of them is Oryza rhizomatis. And you can see here, this is a work of one PhD student. He mined this particular allele, Oryza rhizomatis, and then compared this particular allele with Titop alleles and Nippon bear alleles, and finally validated its function. And we concluded in our F figure here that the allele orthologue which we cloned from Oryza rhizomatis, providing resistance to more than 6 strains of Magnaporthia oryzae. The paper was published, and also we got patent on this particular orthologues. Next slide. The another orthologues which we cloned is PI54OF from another Oryza officinalis, a wild species which was cloned by Dr. Devanna, who is student of this university and now working at senior scientist at CRRI Cuttack, and also sitting here in the audiences. He made a very typical analysis of this orthologue cloned from Oryza officinalis compared with all the alleles which we cloned from Oryza rhizomatis as well as Titop and susceptible lines, and then filed a patent on this particular orthologues. Next slide, please. And showed its function in both Indica and Japonica type of rice, and very clearly we showed that PIOF is providing high level of resistance to multiple strains of Magnaporthia oryzae. Next slide. Now, next question was, if this particular allele and orthologues which we cloned from Titop, Oryza rhizomatis, and Oryza officinalis, where these are expressing in a protein gene, either it is present in DNA, in the nucleus, or in the cytoplasm, or cell membrane. So where it is present, therefore doing that analysis, one of my PhD students, she did this analysis and found that the and showed very typical localization of PI54 and PI54RH. These are localized in the host cell membrane, whereas PI54OF is localized in the cytoplasm, and we showed that this diversity is a boon for breeding program because of their multiple expression in different compartments of the cells. Next slide, please. Now, when we cloned this particular gene, the next step was how to utilize this gene. And for doing that, introgression of PI54 in popular rice varieties was started in 2008, and we looked at their performance in different agronomical backgrounds and different zones of the country. Next slide. And what we did here, you can see here, but what is the material we provided to the breeders? We were providing the gene, we provided gene-linked markers, TRS26 and TRS33, as well as gene-based markers. Not only that, also donor important lines, TTUP as well as a TTUP-derived derivatives, recombinant inbred line 4, which we provided to the breeders in 2008 in a network program. Next slide, please. And this was the strategy which we adopted here. You can see an example of Pusa basmati 1, which is highly susceptible to Magnaporthia oryzae, and the TTUP phenotype, which was crossed with Pusa basmati, and then the subsequent segregating populations were monitored by using gene-linked and gene-based markers. And wherever this gene was present, you can see the band was present, and also we also did phenotyping at Indian Institute of Rice Research as well as at other places to look at the segregating lines and their phenotypes to Magnaporthia oryzae. Next slide. And those lines were evaluated in different parts of the country. This is one of the examples of Karnal, Pusa, Bihar, and then Panipat and IRI field, and this is the monitoring team which are evaluating those lines which contains PI54. Next slide. And also similar to that, this is an example of Pusa Samba 1850. This is a variety which was released by IRI group, and then Pusa Basmati 1885 again contains PI54 along with other genes, and then HPR 2880, which was evaluated at Palampur and released in Himachal Pradesh, and then Shalimar rice 6 containing PI54 was released by our colleague from Sher-e Kashmir University at Kashmir. Next one. And similarly, our colleagues, Dr. Sundaram's group, this is an example of DRR Than 62, which contain PI54 along with other genes, and also at Tamil Nadu, CO51, which contain PI54, and the phenotype of this particular line. Next slide. So when we looked at literature this year, and this hard work was done by Dr. Devanna, we found that this gene was transferred in more than 40 improved varieties of rice and 22 advanced breeding lines. And this is the pan distribution of PI54 in India. Wherever rice is grown, all those 40 varieties are listed on that side which contain PI54. And these are the developer institutions, including IRI and also Indian Institute of Rice Research, particularly Dr. Sundaram's group. And also one report from Dharwad, where they transferred this particular variety, this gene in N10 variety. Dr. Prashanti, she did this work. And all these colleagues now made this particular gene, which is present in almost all lines grown in the country. This is the example only from public institutions, and I'm sure that many private institutions, they have transferred this particular gene in their varieties. Next slide, please. So finally, to conclude this particular presentation here, these are the milestones of PI54 discovery here. We started in 1996 on characterization of Magnaporthia populations, identified TTUP in 2002, then a gene was mapped in 2005, and then again cloned in 2005. In 2008, this mass program was started. In 2011, this gene was functionally validated. In 2012, a molecular model was developed, and also in 2012 we cloned its ortholog from Rhizomatis. In 2014, another ortholog was cloned from Officinalis. In 2015, alleles were mined from 96 rice lines. In 2016, AVR PF54 was cloned, and in 2025, if you see here, the gene is transferred now in more than 40 rice lines. And as I told you, the PI54 associated studies gave us more than 70 publications, 5 patents, 3 products in the form of varieties, and more than 22 PhD students were trained on this particular gene, and it helped in developing human resource in molecular biology in the country. Next slide, please. So to conclude my presentation here, we sequenced genomes of rice, tomato, pigeon pea, wheat, jute, mango, guar, black, Lathyrus, and many plant pathogens, and this sequence is available in public domain. We also cloned PI54 blast resistant genes and its orthologs from wild species, developed gene-based markers, and now this new gene or novel gene which we cloned, PI54, has already been transferred in more than 40 varieties of rice, creating a silent revolution in rice breeding. Next slide. These are my all students at that time when we completed, and these 22 students which are listed here, they are exclusively worked on PI54 gene, and I will take only 1 minute to explain here because most of the students are working here, but where these students are present here. The Prem was the first MSc student who developed first cross in 1999 in my lab, followed by Vinay, who did RFLP analysis for mapping of this gene, who is now principal scientist at Indian Institute of Wheat and Varley Research Institute. And then Madhav cloned this particular gene in 2005 in my lab. He is director of Tobacco Research Institute, Rajamundry. Satyapal did functional validation of this particular gene, who is now in Indian Revenue Services and working in Japan. Santosh did this microarray analysis and transcriptome analysis, and he is senior scientist at Indian Institute of Millet Research. And then Archana did allele mining, and she is principal scientist at BARC, Bombay. Alok cloned orthologus of this particular gene from Oryza rhizomatis, who is senior scientist at Indian Institute of Pulses Research. Donald worked on functional validation of OF, and he then shifted to Indian Institute of Science, Bangalore, for doing his PhD. Amit was involved in doing functional validation of PI54, who is running his own company in Bihar. Sangeeta was involved in decoding the genome of this rice blast for identifying defense response gene and resistance gene. Shoham cloned this AVR PI54 gene, who is scientist at Indian Agriculture Research Institute, now New Delhi. Shalu did allele mining, she doing postdoctorate from US. Pankaj did allele mining of AVR gene, who is now associate professor. Devanna did allele mining of orthologous genes from Oryza officinalis, who is senior scientist at Indian Institute of CRRI, Cuttack. Joshita did promoter cloning, and she is senior scientist at IRI, New Delhi. Kirti was involved in doing work on RNAi, and she is now a scientist in forensic lab in New Delhi. Mandeep did proteome analysis, and she is now postdoctorate in US. Priyanka was involved in transcriptome analysis of PI54. She is now associate professor at MIT University. Jyoti did expression analysis again in PI54, and she is doing postdoctorate from Israel. And then Sarkar was doing computational analysis of different alleles. He is a professor at Central University of Assam. Vanita did again this expression analysis of PI54. She is now associate professor in Himachal Pradesh. And Ritu cloned pathogen-inducible promoter. She is doing postdoctorate in US. And this is how we collaborated with scientists from IRI, HPKBB Palampur, and also CRRI Cuttack, and also Dr. M.S. Prasad from Hyderabad. And most of the funding from this whole work came from ICAR, DVT, World Bank Project, and also Department of Science and Technology, Government of India. So to explain this to the student, those who are sitting here, if you invest your mind and also do more critical science, I think there are a lot of opportunities in science also and basic sciences also, wherein you can do a lot of work and make your career in sciences. So thank you very much, and we can have some questions if you wish.
Thank you, Dr. T.R. Sharma, for amazing journey of how field lab discovery can take it to the field transformation and bringing a lot of impact in the rice field. Thank you. Nice presentation. I think we have one question. We have time left for only one question.
There is no question. Thank you. So you can send your question to me by email if you wish.
So now we'll break for the tea. I'll request for announcement.
Now we shall break for a very brief of 10 minutes tea break. Very short break of 10 minutes to have a tea and assemble back in the same hall for the next presentation from Dr. R.M. Sundaram, sir.
ಹರೀಶ್ ತೆಗಿ. ಇದೆ ಸರ್. ಸರ್ ಈಗ ಬೇರೆ ಹಾಕೇನು ಸರ್ ವರ್ಕ್ ಆಗ್ತದೆ ಸರ್. ಅದು ವರ್ಕ್ ಆಗಲಿಲ್ಲ ಸರ್. ಬೇರೆ ಹಾಕಿದ್ರೆ ವರ್ಕ್ ಆಗ್ತದೆ. ಅದೊಂದು ಪಾಯಿಂಟ್ ಮುಂದೆ ಸರ್ಸಣ ಬೇಕಾದ್ರೆ ಬಿಡ್ರಿ ಸರ್. ಹಾಂ
ಸ್ವಲ್ಪ ನೋಡೋಕೆ ಅನುಕೂಲ ಆಗ್ತದೆ. ಇಲ್ಲ ಅಂದ್ರೆ
ಹಾಂ ಇಷ್ಟೇ ಸಾಕು ತೋರಿ ಸರ್, ಇಷ್ಟೇ ಸಾಕು. ಈಗ ನೋಡಿ ಸರ್ ವರ್ಕ್ ಆಗ್ತದೆ ಸರ್. ಸರ್ ಬ್ಯಾಡ್ ತೋರಿ ಸರ್, ಈಗ ವರ್ಕ್ ಆಗ್ತದೆ ಸುಳ್ಳೆ ಈಗ. ಯಾಕಂದ್ರೆ ಸರ್ ಇದೊಂದು ವರ್ಕಿಂಗ್ ಇಲ್ಲ ಸರ್. ಇದ್ರಾಗ ಈಗ ಮನೆಯ ರಿಮೋಟ್ ಅದಕ್ಕೆ ಯೂಸ್ ಮಾಡ್ಕೊಂಬಿಟ್ರಿ ಸರ್. ಇಂಥದ್ದೊಂದು ತರಿಸಿ ಇಟ್ಕೊಡಿ ಸರ್. ನಾಳೆ ಯಾಕೆ ಕೊಟ್ಟಿದ್ದು ನಾನು ಇದು ಕೊಟ್ಟರೆ ಸರ್, ಎರಡು ಮಿಕ್ಸ್ ಅದು ವರ್ಕಿಂಗ್ ಇದೆ ಸರ್.
ರೈಸ್ ಕೋಆರ್ಡಿನೇಟರ್ ನಮಗೆ
ಈಗ ವರ್ಕ್ ಆಗಿದೆ ಸರ್.
ವರ್ಕ್ ಆಗೈತಿಲ್ಲ ಇಲ್ವಾ?
ಶಾಲ್ ಚೇಂಜ್ ಮಾಡಿದ್ವಿ ಸರ್.
ಸರ್ ವಾಟ್ಸ್ ಪ್ರಾಬ್ಲಮ್ ಆಗೈತಿಲ್ಲ?
ಹೌದು
ಸೆಲ್ ಪ್ರಾಬ್ಲಮ್.
ಶಾಲ್ ಸರ್,
ನಾಟ್ ಕಂಪ್ಯಾಟಿಬಿಲಿಟಿ ಪ್ರಾಬ್ಲಮ್ ಅಲ್ಲ.
ನಮ್ದು ಏನು ಪ್ರಾಬ್ಲಮ್ ಇಲ್ಲ ಸರ್.
ಅಲ್ಲ ಅವ್ರು ನೋಡ್ರಿ ಈ ಕಡೆ ಅಲ್ಲ, ಈ ಕಡೆ ಮಾಡಿದ್ರೂ ಆಗುತ್ತೆ, ಎಲ್ಲಿ ಮಾಡಿದ್ರೂ ಆಗುತ್ತಲ್ಲ.
ಆಗತ್ತೆ ಸರ್. ಶಾಲ್ ಡೌನ್ ಇತ್ತು.
ಇದು ಪಾಯಿಂಟರ್, ಇದೇನಿದೆ
ಪಾಯಿಂಟರ್ ಸರ್. ಪಾಯಿಂಟರ್ ಯಾಕೆ ವರ್ಕ್ ಆಗ್ತಾ ಇಲ್ಲ? ಪಾಯಿಂಟರ್ ವರ್ಕ್
ಆಗ್ತಿದೆ.
ಹೌದಾ ಸರ್? ಅಲ್ಲಿ ಕಾಣಂಗೇ ಇಲ್ಲ ಸರ್.
ಅಲ್ಲಿ ಕಾಣಿರೋದು
ಅದಕ್ಕೆ ಅವ್ರು
ಇಲ್ಲಿ ನೋಡ್ರಿ, ಇಲ್ಲಿ ನೋಡ್ರಿ ಮೈಕು.
ಹಾಂ
ಬಟ್ ಅಲ್ಲಿ ಬರ್ತಾ ಇಲ್ಲ, ಅದು ಅಲ್ಲಿ ಬರಲ್ಲ. ಇಲ್ಲಿ ಬಿಡು. ಬಟ್ ದಿಸ್ ಇಸ್ ಆಪ್ರೇಟಿಂಗ್ ಅಲ್ಲ?
ಆಪ್ರೇಟಿಂಗ್ ಆದ ಸರ್. ಇದು ಆಪ್ರೇಟಿಂಗ್ ಆದ ಸರ್. ಹೊಸದು ಕೊಟ್ಟಿದ್ರಿ ಅದನ್ನು ಹಾಕೇನು ಸರ. ಇದೊಂದು ಇಟ್ಕೊಂಡಿರ್ತೇನೆ ಸರ್. ಸರ್ ಇನ್ನೊಂದು ಸ್ವಲ್ಪ ಮುಂದೆ ಮಾಡೋದಿಲ್ಲ ಬ್ಯಾಬ್ರಿ ಸರ್. ಸಾಕು ಬಿಡ್ರಿ ಸರ್. ಬರೀ ಅವನೇ ಹೇಳ್ತಾನೆ ಫೋಟೋ ಬರಲ್ಲ ರೀ, ಹಂಗೆ ಆಗಲ್ರಿ, ಹಿಂಗೆ ಆಗಲ್ರಿ.
Welcome back. Before we move to the presentations, we have an announcement here. There is a special interaction session for the benefit of students. At 6 o'clock, we have an event of interaction between the industry personnel and the students at 6 o'clock. I request all the postgraduate students of the university to kindly be present in the hall at 6 o'clock and actively involved in this interaction with the private sector and industry people present here. And also I request industry representatives, please make yourself available for the interaction at 6 o'clock in the evening. One more request from the organizing committee that all the registered participants should please carry the food coupons while you go for the lunch and dinner. Kindly carry your food coupons to the guest house to have a lunch and dinner.
Okay, welcome back. Now we have very important presentation here by Dr. R.M. Sundaram. I invite Dr. Sundaram, who is Director of Indian Institute of Rice Research, Hyderabad, and also I think one of the first developer of marker-assisted derived line using these for access series genes, and did a lot of work on marker-assisted selection and gene mapping in rice for the past more than 25 years. So Dr. Sundaram, please.
Thank you. Honorable Vice Chancellor of this university, Dr. Patil, respected Chairman and Co-Chairman, and my dear colleagues, I'm going to talk about advances in molecular breeding and tools for multiple pest and disease resistance in rice. What I'm going to narrate is my last 20-year experience, which I've gathered for the last 28 years. And it's not something like a typical Bhagavad Gita or a sermon I would like to give. I would just like to share where we have succeeded, what we have learned in the last, say, 28 years in terms of breeding for multiple biotic stress resistance, and where we have failed, and how we sort of did a mid-course correction. So I'll be dividing my presentation into 5 parts. The first part, I'll give a brief introduction about the scenario of rice, which my colleagues have already explained. I'll also be talking about why biotic stresses are very, very important in the context of stabilizing rice production. Then I'll be talking about one of the most important steps in terms of deploying genes or identifying genes, that is molecular mapping. I'll talk only with a few case examples and talk about an important upcoming tool called association mapping. And I'll then be in the third part of my presentation talking about molecular breeding, what are all the various strategies, including backcross breeding, pedigree breeding, and also some of the new tools and techniques will be the fourth part of my presentation. In last 2 slides, I'll be summarizing what I have learned in the last, say, 25, 28 years in this particular field. So as you are all aware, we are #1 in terms of not only rice production, and we are slowly but steadily increasing our productivity also. And rice is very, very important. And we are also doing extremely well, not only in export of basmati rice, even in non-basmati rice also. So the scenario with regard to rice is looking very green, but it's not so green Because if you look into what we require by, say, 2050 or 2047, when our Honorable Prime Minister is expecting our country to become a developed country or Viksit Bharat, that's where the major problem is. Presently, rice is grown in about 47 to 50 million hectares, but by 2047 or 2050, the total rice area in which rice is grown is expected to decrease to about 35 to 37 or maximum 40 million hectares. We are going to lose outrightly around 7 to 10 million hectares of rice growing area. And there are other associated problems like biotic stresses, rapidly changing climate and all. So we need to work. And while breeders will be developing good varieties with higher yield, with better quality, we also need to protect the yield by protecting the varieties and hybrids against biotic stresses. So if you look into it, any survey says that in any given season, biotic stresses can reduce yields of rice up to about 30%. And what are all the biotic stresses which are of significant or important in rice? As far as insect pests goes, they are stem borer, leaf folder, gall midge, and then brown plant hopper. As far as diseases are concerned, we have blast complex, then bacterial blight, sheath blight, fall smut, and a few other minor diseases which have assumed major disease proportion. There is a reason why I have circled problems like stem borer, leaf folder, and also fall smut. This is principally because there is no known strong host plant resistance. So resistance breeding has not progressed really well, but there is some silver lining. My colleagues have done some seminal work in terms of identifying candidate genes or candidate loci associated with stem borer and fall smut. I'll narrate when I'll be talking about them. And finally, with regard to resistance gene breeding strategies, Dr. T.R. Sharma ji has very nicely, eloquently expressed it. While genes like PA54, XA21 are very useful, but it will always be better if they are deployed in combination, say PA54 with PA2 or PA9, XA21 with XA13 or this thing. Pyramiding or stacking multiple resistant genes is the way for us to move forward. And this is particularly accentuated because of a rapidly changing climate, wherein what we are seeing is we are seeing a transition from many of the minor diseases or pests to major pests or diseases. This is particularly true. This is a slide given by my colleague Dr. Padma Kumari. When Green Revolution started in our country, we had only 3 major insect pests. But as of now, we have got more than 20. The numbers are increasing steadily. And this is also true with disease scenario, which was ably illustrated by Dr. M.S. Prasad. When our country became independent, we had only one major disease problem, that is brown spot of rice. But as on today, it's 12, and the number is growing steadily. And one of the main reasons for this is that the climate change related vagaries like increasing temperature, shifting rainfalls, and shifting humidity patterns are actually accelerating or accentuating this particular problem in terms of making the pathogen or pest strains more virulent or more infective. And now coming to the second part of my presentation, I would just briefly give a glimpse about how we are doing molecular mapping, or how we are, before identifying and then deploying the genes or QTLs, one of the first and foremost tasks is to identify the genomic regions or the genetic loci associated with resistance, and to fine map them, and if possible, do a gene characterization, which was very ably illustrated by Dr. T.R. Sharma with respect to PR54 gene. And I'll be talking about how we work towards this particular task. So if you look into the flow with regard to development of resistant varieties, it starts with identification of resistant genes or QTLs in populations or in the land races or varieties, then identifying molecular mapping or marker development, then deploying those markers in marker-assisted selection, and then finally using crossbreeding or back crossbreeding to transfer them into different varieties. And finally, we have a multiple biotic stress resistant cultivar. So in this, I would like to acknowledge a major role played by All India Coordinated Research Project on Rice, which every year identifies not only very strong or very a high level of resistance with regard to the major pests and diseases. And we also have identified several multiple biotic stress tolerant donors also. And with regard to molecular mapping, if I would like to, since there are many students are here, I would like to spend a few minutes in this particular slide. Whenever you undertake molecular mapping, the first and foremost task is in terms of identifying a good source of resistance. Preferably it should be monogenic, and you need to identify a variety which is susceptible to that particular pest or disease. Then you develop a mapping population. The most commonly used mapping populations are F2, F3, advanced up to F5 or F6 generation, by which time they will become both, they will be heterogeneous, but all lines will be homozygous. Individual plants in a line will be homozygous. But there are alternative rules wherein we can develop backcross inbred lines or near isogenic lines. Once you have a mapping population, a stabilized mapping population is always good. When I say stabilized mapping population, I'm talking about generations like beyond F5, like F6 or F7. And when it is backcross derived line, it should be at least BC1 or BC2, F4 or F5 generation, wherein the trait will not segregate any further. So once we have a stable generation, we have to go for large-scale phenotyping, high-throughput phenotyping, wherein each and every plant should be, every line should be phenotyped at least for 2 continuous seasons. And once phenotyping is done, we have got a lot of options in terms of genotyping. And I would not like to go into what all the different markers or steps used there. But at present, the most preferred marker systems in rice for genotyping are SNP markers. Different platforms are available in rice. If anybody is interested, you can write to me. I'll explain you a detail about which are all the genotyping platforms available for genotyping in rice, particularly SNP markers. And then once you have a marker system used for analysis of this fixed mapping population, then you develop a linkage map using those markers. Even though those markers' physical positions will be known already, but still this is the first and foremost task which you have to do it. Then finally you have to go for QTL or gene identification and undertaking course mapping, and then with a subset of that particular mapping population, you can do intercrossing and then go for fine mapping. And even though it's not essential to identify the exact location of the gene and identifying what is the causative gene underlying that particular trait, it will be desirable. That's why most of the cases what I'll be going to describe as of talking about molecular mapping, we didn't just stop at coarse mapping or fine mapping. We went up to the gene level, not only in terms of fine mapping them, but also characterizing them at functional level through genomic, transgenic, and also transcriptomic approaches. And finally, when you have a closely linked marker, in the case of, even if you don't have a closely linked marker, you can always have 2 markers flanking the gene, which can be used together. And if you have a marker which is part of the gene, these markers are functional markers. An example was presented by Dr. T.R. Sharma ji for PA54 gene. And once you have a good marker which is very closely linked to the gene, or 2 markers which are flanking the gene, or marker which is part of the gene, which are called as functional markers, we can go for large-scale marker-assisted selection using various tools or techniques. And how we are going to develop different methodology to transfer the genes, I'll be discussing later. Now coming to some of the own success stories with regard to bacterial blight resistance, my team together with Dr. Gaurishakar Laha and with the help of my pathology team, they have done very good work in terms of identifying. This is one of the genes which has been used as a flagship gene in many of the resistance breeding program. It's a gene called XA33. It was course mapped using SSR markers on chromosome 7 of rice. But recently, one of my PhD students, he has done the fine mapping, and we have identified the underlying candidate gene also. But since we were dependent on low throughput markers like SSRs, ISSRs, and then RFLPs in initial stages, the progress was very, very long. It took almost 6 years for us to undertake molecular mapping, gene identification, and then gene characterization. But The scenario with regard to this has changed with the advent of genomic tools, which are very well described by Dr. T.R. Sharma-ji. So now that we have got very good SNP markers and also high throughput genomic tools for candidate gene characterization, once my student, PhD student Pragya, she identified that one of the lines provided by Oryza officinalis introgression line, it has got strong level of resistance against bacterial blight. She went ahead and did genotyping with 7K rice SNP markers available, and she was able to identify a major locus on chromosome 11 long arm, which is about 3 kb away from exate 21. And she has also worked to identify the candidate gene also. We were lucky because the underlying SNP was part of the gene, and we were then able to, through sequencing and also through transcriptomics, and then later through transgenesis and gene silencing, we were able to identify that the candidate gene is a gene encoding NBRC domain containing protein. This is another success story wherein my colleague Dr. Seshu Madhav, together with Dr. Srinivas Prasad sir's team, they have worked towards identification of major neck blast resistant gene. It also confers resistance to leaf blast also. This was identified from a wild species called as Oryza glumipetala. The introgression lines were developed by PAU, Ludhiana. So he worked initially with SSR markers and later with the high throughput tools like SNP markers, genotyping by sequencing and other advanced tools which are available. He was able to identify the candidate gene, and this gene has been licensed to at least 3 or 4 companies for deployment in their breeding programs. And this is the power of mapping. So right from a day in which mapping population was developed, within about 1.5 years, we can easily undertake or identify the candidate gene and validate it also. And this is one recent example. I mentioned that sheath blight and false smut are gray areas, and they are not having very strong level of resistance. Of course, there are a few genotypes like TETUP and all, but the level of resistance is towards the moderate resistance side. So we have identified a northeastern landrace by my colleague Dr. Vijaypal Badana, who earlier worked at Nagaland, Manipur and all. So he brought this particular line. We developed a mapping population by crossing it with improved Samba Masuri. So we had a set of 430 RILs. So using that with SNP markers, with the help of NAPGR, Dr. Gopalji Jha's group, we were able to do a fine mapping using SNP markers. And we were also able to identify diagnostic SNPs, and we are now identified 3 putative candidate genes. We are now working towards that validation. And this work, I would say, that will get completed within a time period of 1 year. This is the power of genomics. This is the power of deploying advanced tools in molecular mapping. Then this is another typical case wherein my colleague Dr. Jyoti Badri, she works on population improvement with regard to sheath blight. So from a northeastern landrace called Fogak, which shows strong level of tolerance to sheath blight, she has identified a major locus called QSBR 3.1 on chromosome 3, and she is working towards identification of the candidate gene. And we have been successful to some extent because 2 genes show hyperexpression after inoculation with the pathogen. And similar is the story with regard to another gene conferring tolerance to BPH called BPH33. The original donor is from landrace called Velithachira. From that breeding line called RP2068 was earlier developed. We identified that this particular line shows strong level of resistance against BPH. So we developed a mapping population. This program was led by Dr. Bentur. So together with his team, my team has initially done the course mapping, then fine mapping with SNP markers, and we have identified that the candidate gene controlling this particular, responsible for this particular trait is a heat shock protein. And now we have developed candidate gene specific markers, and we are in final stage of varietal development with this particular new gene. And one important power of like say, we have been talking about biparental cross-derived mapping populations. Other popular methodology is called as association mapping, wherein historical populations or crossbred developed populations larger in size, around 100 or thousands. Generally, they are used for trait mapping, phenotyping, and then validation. Through association mapping, my colleagues Dr. Kalyani and Dr. Somashekar, they have indeed identified some major loci located on chromosome 1, 2, 4, and 8 associated with rice root knot nematode, which is becoming a serious problem, particularly in upland rice and also direct seeded rice. This was done using our eCrypt network, wherein we have started a coordinated testing program even for nematodes also. With the support of our cooperators, we were able to achieve this. And right now we are working towards that major locus located on chromosome 4, short arm, and we are working towards identification of the candidate gene with a biparental cross-derived mapping population with 2 donors. Then this another, I would say, a success of coordinated research. In coordinated research, under the guidance of Dr. T.R. Sharma ji, starting with the year 2023, we have been fingerprinting each and every entry nominated for our eCrypt trials, be it IHRT or IVT or AVT1, AVT2. Every year we fingerprint around 1,200 to 1,500 entries using 1K rice SNP platform. It has got representation of around 1,084 SNP markers. One lucky thing what we got out of it is I requested Padma Kumari madam to analyze the data with regard to stem borer tolerance, because we have a stem borer screening nursery carried out every year wherein all these 1,300 lines are subjected for this thing. Then she carefully selected some certain individuals and then developed a mapping population which doesn't represent a structure. And my colleague Dr. Kalyani, who is a genomics expert, could do an association mapping using this 1K RICA. And we were surprised to find very good associations, particularly on chromosome 5, wherein we got one single marker which has got a large peak. And as far as biparental cross-derived populations are very powerful in terms of trait detection and characterization, I would say that future lies also in association mapping. And we are going to use our All India Coordinated Research Project on rice data significantly for association mapping. And I would like to stop here with regard to, I've only given you a glimpse of molecular mapping because of paucity of time. If any student or anybody is interested, he can always write to me, and I'll be happy to assist you in various stages in which you require some help. Now let's talk about how whatever markers or whatever major genes for which molecular markers are available, how they can be translated by developing better resistant varieties or hybrids. So in this regard, there are several breeding strategies. One breeding strategy which has been very, very popularly deployed for resistance gene breeding is backcross breeding, wherein generally in the background of a very elite variety, which not only has got a higher yield but also a good grain and cooking quality, we try to transfer a few genes, maybe 3, 4, or 5. They may be encoding a single trait, or they could be encoding multiple traits. So backcross breeding or marker-resisted gene pyramiding is one of the most popular strategy. But of late, additional tools like genome-wide association study, wherein you don't look at single genes, which are either major or minor QTLs. So there are certain traits like, for example, sheath blight. We identified that even though there are some major QTLs are there, but even in Vazopec and Fogak, they are controlled by several minor loci also. So genome-wide association study will help you to identify the minor QTLs and then help us to transfer those QTLs into the background of our varieties. Then we have genome editing. I'll talk only about one successful example wherein genome editing has been deployed for resistant gene breeding. And I'll also briefly be touching populations like magic. And also, we also need to understand that conventional breeding is still used for resistant breeding deployment, but there are various challenges, and that's why molecular breeding is the right way for us to go ahead forward. As far as bacterial leaf blight resistance is concerned, based on our intensive analysis of our eCrypt data, wherein we have got extensive phenotyping information about various NALs containing different genes. For bacterial blight resistance, it is my understanding that a 2-gene or 3-gene combination from the genes listed here, XA13, XA5, XA21, XA33, XA38, and XA48 should give durable resistance. With regard to BLAST, already Dr. T.R. Sharma has very nicely illustrated. Gene combination like PA1 plus PA2, PA2 plus PA54, PA9 plus PA54, PA68 plus PA54, all the, in whichever combination PA54 is there, it gives good level of resistance. This I can say with confidence because based on our fingerprinting data for all our equip entries, more than 90% of lines which have been identified to show good level of blast resistance have got the resistant allele of PA54. And with regard to brown plant topper, even though we have got a few genes which have been identified to work very well in the country, but we don't have very good markers for them. We have got only SSRs which you can deploy. So we are deploying for, as a start, BPH33 in our breeding program. And similarly for Galmich, we have got genes like GM3, GM4 and GM8, of which we are deploying the two dominant genes, GM4 and GM8 in pyramid conditions. And we now have some success also with regard to breeding for Galmich resistance. And in all these things, our target is variety which is resilient not only to a pest or disease, but also to multiple stresses. That's our end target of this particular program. Now let me narrate a few important things about how we developed an improved version of Samba Masuri, which has got durable bacterial blight resistance. And even as on today, the area under this variety is more than 9 lakh hectares. And there are farmers who have been growing this at least for the last almost 12 or 13 generations. And if you look into why this program started, this was a time when I joined IARR. There are a group of farmers who are from Rayalaseema area, that is Nandyal area, and also from East Godavari. They grow Samba Masuri regularly. And they expressed, in 1998 it happened when I joined IARR, they expressed to my head of department, Dr. N.P. Sarmaji, that we are facing severe problem of a disease called bacterial blight. Then in collaboration with Dr. Ramesh Sonti's group, who have been a pioneer in this particular regard, we together drafted a NATP project of ICR, and then we worked to deploy backcross breeding for development of an improved version of Samba Masuri. And in this, our target was that we want to develop a version of Samba Masuri which looks like Samba Masuri, which yields like Samba Masuri, and which cooks like Samba Masuri. And it should also have the resistance. We should not lose anything from the background with regard to Samba Masuri. So at that point of time, we were not very sure how many backcross generations we had to go. We went up to BC4 generation, and we deployed markers not only for selection of the target genes called as foreground selection markers. We also deployed a set of SSRs which could distinguish between Samba Masuri and the donor parent. 2 minutes. And Samba Masuri with regard to the presence or absence of the genes. Through this effort, we could develop one of the first products. Of course, there is another product parallelly developed called Improved Pusa Basmati 1. Improved Samba Masuri has got 3 major resistant genes, EXAVE 21, 5, and 13. And this is a success story of not only of deploying the right kind of rice science for improvement, but also about interinstitutional collaboration. Our collaboration with CCMV is continuing for the last almost 25 years or 28 years. And this variety is doing really well. And what is important for you to understand is that under field conditions, it is even as on today, it is standing very, very tall in terms of resistance. As you can see here, on the right side, you have got panel which has got improved Samba Masuri. It is totally free from the disease. Already grain maturity has started. On the left side, Samba Masuri is there, wherein it's severely affected by bacterial blight disease. And this has already been described by my colleague Dr. Srinivas Prasad, wherein the background of improved Samba Masuri, another gene called EXATE38 was added. And this also Dr. Srinivas Prasad has already narrated. And another interesting development is a recent release called as DRR-85, which combines bacterial blight resistance with Galmich resistance. It showed immune level of reaction in several Galmich hotspots along with bacterial blight resistance. And we have added a trait of salinity tolerance in the background of improved Samba Masuri and developed DRR-58. Then another important development is adding low soil phosphorus tolerance, which can reduce phosphorus fertilizers. It is doing really well in Gangavati area of Karnataka. And finally, one important version is development of bacterial blight blast and then submergence tolerance version. This was recently released, wherein Dr. Fiaz has taken a lead role in this development along with Dr. Prasad. And we also have a success story wherein we transferred QTLs associated with high zinc and developed a version of improved Samba Masuri called DRR-48. And finally, this is a flagship product in which it has got multiple traits. It has got bacterial blight resistance controlled by 21, 13, and 5, blast resistance controlled by PA54, and it also has got yield enhancing genes, SCM2 and then OSSPL14. This is a collaborative work wherein we got the donor lines from IRRI, and this variety is doing really well. In fact, this Kharif season, we had a Dharna Bay farmers who wanted more quantity of seeds of this variety, and it's spreading like a wildfire in Samba Masuri growing areas. And this is what I would like to tell, that through proper planning and through proper deployment, we could add value to Samba Masuri by initially developing bacterial blight resistant versions, then blast resistant versions, then BPH resistant versions, garbage resistant versions, and versions which have got tolerance to abiotic stresses, and finally, versions of Samba Masuri which can give higher yield also, and with better nutritional profile. And we could do the same work in Swarna's background also, wherein along with bacterial blight and blast resistance, we could transfer some of the yield enhancing genes also. And I would like to talk about I'll take half a minute, sir, in this. Some new tools are available with regard to accelerating breeding for durable biotic stress resistance. The first and foremost tool is gene editing, wherein a collaborative group led by IRRI, along with Heinrich Heinz University in Germany, they have edited at least 3 or 4 loci. These are genes called as susceptibility genes, like XA7, XA13, and other XA genes, which are known to be recessive-resistant genes. So the promoter part of these genes are binding sites for transcription activators called TAL effectors. They could edit the promoter part of these genes, and now we have got durable resistance against multiple strains of this particular disease. But these developments are parallelly going on in India, identifying different targets. But this is one example wherein we are expecting the lines to be received in India, and then we'll be testing against different strains in the country. And another important tool is called as haplotype-based breeding. You all may be knowing that haplotype is a group of DNA markers which are closely linked or co-inherited. And we have got several favorable haplotypes and then several non-favorable or unfavorable haplotypes for several traits. For example, for yield, these are all the different genes and their favorable haplotypes. Deploying this approach, my colleague Dr. Jyoti has developed a very good variety called DRR-87 in the background of a variety called Swarna. And this particular breeding approach, we followed a recombination breeding approach with a reduced backcross breeding approach. And this version has got tolerance or resistance against multiple pests and diseases. And of course, speed breeding is something which can really accelerate our efforts in terms of rapidly developing multiple biotic stress-resistant varieties as compared to just 1 or 2 seasons in a year. With collaboration with IRRI, we have demonstrated that up to 6 seasons can be taken up. And even in wild rice also, it can accelerate the breeding process. And finally, one important component I would like to touch upon is artificial intelligence. If gainfully deployed, it can be used in terms of predictive risk modeling, AI-powered phenotyping, and it can accelerate the process of genomic selection or genomic prediction. And even in gene discovery also, it has got its own use. And speed breeding can be accelerated through the application of AI tools. And if anybody is interested, I was deeply impressed by reading this paper by Farooq et al. It gives a very good workflow regarding how we can deploy artificial intelligence in breeding process. And finally, just in a slide or two, I'd like to tell what did I gather in the last several years of my work at IRR. So if you look into it, breeding climate resilient varieties through genomics-assisted breeding should be our target. Towards this, we clearly demonstrated that interinstitutional collaboration, not only between IRR and CCMB, IRR and several other institutes, whatever I presented is because of collaboration, not only within the institutes between different disciplines, but also with other institutes. So I would like to mention here that interinstitutional collaboration is the key for success. And another important point is that molecular breeding is already started delivering products. Already about 2 million or 2.5 million hectares is under molecular breeding derived products in the country, in rice particularly. And another take-home point is that it should not target improving only 1 or 2 traits. You should always be ambitious or over-ambitious, and multi-trait packaging through genomics-assisted breeding is a way for us to forward. We have demonstrated in the background of Swarna and Samba Masuri, and CRRA has done it in several backgrounds. IRA has done it with the help of NAPB in terms of adding value to several Basmati varieties. And finally, what I would like to mention here is that genomics should be increasingly deployed, not only for gene discovery, but also for accelerated gene deployment. And what is important for our as a country is that there are several novel sources of resistance. We should work towards identifying the functional basis of resistance or tolerance, and we should work more in this direction, just like the way Dr. T.R. Sharma's group has worked. And of course, the new tools which I described, they can certainly complement the process of breeding. But finally, I would like to mention here is that we started our work like a bullock cart. We were using RAPDs, ISSRs for breeding for biotic stress resistance. When I joined, I started a program for marker-stud breeding, and we were using a few markers like SSRs, ISSRs, CAPs, and all. And now, my team is traveling in an SUV, wherein we are deploying high-throughput genomic tools, not only for gene identification, but also for gene deployment. Finally, this is my concluding slide. A breeder can ask a question, whether can he use biotechnological tools for improvement of a trait. The answer is definitely yes, but we need to remember that biotechnology is one among the several tools available for a breeder. At the end of the day, biotechnological tools like MAS, molecular breeding, genomics, can only complement and then supplement the process of breeding. It will never replace the process of breeding. And we should embrace all these modern tools in day-to-day breeding. With this, I would like to thank all my colleagues who work with me. Some of them are here also, and also my funding agencies who have been very, very extraordinarily generous in terms of funding all these efforts. Thank you, one and all. Thank you, sir.
Thank you very much, Dr. Sundaram. Very nicely presented how we can map new genes for different traits like blast, BLB, and also insect resistance, and then how to deploy. Actually, I think major impetus on molecular breeding was started from Indian Institute of Rice Research, where you are heading now. And almost all varieties which we are cultivating in India are marker-assisted derived varieties in rice. So that is all because of the leadership which you are providing there to this whole rice improvement program in the country. Since we left with very less time, but still we can have one question. So it's difficult to choose between Dr. Tikku and yourself. You can decide yourself. Okay. चलो आप पूछ लो पहले.
What are happening with the molecular breeding?
Sir, I couldn't hear you, sir. Can you keep the mic closer?
Hello. Okay. We have seen in majority of the crops, including rice, that the developments what are happening with the molecular breeding are with respect to the stable varieties. For that matter, Samba Masuri, which is a well-known variety from the almost all years. Now you suggest me, your opinion I want to know, whether we need to start a fresh breeding with like magic populations involving all these genes, or we have to go with a stable variety, means ABT advanced varieties should be taken, my opinion. ABT advanced varieties should be taken into this molecular tool introduction program.
That's one way to look at it. We always, when we started our molecular breeding program, we look at low-hanging fruits. And one easy strategy is marker-assisted backcross breeding. But believe me, in forward breeding, if we deploy the right set of markers, the right set of traits, it can transform breeding. One typical example is DRR 75. It was not developed through backcross breeding. There was only one backcross with some improved Samba Masuri, and further it involved crossing with several parents. But we have been successful because we had a large population size. We were able to identify a plant which was similar in grain quality to Samba Masuri, and we could transfer at least 4 or 5 different traits. And another example is DRR 79, wherein at least 7 or 8 traits have been added in the background. And that was both DRR 75 and DRR 79. They were not released through NAL trials. They were released through ordinary trials, wherein the lines had to be tested against the best variety and tested for 3 years. So I think while there could be a case wherein good variety, we need to make it great by adding a few traits. I think we should invest more on approaches like marker-rested forward breeding and then recurrent selection and magic population, and invest less on marker-rested backcross breeding, sir. That's the hard lesson I have learned.
Okay, from student, any question?
Yes, go ahead, sir.
So, okay.
Sir, good afternoon, sir.
Achha, yeah,
Myself, Harish, PhD scholar, sir. Sir, in GWAS, we are identifying SNPs for particular traits, sir. So now there is pan-genome approaches are available, sir, where whole set of genes are identifying, and we can do large introgression, sir. So can we use some phenotype and genotype matrix of pan-genomes for GWAS analysis as an input?
You have raised the right point. Initially, our approach in GWAS was only using the primary gene pool, that is the varieties and land races. But of late, the thinking process has moved differently, and we have started deploying even some of the wild relatives and other exotic genomes also. But you need to understand that genome-wide selection or genomic selection is a work in progress, because we have just started with our eCRIP trial data, both phenotype and genotype data. Every year around, we have got the phenotype data for at least 12 or 13 different traits every year. But it's a work in progress, and we should certainly move from the primary gene pool to a pan-genomic approach. You are right in that. But progress will come in, say, another 3 or 4 years or 5 years, when we'll have a large set of products developed through genomic selection. Thank you. Thank you. Thank you.
Thank you, Dr. Sundar, for a nice presentation. Now I would like to invite Dr. Amresh Chandel, rice breeder at Well Monsanto, Hyderabad.
Respected Chair, Co-Chair, honorable dignitaries, and all the delegates present here, thank you for this invitation. And we are all very proud of the fact that India has achieved #1 status in rice production. And all my fellow colleagues, scientists, distinguished scientists who spoke before me, have a very big role in that. And also all of you have very big role who have contributed to this success. But this conference on multiple disease resistance is a very apt moment where we know that we have a lot of potential in rice and how we can go forward together as a private sector and public sector. So good afternoon. My name is Amresh Chandel, and I work in Bayer Crop Science. I will be presenting a private sector overview of how we can work together and what are the new approaches we can take in achieving the success for rice production in India and also in Southeast Asia. So my presentation is overview here, context, then modern molecular tools, gene pyramiding strategies and regional evidence, CRISPR advances on multi-stress regulation, digital phenomics, economic and sustainability impact for farmers, and also buyers' rice journey and future outlook, then the conclusion. I know the time is running late. I will try to finish fast because we have some interesting oral presentations also. Now, what are the field realities driving the modern rice breeding? These are the key challenges we see in the field. Insect pests, we all know. Viral diseases, which are becoming very important now. Multiple pathogen pressures and rapid biotype shifts are happening. In the conventional breeding, we do have constraints like difficult to stack multiple resistant genes without introducing linkage. Long breeding cycles delay delivery of improved varieties. Phenotyping multiple stresses simultaneously is inconsistent and labor-intensive. Limited ability to track complex gene interactions using traditional tools. Now, why precision, speed, and data integration matter? because we need to combine many resistant traits cleanly and efficiently. Faster selection cycles enable quicker deployment of resilient products. Data-driven phenomics and genomics improve accuracy, confidence, and decision-making. And integrating molecular tools with digital analytics accelerates genetic gain under multi-stress environments. What are our advanced tools and platforms for rice resistance breeding? We do have molecular tools like marker-assisted selection, which all of you are very much aware. Then we have gene silencing, RNAi approach, and genome editing, CRISPR-Cas, which is a new upcoming technology. On the advanced platforms, we have speed breeding, which is changing the way we are getting genetic advance faster. Then double haploidy, another approach. And genomic-enabled trait discovery. This is how integrated pipeline looks like for multi-pest and disease resistance in rice. First is a discovery phase, then we do markers and edits, then the stack the design, rapid cycling using speed breeding, phenomics, and it goes to the field testing. Then predictive analytics are applied, and finally deployment and impact happen. This all can be done within a span of 5 years now, but previously it took 12 to 15 years. So this is how we have to integrate our multiple disease resistance in rice. And on the genome editing strategies for disease, this is the way it goes: S gene knockout, optimize the alleles, promoter and cis editing, multi-target stacks, and again validation and deployment in the markets where you want to release the product. On gene pyramiding, context and rationale in South and Southeast Asia. Why do we need gene pyramiding? Because we have major regional threats like bacterial leaf blight. It is very diverse, evolving races across India, Bangladesh, Vietnam, Philippines. Rice blast, again, a multiple magnoporte races are there. BPH has different biotypes, and emerging bacterial leaf streak. BLS is becoming very important, especially in Southeast Asia. Our production goal is to enhance durable, broad spectrum resistance without compromising the yield or grain quality. What are the breeding strategies which we employ? First is a marker-assisted pyramiding, which remains the most effective and scalable approach. There have been recent evidence where ERI, Bangladesh Rice Research Institute, and many Indian programs have effectively utilized it and released the products. Optimal pyramids include 3 to 5 resistant genes or QTLs to avoid linkage drag or yield penalty, and increasing use of newer R genes like XA23, 47, 50 in the elite backgrounds. And what is the target profile we are looking at? A high yield product under Swarna or MTU 1010 mega varieties background, suited to irrigated and rain-fed lowlands, and strong tolerance across multiple pathotypes, which is very evident from the ECRIP trials where both public and private partners give the products for testing, and we are seeing a very good products coming out of that. Gene pyramiding recommendations from my side here, based on evidence. The first recommendation is, I'm sure many of you might be following this, but this is just to reiterate that broad BLB plus blast resistance is the most validated, where XA5, 13, 21, and PI54, PI9, these combinations are proven durability, where many products have been released. The second can be on XA23, 21, 5, and PI54, or XA47, 21, and PI9 for the enhanced durability. Then the third is a multi-pest, where BLB, BPH, and blast, all three can be combined in one product. And the fourth will be advanced next generation options where we can use CRISPR strategies also. What are the practical deployment and expected outcomes? On the breeding methods, marker-assisted selection takes 2 to 4 years for introgression, where you use tight markers for major genes and validate with background recovery phenotypic screening, which you all very well aware. Outcome is 20 to 40% lower disease severity, maintained or improved yields, and stronger resilience against the evolving races. So this will help us in regenerative and climate-smart rice systems. Recommended target backgrounds are again the mega varieties, MTU 1010, IR64, Seherang, PSB series, and partners and pipelines. So we have to work together. As Dr. Sundaram told that there is a very good linkage between partnership between national programs and state programs. I would add a private partner also there. So come up with the projects where public-private partnerships can also work together in a NARS program, which will help to fasten the process. On the CRISPR, let's see what are the rapid progress and why the CRISPR matters. Because this is a precise targeted edits with no foreign DNA. Faster breeding cycles, minimal yield penalty, and ideal for South Asia and Southeast Asia. Which key traits are improved? Yield and plant architecture, higher yield, unchanged quality, D3 knockouts give 22 to 38% BLB lesion reduction, and also OS, APL, GLW7, GLW7 edits improve the grain size, nutrient flow. Grain quality and nutrition is also improved. For the BLB blast and multi-stress resistance also, CRISPR has been utilized. These are the major breakthroughs where broad spectrum BLB resistance effective against diverse Asian and African strains was demonstrated in IR64, Seherang, Sub1, and other mega varieties, and there was no major agronomical penalties. Additional disease resistance targets where this B9 mutation can enhance BLB resistance, D3 edits, and both BLB to enhance architecture benefits and multi-disease packages. For abiotic stress also, drought, UV tolerance, arsenic reduction, and other enhancements like herbicide resistance and grain appearance can be done using this technology. What are the regulatory status and application in South Asia? India is the fastest deployment. You all know, last year we released Pusa rice DST1, DRR Dhan 100, Kamala. These are the 2 gene-edited varieties. On the regulatory part, SDN1 and SDN2 edits exempt from GMO rules under India EPA Rules 7-1 and expanded field trials after 2024 regulatory relaxation and high relevance to India, BLB drought and salinity. In Southeast Asia, there is an ongoing ERI and national collaborations, testing of CRISPR suite edited BLB resistant lines in IR64 background, and commercial approvals are slow, but edits without transgene may be non-GMO depending on the national policy. Then integration with the traditional breeding, where CRISPR suite promoters are edits or complementary pyramids like XA23, 21, PI54. This enables durable resistance without relying only on the resistant genes. These are the challenges. Off-target editing reduced in newer tools. So this is reduced now. Public acceptance and IP constant. This is one of the biggest challenge. And harmonization of the regulatory framework. But we all are working together to overcome these challenges. I am sure within few years it will be overcome. These are some of the reports of pyramiding BLB, blast, and BPH in elite backgrounds. On the left side, you see marker-assisted breeding from the India. Popular cultivar Telahamsa was improved by pyramiding these XA21 and XA13 gene. And also in Tamil Nadu, TNAU, stacking of different genes was done in ASD16 and ADT43. Similarly, in China and from Bangladesh, there are some reports. These are very latest development. And multi-local validation, evidence from the trials. Telahamsa pyramided lines validated at 3 blast hotspots and 2 BLB locations. They exhibited high resistance across environments, supporting the spray reduction potential. Similarly, BRRI Dhan 63 pyramided lines, they achieved near immune scores 0 to 1 across test, and multiple lines demonstrated higher yield than the elite check, implying fewer fungicide and bactericide applications for equivalent production. Again, for ASD16 and ADT43 with blast, BLB, and sheath blight resistance. Farmer impact, yield and resilience. So here we see several lines in BRRI Dhan 63 having more than 28 to 30 gram per hill and near immune disease scores. This reduced input cost and lower the crop loss risk. And stacked packages in the elite background, both in China and India, they maintain agronomic traits, broaden the resistance spectra under the biotype shift. And again, this is a public-private workflows where we can combine resources and work together. Genomic resources for BPH. Digital phenotyping and data-driven breeding decisions are now being taken across in private sector and public sector, where digital phenotyping and data-driven breeding decisions are very important. And then it combines analytics and gives us a digital dashboard where we can take decisions faster. What are the sustainability benefits of resistant rice varieties? Outcomes. First is a fewer pesticide application, lower pesticide residues, improved safety of ecosystem, enhanced resilience of climate variability. And for the farmer, what is the value? Cost saving from reduced spray, higher and more stable yields, reduced production risk, and better access to the premium markets. Now, Bayer has a rich history of bringing disease-resistant products in the market. Bayer is the first company one of the first company which released the BLB-resistant hybrid, BLB and BPH-resistant hybrid back in 2008 with the name Arise Dhani DT. This is the hybrid for the late maturity market, and it provides protection against both BLB and BPH. Later on, Arise 6444, which is one of the best-selling hybrid till date, was also introgressed with BLB and blast genes, and it is available in the market. And over the years, you can see we have been bringing products which are climate resilient and having both biotic and abiotic stress tolerance across multiple segments and markets. Today, at Bayer, we are giving products for all the segments, all the market segments present in India and Southeast Asia, and with these resistant genes. Arise 8433DT, 8455DT, 7006, all these products are there. This is the Arise portfolio where climate resilient performance across diverse and variable environments, abiotic stress tolerance including drought, submergence, and lodging, biotic resistance package against major pest and disease like BPH, blast, and BLB. These are some of the pictures which validate our products. In the middle, you can see Arise Dhani on the right side gave 8.3 tons per hectare. And on the left side, the other cultivar gave only 1.7 tons per hectare under the infestation. This is a farmer field photograph, real time. Similarly, you can see other photo where Arise RNDT, Arise 6444 Gold crop is standing very well, but other cultivar which are not having resistance gene are affected by the disease and pest. Now, what is the economic contributions of disease-resistant rice hybrids to the regional system? Here, we can see over the past 10 years, Arise products have given a better return to the farmers compared to the other cultivars when grown in disease-prone geographies. And this is the economic value of the disease resistance in rice, which impact across the yield levels, where we see in 3 to 16% yield loss. Still, if you grow resistant products, farmers can get better returns. These are the conclusions now. The first is integrated molecular tools make multi-stress resistance practical. Digital phenomics accelerate accurate selection. And collaboration is the key to scale. Thank you for your attention. My contact details are here. If any questions, you can send it across. Yeah. Thank you.
Thank you, Dr. Chandel, for excellent presentation. And you have covered various aspects on rice, what we should do in India and globally, and what are the options available with us, and giving little bit information about your company's work. That's too in a very confidential manner. I was discussing with Dr. Saab that what are the genes which you have deployed in various cultivars, probably you don't disclose. So anyway, it's your policy, but I think students or anybody can ask question. Dr. Saab, Dr. Krishnan Kumar.
I was listening to all the presentations on rice. We have a tremendous post-harvest loss after rice is harvested. And most of the time, our products or our rice exports, whether it is in broken rice or it is in the other formats, often get, I think some effort now slowly should start with respect to the breeding those genotypes which can also confer post-harvest resistance in whatever is because we almost take it to the grain, and it's a very sad story to see that if our product is rejected both within and outside. I don't think, in my view, that kind of an approach has been very intensely done. But since you said it's a institutional collaboration, I see that Bayer, if they can join hands with others and also see if we can take it forward, it will be very, very great advantage to our country, since we are also one of the largest producers and exporters.
Thank you, sir. Thank you very much for your comments.
Yeah, thank you, sir. This is very well commented, and this is need of the hour. But I would also like to say that if we harvest the grain at right moisture, we can still reduce all these post-harvest losses. But we have to work together on these technologies. Yeah. Thank you.
Okay, thank you. Any other question? Okay, Dr. Chandel, there is no question. Thank you very much for your excellent presentation. So now we have 3 oral presentations, and each presentation will be for 10 minutes. I will request my co-chair, Dr. Gowda, to kindly invite all the presenters, and we can listen to them. Dr. Gowda.
Yeah, thank you, sir. Now, first, I would like to invite Ashita Joy to talk on decode computational and metabolic insights into anti-no-bacterial mediated management for rice PLB. Thank you. You have 10 minutes, Roy.
Good afternoon to everybody present for this session. Today I'm going to talk about the computational and metabolomic insights into the Actinobacteria mediated management of bacterial leaf blight of rice. Rice feeds half the million population of the world, and a major hindrance to the rice production in this world is caused by bacterial leaf blight of rice, which is caused by Xanthomonas oryzae, Petavar oryzae. And you can see that chloropleth map in which India comes under a high risk zone for bacterial leaf blight susceptibility. As the world is shifting towards sustainable management, there is a high need in the hour for the biological control management of rice. And why Actinobacteria? Because they are a very good plant growth promoting agents, and also they are elicitors of plant defense, and also they are a very good biological control agent. Isolation and enumeration of the Actinobacteria from rice rhizosphere and the screening of these Actinobacteria against the Xanthomonas oryzae, Petavar oryzae, potent Actinobacteria which are screened through the primary and secondary screening were further tested in glass, house and field condition, metabolomics profiling of the potent Actinobacteria using the HRLCMS method, and the molecular interaction of the pathogenicity factors of SU with the actinobacterial metabolites in silico conditions under molecular dynamic modeling. This is the flow of the study. These are the diverse actinobacterial isolates from the rice rhizospheres, isolated using the serial dilution method in starch casein agar medium. And primary screening of the actinobacterial isolates was done using the dual culture plate, and the 8 isolates were found to be effectively reducing the disease suppression, and they were taken for secondary screening in disk diffusion assay, and AMGD 16, 20 and AUDT 502 were used to were very effective to control the bacterial leaf blight of rice. And molecular characterization of the AMGD 16 was found to be Streptomyces gingivanus, and AMGD 20 was Streptomyces xiamensis. And when we're biochemically characterized, they were producing sidrophore. They were positive for IAA production, catalase production, and also HCN production and phosphorus solubilization. And when taken under field evaluation, the seed treatment at 10 liter per kg and 2 foliar spray of 10 ml per liter with AMGD 16 at 10⁸ CFU per ml had enhanced the root length, the plant height, reduced the lesion length, and which showed that they are good plant promoting and disease suppression agents. The AMGD 16, which was a very good biological control agent, were further taken for HRLCMS analysis to understand its metabolome. And based on the area of abundance, different metabolites were selected, and literature mining was done to understand the function of those metabolomics, and we saw that there are different indole derivatives like indole 3 acetic acid, indole 3 carbinol, methyl indole 3 acetate, and also indole 3 acrylic acids and pyroxidine. They are all different very good growth promoting or metabolites, and also lumichrome, fusidic acid, and manumycin, oleomycin. They are all different antimicrobial agents which can be used for further reducing the Xanthomonas disease suppression of bacterial leaf blight. This is the total ion chromatogram of the actinobacterial isolate plotted by relative abundance against the time, retention time. Different peaks show the relative abundance of different metabolites, and several peaks show the diverse nature of metabolites produced by the AMGD 16. And in order to understand where each metabolites belong to which pathway, the global pathway enrichment analysis is done, and each bubble represented the different pathways. And the major pathways were found as valine leucine isoleucine biosynthesis and degradation pathway, the pyrimidine metabolism, purine metabolism, tryptophan metabolism. They all have different roles in plant growth promotion and also microbial suppression. And further, to understand the molecular level of interaction, two pathogenicity factors, that is PTHO1 and peptide deformylase, which are very important for the virulence and the viability of the zoo, were taken. And the molecular docking studies was conducted between the different metabolites and also the pathogenicity factor. And PTHO1 binds with the promoter region of OH sweet gene 11, which will upregulate and lead to the sweet sugar influx. And this is the Ramachandran plot showing the backbone dihedral angle distribution between the phi and psi angles of the amino acid bases. And most of the amino acids were in the allowed and disallowed, in the favored and the allowed regions. And you can see that lumicrome has bound with a minus 6.6 kilocalorie, binding energy of minus 6.6 kilocalorie per mole, and this had the least binding energy, and this was and had three number of hydrogen bonds, which was further taken for dynamic modeling to understand the stability of the protein. The RMSD plot also shows that in the initial phases there was fluctuation, but towards the 100 nanoseconds the entire complex was stable. And when we had seen the RMSF, that is the root mean square fluctuation, only controlled fluctuation was there, and the radius of gyration also showed that the entire protein structure was compact, and the more number of hydrogen was bond was found throughout the entire entire over time, which shows the stable nature of the complex. And the superimposed structure also shows that it is an induced fit model, which shows the stability of the protein and the ligand. And further, PTHO, the peptidyl deformylase protein also was taken, and here also the stability, the stereochemically stable protein was present. And here also lumicrome had bound with the peptidyl deformylase with a binding energy of minus 8.3 kilocalorie per mole. And here further you can see that the RMSD plot, fluctuation, RMSF plot, and all the plots show the stabilization of the protein. And the superimposition showed a induced fit model. The key outcomes are AMGD16 as seed treatment and 2 foliar spray reduce the bacterial leaf blight incidence to 32.17%, and the lumicrome effectively bound with the pathogenicity factors of the xanthomonas. And lumicrome can be considered for the future applications can be lumicrome can be suitably a commercial scale production can be undergone through microbial fermentation, and it has both antimicrobial and as well as a growth promoting character. And till now, no direct antimicrobial agent, like lumicrome was never used as an antimicrobial agent in plant disease management. So this is one of the studies where the importance of lumicrome is well documented, and the structural stability is being shown through dynamic modeling and molecular docking. Thank you.
Thank you, Ms. Ashita Roy, for an excellent overview and also sharing some insights on Actinobacteria-mediated management of rice BLB. Thank you so much. Any quick questions to Ms. Ashita Roy? Thank you. I think last 3 hours we talked about, and we heard a lot of story or findings on the rice. Now we have 2 next presentations, which are on, one is on brinjal and other one on sorghum. So first I would like to invite Mr. Nikhil Kumar to talk about decoding the metabolites during the fruit development for a disease tolerance in brinjal.
Good afternoon, and one and all present here. I am happy to present one of the interested crop other than the major crops. Here I will be presenting our work decoding the metabolites during the fruit development for disease resistance in brinjal, Solanum melongena. Basically, eggplant is a widely cultivated and economically significant crop in the Asia, and like in India also it is like major crop used as a food. And it is susceptible to fungal, bacterial, insect pests, and it leads to yield loss and reduced marketability. The secondary metabolites like phenolics, flavonoids, anthocyanins, glycoalkaloids not only contribute to taste, but also it plays an active role in plant defense against biotic stress. Defense responses like secondary metabolite accumulation depend on developmental stage and tissue type. This untargeted metabolite captures the global changes in defensive metabolites. which helps to identify the biochemical markers and pathways involved in fruit defense and disease resistance. This is the one special variety called Solanum melongena variety Mattugula, which is only grown in Mattu village of the Udupi district, which bagged the GI tag in 2011 from Chennai. For this experiment, we have selected 3 fields in which we have selected 25 plants. In each plant, we labeled 5 flowers, and later, in the time interval of 15 days, we have collected the samples. Later, the peel and pulp were separated and metabolite extraction were performed using acidified methanol, followed by homogenization, sonication, and concentration. This table 1 represents the gradient program for LCMS and the instrument parameters, and this that is that figure 4 represents the workflow which has been performed in the Compound Discover software. This figure 5 represents the multivariate analysis for the 15 dpa peel and pulp samples, where we have found 400 plus metabolites, in which we have found 123 common metabolites, where 44 metabolites were upregulating and 47 metabolites were the downregulating. Whereas in 30 dpa we have found that 330 metabolites, where 77 metabolites were found in both peel and pulp, where 10 metabolites were upregulating and 19 metabolites were downregulating. In 45 dpa samples, whereas the pulp sample we have found that more metabolites, and for only 45 metabolites we have found a common in both. When compared to the initial stage of the development to the final mature stage of the development, the common metabolites are reducing and the unique metabolites were increasing. So we thought to check what are the major disease resistant metabolites and the metabolites related to defense. So we performed that analysis, and we have found that here we have compared in between the stages. Previously we performed the analysis among the peel and pulp of the same stage. Here we performed in between stages. This slide represents the disease resistance metabolites in peel samples, among 15 versus 30 dpa, 30 versus 45, and 15 versus 45. We have found that only few metabolites which are found common in both stages. Other than that, there are the many unique metabolites. This is the disease resistant metabolites in pulp sample. Here, when among the development, the number of resistant metabolites were increasing when compared to the early developmental stages. And later we performed the metabolite pathway analysis, where we have found that the compounds which we have identified for disease resistance, they were undergoing pathways like phenylpropanide and flavonoid pathway, oxylipin and fatty acid signaling, amino acid defense pathway, steroid alkaloid and cyanogenic glycoside pathway, and terpenoid metabolism. To conclude, in this study, a wide and targeted metabolomic approach was performed to identify disease-resistant metabolites. A total of 1,041 metabolites identified in 3 stages, among which more than 45% of the metabolites were identified as disease-resistant metabolites. This fruit development involves the progressive metabolic reprogramming, where peel tissues accumulate defense-related metabolites in stage-dependent manner, forming a chemical barrier against the disease. These tissue-specific metabolite profiles suggest active metabolism, likely reflecting differential roles of peel and pulp in defense, transport, primary and secondary metabolic process during fruit development. These are the key references used for this study. And I would like to thank my mentor, Dr. A. Muthuswamy, for his constant guidance and support. I would like to thank Manipal Academy of Higher Education, Manipal School of Life Sciences for their infrastructure and facilities. And I would like to thank SERB and ICMR for funding to our lab. And this is our team working on the broad aspects of the plant biology. Thank you.
Thank you, Mr. Nikhil, for sharing some of your work on metabolic pathways and also profile. Thank you for the excellent work and sharing with all of us. Any question? Yeah, please, sir.
Thank you, good work.
Thank you, sir.
There is one species called Solanum macrocarpon.
Yes, sir.
It is called tomato brinjal and also edible. The good part is it is edible.
Yes,
and if not immune, it is very highly resistant.
Yes, sir.
I would request you to parallelly compare the metabolites with the macrocarpon with the melangina and see where exactly we can gain.
Sure, sir. Sure, sir. We'll carry on.
Okay. No more question. Thank you again.
Thank you, sir. Thank you.
So I'll invite now the last presenter for the today session, Ms. Ranjita, to talk on identification of marker trait association for fall armyworm in resistance in case of sorghum.
Good afternoon, everyone. I will be presenting on the topic Identification of Marker Trait Association for Fall Armyworm Resistance in Sorghum. This study focuses on identification of the genome-wide SNPs and their association with fall armyworm resistance. This pest is recently invaded across the globe. So fall armyworm, Spodoptera frugipeda, is native to America. In 2016, it was first reported outside its native in Western Africa. Then in 2018, in India, at Karnataka. So till now, it has invaded more than 80 countries across the globe. With this rapid dispersal ability, it has become a polyphagous pest, infesting more than 80 crops, host crops, majorly maize, sorghum, and rice. And we know that sorghum plays an important role in food and fodder system, especially in semi-arid tropics regions across Asia and Africa. So this pest is causing yield losses up to 30 to 80%, and currently chemical control is in management, but it is ineffective as fall armyworm feeds inside the rolls, and this pest also has developed a resistance to many of the insecticides. So host plant resistance is the best alternative approach. So with this background, we have framed our objective is to identify the molecular basis of resistance to this pest. We have taken 242 minicore sorghum accession from ICRISAT, and this minicore panel represents the global diversity. It helps in identification of rare alleles as well as novel genes. So this is our, we framed in multi-step screening, including a field as well as laboratory. So in field, we have screened under natural infestation condition as well as in artificial infestation condition using a screen house facility available at ICRISAT for the across 2 seasons. Here, the observations we have taken are like foliar damage score, glossiness, vigor, and leaf sheath pigmentation from the scale 1 to 9 Davis scale for the foliar damage score, and other traits 1 to 5 scale, and under artificial also foliar damage scale for 1 to 9 scale. And to take the account of antibiosis mechanism, we have also conducted detached leaf bioassay for the same panel, 242 mini core collection. And we have here we have taken leaf sample and kept in agar medium, and we have infested the 10 neonate larvae, and after 5 days we have taken observation for the leaf damage score, then number of larvae survived, and the weight of the survived larvae. So from this we have taken bluff values, and we taken further the genome-wide association study. That is all about phenotyping, and genotyping. We have GBS data already available at ICRISAT. So we have aligned that GBS data to sorghum reference genome, and we have got 1,73,000 SNPs. And after filtering the minor allele frequency, we have got 1,70,000 high quality SNPs. So after that, we have corrected with the population structure, then did GWAS at using GAPIT package in R, using a multilocus model like models like Blink, Farm CPU, MLMM, at a significant threshold of 2.9 into 10 to the power of minus 7. So after that, the resulted SNPs we have passed with this GIF annotation file taken from NCBI version 3.62. Then after that, we have checked for genes present within plus or minus 50 kb LD region. So coming to the results, all the traits showed significant genotypic variation with more than 0.8 heritability for leaf sheath pigmentation and moderate heritability for glossiness and others traits with low heritability. And when coming to the correlation between the traits, the trait vigor, glossiness and foliar damage score are they are moderately associated, but here leaf sheath pigmentation we got weak correlation, but earlier studies showed that leaf sheath pigmentation is contributing resistance to pest, and recently in sorghum also the flavonoids are shown detrimental to the fall armyworm. So as they belong to ordinal data, so we did correspondence analysis where we converted the scores into resistant classes for both the trait leaf damage rating and leaf sheath pigmentation. So after association, we got certain classes like partial resistance and susceptible classes were strong. They showed a strong association. So after the GWAS, we have got a total 45 significant marker trait association for the 4 traits: foliar damage score, leaf damage rating, leaf sheath pigmentation, and larval weight. And some of the traits you can see leaf sheath pigmentation and larval weight as well as LDR showed more, they occurred in more than 1 models. So these are the total 45 significant MTAs. And through Blink model, we have got 10 significant MTAs where you can see the p-value ranging from 10 to the power minus 7 to 10 to the power minus 100. And it also showed good phenotypic, they explained a good phenotypic variance ranging from 3 to 66% of the variance. And so you can see the Manhattan and respective QQ plots of the association. The foliar damage score 1 for 1 SNP and 4 significant SNP for leaf sheath pigmentation, and 2 are on the same 6th chromosome, and 1 for LDR, and 4 for survived larva weight, where 2 are on the 1st chromosome. So after the annotation, we got the genes, candidate genes present within plus or minus 50 kb, where we got this description of the identified candidate genes. Example, FDB for the trait LDR, and then other proteins for the leaf sheath pigmentation like kinase, and as well as CBS domain. And then this larval weight actually showed a good related proteins like proline dehydrogenase and pectin acetylase, and as well as peroxisomal nicotinamide. And also for LOH2 related to lipids and related to calcium flux and glycosylyl transferase, as well as serine or threonine specific proteins. So previous studies also have reported some of the proteins like peroxisomal nicotinamide. They play important role in jasminic acid, other oxidative stress-related pathways, actually through flavonoid-based host defense. And calcium binding family proteins, the calcium flux will help in signaling, and also the other proteins like pectin acetylase and lipid related yellow H2, and they are involved in cell wall remodeling, and other things like glycosylcell transferase involved in salicylic acid mediated signaling. So in conclusion, our study GWAS on 242 sorghum mini-core collection, we have identified 45 significant marker trait associations for the fall armyworm resistance, and through blink 10 MTAs for the 4 traits, explaining 3 to 66% of the phenotypic variance, supporting the involvement of multiple loci from small to large effects. And annotations showed that they are involved, those candidate genes are involved in some signaling, redox regulation, flavonoid biosynthesis, as well as cell wall modification. So in future, the validation of the identified candidate genes are needed, then using of transcriptomics and metabolomics to understand the mechanism of resistance, and then next the development of markers for these candidates, and next further involvement of them in marker-assisted breeding. Thank you.
Thank you, Ms. Ranjita, for sharing outstanding work, and also to demonstration of some of the tools what our earlier presenters presented, both in analytics and also molecular tools. Thank you for that overall. And now I request Dr. T.S. Sharma to share some conclusion.
Thank you, Dr. Gowda. So in this particular session, we had 8 presentations, including 3 oral presentations by our young colleagues. And you all will agree with me that this was one of the most enriching and knowledgeable session which we had on rice. And I would like to say 4 to 5 points on this particular sessions. Particularly, we learned about disease and insect pest scenario of rice in the country, their prevalence, significance, and management practices. Then a success story of discovery and deployment of blast resistance gene PI54 and its impact on rice breeding. And then mapping and characterization of genes for various biotic stresses and their introgression in rice breeding using marker-assisted selection, and also successful examples of mass-derived products which we have in the country now, and then challenges and opportunities of rice improvement globally, and available new strategies with the breeders. And finally, we had 3 presentations by our young colleagues showing their work, and I would like to congratulate all the speakers, particularly our young speakers, for their nice presentation. And I would also like to thank my co-chair, Dr. Viresh Gowda, head R&D Wire Biosciences, and 2 reporter, Dr. Suma and also Dr. Gurudatta. Besides that, thanks are to the organizers for nicely conducting these particular sessions and giving us opportunity to be here this morning. Thank you very much.
Thank you so much, sir. Now we take the privilege to extend our sincere gratitude to our learned speakers. I call upon Dr. M.S. Prasad, sir, Dr. R.M. Sundaram, sir, and Dr. Amresh Chandal, sir. Please grace the dais, and I request our Honorable Vice Chancellor, Dr. P.L. Patil, sir, to do the honors. And I request chair and co-chair also to join the felicitation of our learned speakers. Yeah, now further we take the pleasure of felicitating our respected chair and co-chair also on behalf of University of Agriculture Sciences, Dharwad. So to Dr. Videsh Gowda, sir, who is a proud alumnus of this prestigious University of Agriculture Sciences, Dharwad, who did his masters in this university. And to chair now Dr. T.R. Sharma, sir, former Deputy Director General, ICAR, New Delhi. Thank you, sir. And we also extend our sincere thanks to both the rapporteurs, Dr. Suma Biradar Madam and Dr. Gurudat Hegde sir, and we request them to hand over the proceeding of the session by tomorrow morning. Thank you, sir. Now we have a very special event of exchange of MOU between University of Agricultural Sciences, Dharwad, and a private maize company. I request Director of Research, US Dharwad, to join us on the stage. And also the representative from the company. And I request Honorable Vice Chancellor, sir, to speak few words on this occasion.
Yeah, thank you, Dr. M.D. Patil. So you can take this is a commercial break or a commercial opportunity. So we want to commercialize our technologies released by our own scientists, maybe varieties, hybrids, and also the important inputs. So kindly refer to, I request all the industry partners, please refer to Agri Innovate India Limited website. We are commercializing all our technologies through Agri Innovate India Limited, and choose the technology required and express your interest, and through the material transfer agreement, we will have the commercialization of our technologies. And we have already commercialized 2 color cotton hybrids, maize hybrid, and biofertilizers. So like this, the list is go on. And also we have several the value-added products from College of Community Science. They are also available for commercialization for the young entrepreneurs or ladies to take up the activities. So here, today, so we are having a MOU between one industry, which is that, the seed company?
Sir, this is
Gen G Seeds and US Dharwad for our Dharma hybrid and another the line in the another hybrid in the pipeline. So Dharma hybrid, the features will be indicated by our maize scientists. So both are US Dharwad products. Each are sold for 3.6 lakhs, I think 3.3 or 3.6 lakhs. So in addition to Agri Innovate India, so you can also consult Directorate of Research US Dharwad for commercialization. Thank you very much.
Just to brief you the variety details, and with the support of our Vice Chancellor and successful licensing of our PPMC head Dr. C.R. Patil sir, we organized one maize hybrid field day during September. There around 15 private partners participated, and they shown the interest in some of the hybrids. And this is one fruit we are getting out of that one. And there are so many people, hybrid, they are asking for to evaluate in their field, and they may come. They have shown interest in next course of season. They will come and to have MOU so that the fruit of our scientists will go to end user, that is a farmer. These are the effort of our two breeders, Dr. S.C. Talekar and Dr. Kachapur, sir.
To move further, now we have a panel discussion on this theme of on the crop rice. I request Dr. R.M. Sundaram sir, Director ICR IRR Hyderabad, to chair this panel discussion. And then I call upon Dr. N.G. Ramachandra, Lead Breeder, Rice Breeding Bioseeds Hyderabad. Dr. Venkatesh Kulkarni, Director R&D, Nath Biogene Seeds. Mr. Devaraja Achar, Head Biotechnology, Indo-American Hybrid Seeds, India Private Limited, Bengaluru. Dr. Chandrasekhar S., Rice India Breeding Lead, Koteva, Hyderabad. Dr. S.K. Prashanti, Professor and Head Biotechnology, UAS Dharwad. And Dr. Viresh Gowda, Head Asia Rice Breeding, Bayer Crop Sciences, Hyderabad, to join as a panelists.
Yes, sir. Yes, sir, you can come.
Okay, that'll be fine. I look the odd man. Good morning to you all, rather good afternoon to you all. We are here for an important panel discussion on the theme, particularly breeding crop varieties for durable resistance to multiple biotic stresses, how we have progressed from genes to fields. And in this particular panel discussion, it's one of the imperatives because we have to address increasing complexity of pest and disease pressure. And we also need to discuss about the various strategies in terms of rapid or quick development of better breeding lines, which can tackle multiple biotic stresses, and how we can integrate conventional breeding with molecular tools, AA, and also phenomic tools, and how to bridge the gap between research, varietal release, and then farmers' adoption. There are a few questions I would like to ask the panelists. I'll just throw a few questions. We can have about a 3-minute submission by each panelist, following which we'll have a short round of discussion with the audience also. So the broad questions which the panelists can answer in addition to telling their own views is that, my first question is, are we over-relying on major genes? And is there any scope or limitation of deploying the minor genes or QTLs in a breeding program? How do we predict resistance breakdown before it actually happens? Are there any strategies? And the third question is, can artificial intelligence and machine learning reduce breeding cycle time meaningfully? The fourth question is, what is the ideal gene combination, particularly for Indian ecosystems? Are we there? For example, for bacterial blight, I can say with confidence. And similarly, based on the presentation given by Dr. Srinivas Prasad and Dr. T.R. Sharma, we are also there with regard to blast resistance. But with regard to the other insect pests and diseases in different crops, can we tell what is the ideal gene combination depending on what particular ecosystem in which you are growing. And my final question is, how do we ensure pyramided genes, how can they remain effective post-release, or how to prolong the life of the pyramided gene products? These are all broad questions which the panelists can attend, but I would like each one of them to submit their views or and try to answer at least one of these questions. I now invite from my right side, Dr. Viresh Godaji. You have about 3 minutes to give
Sure.
submission.
Thank you, Dr. Sundaram. So my viewpoints, especially right kind of a gene combination or right approach to tackle some of these challenges is it's always what customer needs. First, we need to think from the farmer what farmer is actually facing a problem. And it should be not across India or across overall rice community we want to solve. It is always essential to know that what product and what product concepts we wanted to build. Based on that, I think if we can really design a right gene combinations, looking at the challenges what customers are facing, maybe that will help us to address some of the challenges. On the top, yield is always important. Every time, without yield, any upgradation we do on the particular product, it will be not completely accepted by customer, because customer always or a farmer always wants improvement over what he is doing today. Not only just addressing the challenges, it is also essential to have yield improvement. That's where I can say a lot of now new technologies which are available, improving yield along with bringing a lot of this the tolerance. It will be a right approach, I can say. And most of the genomics what we learned today, if you can look at the animal genomics, I think a lot of advancement happened even before the plant. So maybe it's a good approach to look at what advancements are happening in animal genomics, which will actually help us to bring a lot of quantitative approach to tackle a lot of this major and minor genes what we are looking at. So always we need to keep a open, high, and thinking process to look at some of the advancements so that we can really tackle site-specific or target-specific problems. We can bring it for the customers.
Thank you very much, sir. I now request Dr. Devaraja Achar from Indo-American Hybrid Seeds to give his perspective on the topic.
So I would be discussing on marker-resisted breeding. That is one of the important key aspects for any kind of a trait improvement or development of a new varieties, in fact. As Sundaram sir said, from car to SUV, in fact, it was if you see the advent of the markers in almost 22 decades, majority of the varieties have been, mega varieties have been developed and have been commercially, viable in the market. So therefore, identification of key traits is very important. But if you see the complexity of the trait and the evolution of these races, in fact, with respect to the fungal and bacterial races compared to the virus, in fact. As we are pyramiding the time taken, what we are taking the pyramiding the genes or something like that, it is tenfold higher than what the strains or the pathogens are evolving, in fact. To combat and cope up such kind of things, it's very difficult, in fact. So that's the reason where the gene editing approach is one of the new approach where you can able to do it within short of 2 years, where you can anticipate, in fact. And now, not the developing or incorporating the resistant gene to the background. Now you are going to edit the same plant by recessive ways. That means either you are making the plant to be not recessive or resistant for a particular disease, in fact. So therefore, the fast tracking of those incorporation or editing the gene of the native genome is one of the best approach than putting the donor gene into this, donor genetics into this one, because of the lot of linkage drag and that and this. Okay. Of course, there are many approaches have come, genome association or other ways of these things. But pyramiding the genes in the native, in the major genes are fine. But if you take the QTL, again, it is a complex with the genome editing. So therefore, a lot of issues are there, but as a lot of marker-assisted breeding is need to be fast-tracked with the multiple tools, in fact. The best approach is presently the gene editing approach, which we need to take it forward for the varietal development or hybrid development in rice. Thank you.
Thank you very much. Now I request Dr. Venkatesh Kulkarni, Director R&D, North Biogene Seeds, to give his perspective.
Good afternoon, everybody. I am actually placed between two people who both are helping us in especially rice marker marker-assisted selection in rice. Whenever my laboratory is overflowed, Dr. Devraj Acharya helps me in analyzing our sample and sending the results to us. The way in which the cost and the speed that he manages is very, very appreciable. He has helped us a lot in developing our population, etc. The second one is Dr. Sundaram, because in my first part of rice marker-assisted selection, honestly, we suffered a lot because of the source. The markers were in such a source that at the end of the day, product was not coming. Now, I am very happy to say that the elite sources are being impregnated with the markers, and those are available for as a source for much more validated sources are available for development of especially blight and blast. I have some of the points that to be successful in these marker-assisted selection are the hotspots and as well as screening protocols. And a source of the material is very important. These three things, a consistent management of hotspots and pure culture of the organism will help us in getting the fastest and accurate results. In addition to that, your period of marker analysis and before flowering, if the marker data is available, then we can really manipulate within the field and get the things. When we said that we are going for 4 markers, then we means 1 out of 16 will be our homozygous plant. So based on that, we increased the population and we got the results. That actually helped us. On and whole, in rice, it is working very well for marker-assisted selection to address the biotic stresses. And in addition to the rice, in tomato, we are seeing a lot of success on that. Here, one of the challenges as a private sector person, we are also working on hybrid breeding. So one of the protocols that we have developed is that once we develop a B line with the marker, then integrating it, making it an A line, we are reducing the time span, and we are doing some very good work on that. However, a lot of our platform getting shifted to TGMS line. There will not be any big difficulties because it's as good as any pure line development. Durability question is consistent. Even we go through the marker-assisted selection. In the hotspot, consistent screening would help us in deriving a durable thing. Further, first few multiplication of breeder seed in the hotspot itself will also help in enhancing the durability of the material that we have developed. On AI, recently our team got, our R&D team got exposed to the AI from an US AI specialist. Our company is trying to make an AI agent for our R&D, and we are working on building the data. Historic data is very important for us. So I feel whatever I have learned till now and whatever I have experienced with a limited exposure to that, AI will have a lot of solutions to us with the quickest solutions from our historic data and future model building. AI can help us in a much better way in the coming days, because most of the agricultural science is built on a vast amount of data. For example, if you take your initial varietal trial, the amount of data that in that varietal trial is so humongous that it will be humanly impossible to assess the variation and means to judge the right variety as a superior. So I feel that AI will be a very great tool in the coming years for developing new models and identifying or decision making in breeding. With this, I thank UAS Dharwad and other organization for giving me opportunity. Sir, thank you for me. I feel very proud to be sharing with you platform.
It's my pleasure also, sir. Now, sir, I now request Dr. Prabhugoda Patil, who is a senior rice breeder in Advanta Seeds, to give his own perspective. Okay, sorry.
He's not here.
Okay, go ahead, sir.
So I'm Chandrasekhar from Kotiva.
Okay.
So,
okay, sorry, Dr. Chandrasekhar from Kotiva.
It's okay, sir.
Okay.
No. So I think we are like fortunate. Again, as I said, like I am also alumni of this college. Coming back here and talking in front of, especially students, is very encouraging. So from morning we saw like multiple presentations about the disease emerging situation and the new tools, how we can use those to effectively monitor, breed for the resistance, and the new question is about durability. I'll just try to address each one of those things. Like one, most of the time we discussed about deploying this major effect QTLs. I think which is progressed very rapidly, and especially on rice. I always think like in the insect world, Drosophila, and in the plants world, we always refer to Arabidopsis. But on a cultivated species, rice is having so much information generated in like multiple million base GBs, you can say. Like that much data is available on Genomics, Phenomics, everything is there. How to put that data into effective use and make meaningful decisions for breeding is actually a challenge. If that much data is available, that is where these new tools, AIs, and all those things will be helpful to make that right candidates. Because as a plant breeding students, we know that the simple genetic gain what we make is time and cost. Making those decisions in time and putting it in the cost-effective manner is. Sometimes we always think that it is only private, but it's happening everywhere because resources are infinite. So we need to make those decisions in right way. So marker-assisted backbreeds, as I said, like multiples we have seen. One of the area which we have significantly developed and we are seeing a benefit of that is, especially in a self-pollinated crop like rice, genomic selection. Because we are trying to make a self-pollinated crop into hybrid crop, and there are challenges, like for example, I want to evaluate 10,000 test crosses. It's very difficult to generate those many test crosses, put those in field and evaluate. So that is where your genomic selection tools will help significantly and make a lot of predictions and generate thousands of recombinants and advanced right recombinants. Otherwise, it would have been not possible without these genomic tools. And another thing which we want to discuss about pyramiding. So in pyramiding is like, again, like you will see in rice, as I said, like significant information is available. You will have multiple genes reported. We talk more of like product, market, customer. Our language will be slightly different from a private perspective, but for everybody it is same. Farmer is a customer. So you can load multiple genes into everything, but you need to have that right strategy, where what works, and put that right combination so that you have to balance the performance for other yield things as well as the disease. So putting that combination is very important from that perspective, so that you don't have a yield drag, you don't compromise on the yield plus the tolerance. So another thing, just touch about surveillance. Surveillance is human, the way we get our physical reports of this is reported there, that is reported there, but that gives more of a disease. But to go to the next level, biotypes, races, one of the approach which is evolving is molecular diagnostics. Like the way you characterize your the pipeline of the genotypes, you also characterize the pathogens for their genetic diversity, how they are emerging, and keep testing on your set of lines, which we typically call as differentials. That is one thing which we can keep you ahead of the cycle and look for those breakups and what is that which is working. So in the climate change era, these breakdowns will happen even faster. So these are my perspectives, sir. Thanks for the opportunity. Thank you.
Thank you very much, sir. Now let's move to Dr. N.G. Ramachandra, who is the lead breeder rice breeding program at Bioseeds Hyderabad. Sir.
Thank you. Thank you, sir. Thank you for this opportunity. I think I thank Dr. Sundaram and all the organizers for this opportunity. It was a great place to be there. Basically, if you look at the rice, is one of the. I should say I'm fortunate to be in the rice, because a lot of information is available. I think from total genome is available with us. So that's a great opportunity. And also, if you look at the public sector, we have a lot of institutions here in India, especially working in Hyderabad, so which are really cooperative and helping us in many of the things. Like we start with the screening. I think screening facility is wonderful in IRR, so which we can utilize. I think we are utilizing it presently and doing the things screening. Art spot screenings are one of the other things. I think these two are the artificial screening and the protocols available in most of the diseases, like BLB, BLAS, BPH, and all. So and also the upcoming diseases like fall smut and also the viruses, I think is developing fast. So, but at presently, most of the companies and most of us, including our company, we are working on basically BLB, BPH, and BLAST. So here the main thing is we have to look at in the farmer perspectives, what is required for the geography and what is required for the business. So if we load too many genes in one of the lines, so we are going to lose some of the yield genes. So yield is the main things. So first, for private company, yield is the one thing we should talk. So other additional parameters like health, hygiene factors are like BLB, BPH are the secondary there. But if we only concentrate the hygiene factors, leaving the yield is not good. So yesterday, I think some person asked a very good question. I think Dr. Krishna Kumar has answered him actually. So if you put everything in one line, so it may be super duper very good. It is doing well in all the gene factors, but it's going to lose the yield. So apart from the yield, I think rice is quality is also one of the important, quality and HRR. So if we don't lose, we have to maintain a yield, quality, and HRR. So apart from that, if you have one or two genes which can complement the yield, and where adverse conditions, especially when the high breakout of BLB, BPH it comes. So that will be complementary for the things. So which is very important. But if you look at the developments in rice, I think most of the companies, most of the institutions are routinely using the molecular tools. So that is the best part of the rice breeding at present. So if you look at any of the companies, even the smaller companies which are using the molecular tools for the disease resistance, they are outsourcing the molecular work and outsourcing the screening facilities. They are doing well. So this is one of the good beginning of the private-public partnership in India, I think. But we have a lot of things to be there. And what our request from the public sector is the actually allied background, genes in the allied backgrounds. So whatever available in present, I think IRI has done a lot of work on that. I think now they have started sharing with the MOVs and also on the payment basis. So that is one of the best things we should recognize. So if we do a lot of background work and clearing the backgrounds and coming with allied background material, that will be very good opportunity for the public sector, private sector to take away from to the market. So, and apart from that, we look at a lot of, I think, Dr. Chandrasekhar has talked on the genomic selections. So, which is one of the really big thing happening in rice. And I think after the corn, I think the rice is one of the things which is most of the people are using, especially accumulating the favorable haplotypes in our backgrounds, our germplasm backgrounds. So that is really helping us to break the yield barriers. But still we have a lot of issues because we deal with a 3-line system, ABR system. So we need to have a lot of things to taken care of that. So, but commercial breeding, we look at only thing what is required for the farmers. So we address that. Maybe the minor one, actually hygiene is one of the things, even it's a market, people come back with a smaller issues also if he is not working in that geography. So if you want to take overall, it is major diseases, but I request public sectors to work on the minor as well as. So we should be future ready for the things. Thank you.
Thank you, Dr. Ramachandra. We have here listened to diverse views from our representatives from private sector. Now let's jump into public sector. I request Dr. S.K. Prashanthi, who's leading the biotechnology program here at UAS Dharwad as professor and head biotechnology. Madam, your views?
Yeah, thank you, sir. Thank you for this opportunity. First, I would like to highlight about the blast disease management. So our previous experience as well as the past experience witnessed the importance of PI54, PI9, and then PI2 interaction individually as well as in the combination. And I would like to say that the PI54 with PI2, it is found the best for our condition. So before this, the very important thing as an academician, I want to say that gene deployment or R gene introgression or the pyramiding should be based on virulence gene information. So that should be done at the national program, like at the national level. So for that, there should be we need the helping hand from the national partners, and it should be a program in the universities or in the different academics. So the gene integration should be for the location specific, because Bagnaporte vora is it highly diverse and a lot of transposable elements are there. It's all based on our student study, I am telling. So that's why the gene integration, fine, PI54 is, and PI9, PI2, in South India it is performing good, as well as in the North India also. But what is the future for us? So my second thought is that, why can't we just think about pyramiding the NBS LRR with the other type of resistant gene? So that is the second thought where the academicians have to work, and then the private people can take that information to transfer into their different varieties or the hybrids. So this is regarding how best we can manage the blast for the durable resistance. And because the R genes, there are plenty, and then majority of the R genes are characterized, but the virulence genes, still we have to go further. So this is regarding the blast. And then the next thing is about the BLB, because in the upland situation, we never heard about the severity of the bacterial leaf blight. But since a couple of years, because of the rainfall, erratic rainfall pattern and then the climate change, we are experiencing the incidence of bacterial leaf blight in our situations, particularly in the drill zone situations. So we have to address it. Anyway, the genes are there. We have started pyramiding with the known blast genes, with the BLB genes. The program is there, already started in our university. And then the next challenge is the major challenge at the national level is the sheath blight disease management. So for that, we are stumbling at the first stage itself, that is identifying the different resistance source. For that, actually, I feel the work has to be still progressed where we can identify the very good source. Already the TETAP is known, which shows the moderate resistance, but there is any chance to find out the highly resistant source. We have the good germplasm collection, so we have started working, and then we have identified some of the highly resistant land races in our university and as well as in our germplasm pool. So again, it should be done at the national level so that we can go ahead with the management of sheath blight, which is very tricky. Already we have transferred the QTL, QSBR 11-1, with the PI2 and then PI54, which are all there in the testing resistance. It is showing fine, but at the same time, we have to select the lines for their yield and then the quality parameter. Here, the rice, the quality is also the prime importance. So these are the major challenges with us in front of us, and then for the sheath blight, still the long way is to go, and then coupled with the false smut. So we have to address these issues, and also again, several forum it is being discussed about the leaf blast and then the neck blast problem, and then how best these genes or the different genes. Can we have the such kind of genes, or the totally they are different types of genes that should be addressed? Even we as the academicians, we have to do some groundwork regarding that, and then fine. As my experience, 9 and then PI54, both at the both level, leaf as well as the neck blast, it shows resistance because our center, that is Agriculture Station Mugad, is known for hotspot for leaf as well as the neck blast. So private people can take the advantage, and then we are there to work on that, and then you can share some of the lines if you particularly want to screen for the neck blast, where actually this has to be addressed on the priority. So this is all my viewpoints. Thank you.
Thank you very much. I now will be floating one question to each one of you. First, let's start with go in the reverse order. My first question is for Dr. Prashanthi. I'd like to know, are breeding programs particularly relevant to blast resistance, since you are also leading a program in this direction? Are they fast enough to stay ahead of evolving the pathogen, more virulent pathogen pathotypes or biotypes? What's your opinion?
Yeah, we are, because our choice is good, because they are the stable genes, whatever in the program we are at the national level, like we have seen 9, 2, and then 54. I think for a long period they can sustain, because we have seen the other part also, because AVR 54 also in the NIP program we have seen that part, so that's why it is hoping so. But anyway, we should not keep silent. We have to go ahead with searching for newer, more alleles for that.
My next question is to Dr. Ramachandra. Sir, I would like to know whether we should continue. See, most of us have been deploying something called as marker-assisted gene pyramiding. We should continue doing this, or possibly in, say, another few years, we should go ahead with genomic selection in full steam. What's your opinion, particularly considering the pest and disease scenario in our country?
Yes. I think at present scenario, maybe another maybe until you get the germplasm loaded with the most of the traits, favorable traits. So we need to continue for maybe few years, maybe I see 2, 3 or 4, 5 years at the most. After that, I think the genomic selection is the only answer for all the pests and diseases, including yield. That's what my...
Thank you. My next question is for Dr. Chandrasekhar. It was the Rice India breeding lead from Kortiva. Sir, my question is, how many, you have transferred several genes in your breeding program in different crops. My question is, how many genes or QTLs should realistically be pyramided before agronomic performance suffers? Or the balance between multiple gene deployment vis-a-vis agronomic penalty? What are your views on it?
Yeah, I think it also depends on like which disease, sir. Like for example, blast.
I said blast.
Yeah,
good example.
Blast is like you will have breakdowns because these fungals generally they will have faster life cycle and they evolve fast. So you need to keep on doing that one. And for example, BLB, which is still very endemic to some areas, not very widespread, and also it depends on seasonal to seasonal. You need to be very, and it's like the most important factor which as a breeder I see is like if before deploying, we create base and converted and see is there any effect. So you need to see that for each of the gene. And for example, my experience, it may be different from others, but BLB have some drag is there, or some like blast, we don't see that much. And BPH, we have deployed a gene and in fact, we have seen reverse effect. Actually, it is positively influenced the performance. So it depends really on the question, on the gene in target, and also your own germplasm, because I cannot say what works for others. So you should know your germplasm and see before in that deployment, and based on that, you have to take a decision. There will not be a single standard answer for this.
Thank you, sir. You aptly put it. My question to Dr. Venkatesh Kulkarni is, what is the single biggest bottleneck slowing progress today in terms of large-scale deployment of molecular markers or genomic tools in breeding? From a private sector perspective.
From a private sector person, sir, if you're saying it is only in rice or in
No,
other,
any crop.
in
Say,
other crops?
Why, like say, the progress with regard to molecular breeding is good, but it's not excellent or outstanding? That's my assessment.
Yes, yeah. I go by you on that front, that it is very good, but breeder-ready markers in most of the crops are not available. For example, if you ask me as a private sector, after this rice, bacterial blight and blast, and BPH markers, in which crop you need breeder-ready markers? That I say that pearl millet blast. Pearl millet blast is one. We can take the similar line and develop. It is one of the most deadliest. Now, downy mildew is not a big problem in pearl millet. It is blast now. So breeder-ready markers, in maize at least, we don't have one. In millet, we don't have one. So in cotton, we don't have one. So we really need a breeder-ready markers. Those can be deployed very well. And of course, backcross method, which is to be used for integration purpose, a lot of us, we have standardized fastest backcross protocols, wherein background and foreground selection can be done, and we can deploy them as fast as possible. Coupled with now with speed breeding facilities, this can be done very, very quickly. Like speed breeding facilities and double haploid facilities of CIMET in maize, we can do very fast. I presume that with the success that what we have seen in rice and even tomatoes, the lot of efforts on developing breeder-ready markers for different problems that we have associated. Another experience with me is that if you ask me markers for insect tolerance and markers for disease tolerance, I prefer markers for disease tolerance would be more successful than insect tolerance with the experience that we have. So in such a case, the private sector, which has got good investments in developing hotspot, probably most of the private sector is also into heavily on line development programs also. That means to say backcross breeding, forward breeding are integral part of our breeding. So in such a manner, when all these are available, only thing, if you ask me that, okay, can we have breeder-ready markers developed from the private sector? When there is a question mark on the result of the experiments, in private sector investment gets curtailed. So we are seeking some proposals through BIRAC or DBT to get the grants and develop for important. In our company, we have listed 10 biotic stresses as important, and we are working on them on all fronts, including transgenic gene editing, conventional breeding, and management approaches also. So I love the marker-assisted selection, and we need some weapons and tools to handle them in different crops. Thank you, sir. Thank you.
My next question is to Dr. Devaraj Achar from Indo-American Hybrid Seeds. My opinion is that we are over-depending on major resistant genes, particularly in rice. My experience is limited to rice, and we should focus more on QTL-based resistance also. So in your opinion, based on your experience in multiple crops, what should be the ideal balance between major R genes and QTL-based resistance?
Yeah.
Take any one crop as an example, sir.
Yeah, if you take any crop, of course, you have a monogenic, oligogenic, and polygenic. If you see the same example for the blast, earlier we were talking about 1 or 2, 3 genes. Now the concept of monogenic and oligogenics has gone out now, because the multiple genes are being gowned. If you see the particular gene, the phenotypic variance of that particular gene, hardly it gives 30%, 40%, something like that. So we call it as a major genes. Now we call it as a major QTLs. The QTL approach is the best approach to introgress to any background, so where we can able to bring a maximum resistance. Even in the QTLs also, you could see that major QTL again minor QTL. So this is a very complex. So it depends on the what kind of a trait which we are targeting. For example, if you talk about the BLB in rice, hardly, as you said, these 4 or 5 genes are enough. But if you talk about the blast, it's not so. We are claiming PA54, PA2, PA9 gene. But if you take the validation, the level of validation, if you take the publication versus validation, we have more publication than the validations. So the lack of validation, sometimes it gives the inconsistency whether I should use the markers or not. So there is no proof of concept. See, for example, if you take the buyers which have been released, the rice, so hardly they have 4 or 5 genes and PA2. So if you see any private companies, every successive years or every 3 years or 4 years, absolutely they have to release some new hybrids. Otherwise, it's not possible. So the complexity of the diseases keep on increasing. So plants cannot combat immediately after introducing 1 or 2 genes. So therefore, I would suggest, it's my feeling, so my experience saying that complexity of the diseases is more. So therefore, QTL approach is the best approach where you can combat the multiple races, in fact. So not a single gene which is particular race. So that would be the best approach. And also as well as the genetic background. Sometimes if you see a particular variety, or if you have 10 varieties, you are introducing multiple genes or multiple QTLs. But all 10 varieties which have been introduced, they will never perform the same way for the resistance sometimes. So it is not a monogenic or polygenic, but it is oligogenic as well. But the genetic background which always complement your whatever the targeted QTLs. So we need to think about those genetic background as well, rather than focusing on those targeted QTLs or genes. So QTL approach is the best approach.
Thank you very much, sir. Now I have a question for Dr. Viresh Koda. Bayer, I understand, has invested a lot in multi-stress resistant or tolerant breeding, not only in rice, but also in many crops. What is your outlook in terms of multi-stress resistance breeding? You can take rice as an example. Say in 2035, how do you look at the scenario?
Yeah.
What actually we'll be doing?
Yeah, sure. I think I'll go back to one of the suggestions made by Dr. T.R. Sharma, sir. Sequencing is very cheaper now. We can make use of that sequencer. How can we generate more insights, and also to address some of the, the challenges what we have with new pathotypes which keeps emerging with the cloud of climate changes. How can we go and start using those information and bringing that part of our, genomic selection, which takes care of all the required traits for the customer. So I can say maybe we have gone through some of the era on the marker-assisted breeding or MEBSA. I think the way forward approach should be to go with the sequence information, which is much, much cheaper. I think even $100 what Doctor was mentioning, it is even lesser if you start thinking of a small crop like a rice. It can be done even at $30 per sample. So how can we get into that to tackle those is the approach which we have started doing already, and I think that's the way forward we are looking at, particularly in the Bayer.
Thank you. We had perspectives shared by representatives from both public and private sector. Before we close today's meeting, I would like to invite 1 or 2 students to ask any questions which are pertinent to the topic of today's discussion. Any questions from the students? Or even members of audience can also ask pointed questions to 1 or more of our panelists. Okay.
Thank you, sir. Since the whole lecture, in the morning, it was very interesting, and people were talking about rice. It is not directly on the topic, but my concern is about, instead of developing disease-resistant variety for multiple traits, can we focus on the indigenous variety, which is very popular in a local area, like Kala Namak or brown rice or red rice? What are the efforts being done for increasing the yield of those varieties which are still localized in certain area of India? Thank you, sir.
That's a question I can answer, because with regard to private sector, I think Prashanti madam can also answer. Private sector, the investment with regard to local land races may not be there. Madam, maybe do you have any perspective to share?
Yeah, we have witnessed the very good performance of the land races. They are unique for maybe the quality or for the resistant genes. That also we have already using those land races, resistant land races, and then identifying the genes and then transferring from that.
But the yield is the constraint in those varieties.
Yeah, but we have to take the permission from the biodiversity board, and also as I know there are some issues regarding we cannot improve the land races, but we can do the selection and then conventional breeding we can practice for the improvement of the land races. Or anybody can add to this?
Well said by Dr. Prashanti. I would like to add one more element to it. One of the most easiest ways in which we can manipulate or improve land races or land cultures is through genome editing. My colleague Dr. Satyendra has already started doing this in one of the local land races, of course with appropriate permissions from the community elders. He's improving a local land race called Chittimutyalu, which is known for its aroma, small grain size, and then extraordinarily high zinc content. He's working to improve because it's very tall, it has got photosensitivity, and it has got a few other problems. So by targeted editing of a few genes without changing the genetic background much, so that the useful traits will not be lost. So genome editing could be one among the many strategies which we can deploy for improving local land races. That's my sir.
I want to have one perspective, because Nath Biogen has one variety called Nath Poha, which is very, very popular. We sell a large quantity of that. It's fantastic poha making ability of that. We employed an MBA intern for assessing that. By inspiring that, we have a small breeding program. We spend around 10 to 15% of the time, called Specialty Rice Program, which is called Use For. So one of our board member brought up an issue saying that why should not be poha be in pink or red color? So we are now currently introducing semi-dwarf gene in those land races which are normally tall and bold seeded. So we have some red rice developed in semi-dwarf. Only thing, because our source of semi-dwarf is not very bold. So 2-3 more back crosses have to be done for getting semi-dwarf and heavily bold. The seed weight is between 33 to 36. So this is one, only we are focusing on semi-dwarf. On biotic stresses and other kind of things, they should follow. I know sources are very limited, and we should do it in the future.
Thank you.
Thank you, sir.
Any more questions from any members of audience, please? Can I ask a question in general, or ask a relevant panelist also to answer? Okay. I think. Yeah,
hello.
Yeah.
Good afternoon, sir. I am.
Yeah, go ahead, sir.
Yeah. So I am Chidanand.
Second last question, okay?
Yeah. So from Kottav Agri Science. So my question is, we have seen one virus, like rice dorp streaked virus in Punjab and Haryana area. So I have not seen anything in this public research. Now you have not yet started, I think so. So my question regarding that, we have any tolerance genetics available for this disease, or it may not be a threat to the rice production in India?
My perception is that the disease comes sporadically. Just like private sector, we need to prioritize our investments. So what we have decided as part of All India Coordinated Research Project on Rice, we have identified 2 research stations. One, our main rice breeding station at Ludhiana as part of PAU's group. And another egrip station is located at Kaul in Haryana. Around Kaul locality also, we observed a lot of incidents even this year. So those institutes are working. And PA has developed a field-based screening protocol through ELISA, wherein quickly you can identify the infected plants. And Kaul, together with PA, is working on identification or validation of the resistant sources. But this is a local problem, particularly in northwestern part of the country. The entire country cannot work on it. So we have prioritized this particular problem for Haryana Agriculture University Rice Research Station located at Kaul, and Punjab Agriculture University, their station at Ludhiana, to focus on this problem. Thank you.
Thank you, sir.
Last question, please.
Pathogen and environment. We tap anything like a host. In the host, we do everything we need from that side. So in pathogen point of view, how far we came to know the pathogen biology and whether, say for example, if we collect the isolates from different hotspots, we screen for most virulent isolate and we check it in some other place. So we got some resistance in that location. If we take that variety or a hybrid to a particular hotspot, it becomes susceptible. So my point of view, how far we know the pathogen biology relevant to particular disease. And my second question is, we talk about the terms like resistance and tolerance. When we have to use that? See, for my view, tolerance, in case of tolerance, if the pathogen and host is fighting and the host is...
Sir, since time is short, I got your two questions. First is regarding...
First question I will answer. So in the beginning only I told that we should once, before we start the breeding program, we should have the perfect knowledge about the pathotypes or the pathogen variability which is there, or where actually our product will go and then hit the market. That information, based on that only we have to start, and then we have to choose the R genes.
Yeah, there are multiple ways we can learn and know, but one simple way is always, we do conduct multiple multi-location trials. How to enhance the same opportunities to monitor the pathotypes or even whatever the races we want to know. If we can collect and start monitoring over the years, we'll know a lot of diversity and also the trend, the change, whatever is happening. Maybe that is one approach. I think it can be helpful as a simple way.
So adding to Veera Gowda's this one answer, you cannot forecast how quickly the pathogen is going to be changed, in fact, okay? So only the way is every season, every time. So we have to collect, and we need to, whatever the pyramided lines which we have, keep on screening only. That is the only way where we can able to say that, yes, whether I can go for this year, next year. The moment what we have been developed for the variety which is resistant for the same hotspot, so another next season itself we cannot expect that particular hybrid is resistant. So therefore it's a continuous process. We don't know exactly in what way they are going to mutate and how fast they are going to mutate. So it's a continuous process as a pathologist. So we should coordinate with the breeder who should work together. Then only we can able to come out something. Thank you.
I think we cannot take any more questions because of paucity of time.
So just want to add about his last question about resistance and tolerance. As a biology student, we should never use resistance, because we just talked about like next season only it may become. So there is nothing called in resistance in the biological world. There is a lot of multiple things: environment, host interaction, how it is evolving, races. So you should only use tolerance. That's my perspective.
No. It should not be like that.
Because viruses, we don't want to use the word tolerance.
In viruses, sir, I'm telling about more
when you
about
said biology, that's why I said, in viruses, we don't want to use the word tolerance, because in spite of the yield, there will be source inoculum will be there for further spread. So never select a tolerant variety if it is a virus. My other point, which I thought time is over, but I'll still share. In many horticulture crops, resistance is breaking down at high temperatures and extreme weather. My suggestion to all the panelists is also subject your genotypes to slightly extreme weather and see their performance.
That is something very well taken, sir. So now I'd like to close today's discussions. We had sharing of viewpoints or perspectives by many members from private sector, and we had one lone but not the least member. We had one representative from public sector, Dr. Prashanti, who shared the perspectives, particularly based on her experience regarding management of blast disease. And I can take at least 4 or 5 concluding points or points which you can take it from today's panel discussion. One, I would like to mention that resistance breeding program must also be dynamic, just like the way the pathogen and pests are dynamic, and they should be multi-layered. And along with deployment of the major genes, we should also explore increasingly the QTLs or the minor genes also. And I would certainly say that future breeding will or certainly should involve deployment of large-scale genomic tools, artificial intelligence, and also speed breeding. And we should not take away the fact that continuous monitoring of pest or pathogen evolution is the need of the hour. And along with resistant gene deployment, we should also do how these genes work at different locations and how the pathogen is evolving to overcome the different resistant genes. Last but not the least, collaboration across breeders, pathologists, entomologists, and data scientists, along with molecular biologists, is vital for success. I'm talking from my own example at IARR. We had a cross-section of team of molecular biologists, pathologists, entomologists, and also other trait specialists who worked together to deliver a product in terms of gene identification or gene deployment. And ultimately, farmer adoption is the ultimate success metric. And I'm sure that in rice, at least, we have recorded a lot of success stories wherein varieties with single trait resistance or multi-trait resistance or hybrids, they are increasingly adopted by the farmers. So I'd like to thank each one of our panel members. Yes, sir, just quickly.
No, sir. I just wanted to say because today rice breeding sets an example of in two-way direction of crop improvement. One, they are attempting to reduce the losses due to biotic stresses, and also they are increasing the yield by manipulating yield genes. I have visited his program, and both way, it was a visual delight when I saw the yield gene plot. So I think other programs can also be on this, increase the yield potential of the varieties, plus also reduce the losses due to biotic stresses. This two-way approach will enhance significant productivity in each of the crop, and I salute the IIRR's efforts in this, and the way in which the field visit was something which is memorable to my life, sir. Thank you for that.
Thanks, Dr. Venkatesh Kulkarni. I would like to thank all my panelist members for today's panel discussion and also the audience for active participation. Thank you, one and all.
Thank you, sir. Before we move for lunch, we take the honor of felicitating our fellow panel members. I request chair of the panel, Dr. R.M. Sundaram sir, and also Dr. Viresh Gowda to felicitate other fellow panelists. And I request all the registered delegates to carry the food coupons while you go for lunch. And there is a special announcement to the students. There is a special interaction session with the industry personnel at 6 o'clock. The interaction between students and the industry.
Registered delegates can collect the receipts.
And from today evening, all the registered delegates can collect their registration fees receipts at the registration counter. Now we'll break for the lunch, and we'll move to the guest house to have a lunch, and vehicles are waiting outside, and we'll join back again for the next session at 2:40. We'll have a lunch break of half an hour, and then we'll assemble at 2:40 for the next session on maize. Thank you. It's Hyderabad to co-chair the session. And Dr. Prema Juyu and Dr. Harshia Banu will note the proceedings of the session. In this session, we have 4 presentations, and we'll begin with the presentation from Dr. Satish Parikh sir, and then we have presentations from Dr. B.M. Prasanna sir, and then from Dr. Jarka Rashid, and then we have a presentation from Dr. Sobi SB. Now I'll hand over the session to Dr. P.U. Krishna sir.
Just looking for my rapporteurs. Please.
Hello, everyone. So, had your good lunch? Now, I do not want anybody to sleep here. And for that, big hello. So, with all due respect to Krishna sir, Arora ji, and talking about my presentation, it would be good, like, if I had a screen over here, but then I used my phone, I'm using the technology to go through it. So, now we have maize session. So let's talk about maize. We can go to next. So because they have put it at the first screen only, so I will say, like, I can introduce myself. I am Satish Parikh. I am very young for Dr. Tiku to be in the industry, because when he already joined the service, at that time I was just maybe still to be born. So, but then I am 30 years in the industry, and out of that 15 at one place only, that is Koteva. And other than that, worked at ICRISAT and ICR as well, so I know both sides of the table, probably a little bit under my Gurujans. So talking about corn, let us go to next slide, please. Okay. So if you look at corn, why talk about corn? It's an important crop. And now, scenario is changing very much in India. If you look at by 2030, so 60 to 70 million metric tons, and by 2047, we are imagining that it could be 110 million metric tons. That is what is going to be the demand for corn. Now, why? There are so many things which are leading to that demand, and there is a very much shift from corn as a normal crop to an industrial crop. That is what is happening in this country. Because of what? It is that is suitability of biofuel. Environmental footprint is less than crops like sugarcane, like rice. It doesn't demand that much of water. Prabhu sir, don't mind it, please. So, demand for feed and processing. Strong policy backing is there for ethanol. Like, you all know that government wants to blend around 30% now. So, sustainable alternative to water use intensive crops. Strategic advantages are also there with maize crop. Why? Because high productivity among the cereals with lower water requirement. Year-round cultivation is possible. Climate resilience and everything you also know. Greenhouse gas emissions you also know. Now, there are certain challenges also in this country. And why I am putting those challenges forward? Because we are sitting in a place where our government sector partners are also there. And they do a lot of basic innovations, and we expect them to do a lot of basic innovations which take the crop forward to the next level. And that is what is required. So, key challenges are dependence on monsoon, lot of rain-fed area is there, hybrid adoption and inconsistent seed availability. Seed availability is still a challenge. Government is doing a lot of work for promotion of HYVs. Gaps in drying, storage and market assurance. You all know that that causes aflatoxin issues. Quality issues in DDGS, because that's an animal feed, that quality is also important. There are lots of areas of expansion. you may call it as seed replacement rate, and how many farmers have access to seed. You know, there is an institution that also does the access for seed index. So those things are also important. Area expansion to certain states is also important. Some of these states have very low seed replacement rate in terms of HYVs. Then high ethanol recovery and diversification could be another area where opportunities exist. And resilience on the, and then reduction of the crude oil imports. That is also one of the primary things. Based on that, if you think of, there are so many research and technological priorities to come up to the standards of EBP 30. Germplasm diversification could be a very big thing through genomic selection or other methods. Then regional seed hubs, strengthen CGMS-based systems, like, you know, some of the crops like palm millet and rice have a very good CGMS system. For that sake, palm millet is a very, very extremely good crop. Then integrated solutions for aflatoxin, stalk rot, and other diseases. Now, if you look at the ecosystem, I highlighted something, and that is there are over 15 improved hybrids, more than 65 plus MOUs, still counting, every day MOUs are happening. And then 35 plus seed companies are there. Breeder seed indents are also, like, you know, increasing day by day. Collaborative efforts, expanding seed production hubs across Indian states. There are so many industrial and economic prospects. They are more like, you know, additional uses or secondary or tertiary uses from the corn. So let us not expand more on that. Now there is a certain key message which I wanted to deliver on that. It's not all about the NBTs, because, you know, there are so many molecular biologists in the room that will tell you that a plethora of technologies do exist. But I just covered a landscape saying that, you know, when we talk about gene editing, gene transfer technologies, XYZ, you know, that gets misspelled by some of the community, saying that, you know, anything related to gene, it could be transgenic. So basically speaking, in true breeding sense, if you call them, they are not transgenics. These are more like new breeding technologies. So what if we start promoting the word new breeding technologies? That could be more important, because that really concisely depicts the thought process behind what we are doing as scientists today. So if you look at, there are precise targeted DNA changing technologies are there, like CRISPR is there, which is very, very affordable also, cheap also, very easy also, and does a lot of good job. And then for specifics, there are like TALENs or ZF nucleases. Then there are another mutagenesis methods using the ODMs. And then there are reverse breeding technologies, like, you know, you can, rather than, you know, earlier days, some of the people say that they used to break hybrids. Now that is not required, because you have reverse breeding technologies. You can use RNAs just to stop the chromosome getting recombination will be stopped. Meiotic recombination will not happen. And then you can have parental chromosomes directly inherited, and you can go for complementarity. And you can utilize through those complementation, you can actually develop the hybrids much more quickly. So those kind of things are there. Grafting of GM root stocks is there. Cisgenesis and intragenesis, where these things are delivered, although you can do that cisgenesis traditionally also, like, you know, we have been transferring those genes of importance into the same species, but that can also be done now through new methods. And then always there is a speed breeding coupled with all these technologies to bring in quicker generation turnaround time. So these precisely put together can be called as new breeding technologies. Now coming to regulatory scenarios, how we see. So this is a map which was edited by the site in 2024 probably. Out of that, if you look at, those are no restriction zones which are dark green. And then those are where approvals are required in lesser green. And then the other ones are there, red ones are there, where those are not allowed. So, but after this, there has been a change in European Union, because SDN1 is now approved for a European Union. But still herbicide tolerance is kept out of that, because still that is treated more like SDN3 and approvals are required. So I have provided an additional link also on the presentation. That is the place where you can go and find this map. I don't know how fast they will once again update it. But Now, when you look at regulatory scenarios in India, so SDN1 and SDN2 events are more often, they only require the IBSC to present them and document them. Otherwise, mostly they are exempt from the DNA regulations. But wherever you talk about SDN3s, which actually involves the gene sequences, insertions and other things, so there only GMO regulations are applicable in India. So this is very, very fortunate, like, for rice, they have mutated the GN1A gene and you see the change has happened. It is more like changing the harvest index of the crop because you increase the number of grains that a panicle can produce. And then, this could have been done by normal breeding technologies that we are very much used to that would have taken a lot of time. So these technologies present a lot of opportunities for us. If you see progress across crops, this is not directly related to corn, but just wanted to give a small snapshot to you. Like, now you're talking about GABA tomatoes that have chemical that can lower your stress and reduce your BP. So stress reliever, some kind of a thing, that is becoming more popular. Mustard greens, then fungal resistant wheat or non-browning lettuce. These are some of the examples where crops have been released across the globe, and these are the live examples of how people are utilizing NBTs to develop the new crops. Now, when we talk about crops, especially maize, this is a very busy slide, but I can tell you, if you look at purely that segment of, if you look at the maize, then haploid induction or those diseases that we talk about, all the 6 important diseases, NCLB and others, then low phytic acid and high amylose, and then kernel number per plant. So these are some of the important traits in maize. So they are like, more amenable for targeting. How much we have targeted in our institutions, that is still a question to think of. So now, this is a very busy slide, but I am just trying to think of an idea, like, I just want to throw an idea to the hall, that are we going to be, when we have commercial institutes, like, private companies do not afford more than 1 or 2 breeders, or maybe if those programs are very big, then maybe 3 or 4 breeders. But, look at the institutes and look at the universities, how much big talent they have. And if you pool the talent across the universities and public institutions, including that of CGIR, then a big, huge talent pool is available. And then, there are a plethora of genes. This slide would have been very small one if I would have thought of accumulating everything across the globe, whatever is going on. Why not in India? And any of these, not only for the crops like corn, but if you speak of rice, there are very less number of examples to be counted, like GN1A, that's a very good, like Dr. Satyendra, what he has done. So, or what Corteva has done, like, pooling up all 4 genes together for resistance. So, utilizing the HT1 genes. So the thing is that in our institutions, what are we going to do? Do we need a change of mindset, like, are we going to do basic research that will change the crop's future going forward? Now, in the university, when we were going across, going through the fields with Dr. Pathak, we found that, for some of the things like, naturally growing the crops, there is data available for almost more than 20 years. That is just amazing. But what about, like, this is a global study where US government created that study. I have not included the pioneer Corteva example. Corteva has done an example of analyzing they call it as era genetic gains. So those studies were conducted, but this is from the USDA data, which shows the data analyzed from 1930s to 2010. And you see that what is the conclusion, that through normal breeding, which we usually called as a conventional breeding, also there is a no plateau for the genetic gain. But yes, of course, it happens thorough slowly. Now, increase was approximately 128 kgs per hectare per year from 1934 to 2020. Now, there was a study on the short season hybrids which said that it was 105 kg per hectare per year, so almost 1% annually. but they also did not observe any plateau. But now if you see the lower side, which is highlighted one, that what they found was, if you talk about resistance genes, then those are the genes which you incorporate. Of course, these examples are not from gene editing, but these are purely like, examples of transgenics. But if you look at biotic resistance, that is the one very low-hanging fruit. If you change the biotic resistance from a susceptible to a resistant component, immediately your yield levels increase, your yield sustainability increases, farmers' profit increases, so these are considered as low-hanging fruits. How they fit into the genetic gain delivery? So everybody knows, many of you are breeders, and you all know there is a breeder's equation. So if you look at the breeder's equation, so selection intensity, when you talk about genomic selections, shelf and test strategies through genomic selection, which help you utilize large number of data sets you can create, and you can hypothetically test large number of hybrids, just test and shelf. So utilizing genomic selection, large evaluation sets can be created and visualized. And some of those studies have reported that 0.6 is the selection efficiency for genomic selections. It can be gained, 0.6, 0.7 also, if you use additional covariates. Now in those situations, practically speaking, this becomes equal to our field trials. Because if you look at field trials also, that same kind of efficiency it delivers. Then, so selection intensity can be increased, selection accuracy, correlation between the predicted and true breeding values, I just now talked about. Then additive genetic standard deviations, utilizing the additive genetic variants. So when you talk about editing epigenetics for novel germplasm creation. So epigenetics is basically not to transfer, some of that is transferable also, but majority of it is just for studying whatever you have created, or suppose if you want to study a gene, so you cause it could be DNA methylation or something else, so you cause epigenetic variations and try to study the gene pathways. So, but that is not heritable. Some of the cases it is, of course, but these provide you, and editing directly, grain number gene. So those provide you chances of creating an additive genetic variation that can be directly utilized into your breeding system and breeding pathways. Then cycle length. When you talk about double haploids, those F8 generation comes to just one generation. And then speed breeding. So wherever that is not possible, double haploids, of course, corn is a lucky crop to have double haploids, but some of those recalcitrant crops, like pearl millet, still people are trying, but some of those crops are very recalcitrant and they do not respond to DH. So then, in those cases, speed breeding is an additional alternative. So these are all the new breeding technologies. If you look at them, most of conventional breeders, probably like me, would say that, arey yaar, ye biotechnologist kya karte hain? But technically speaking, all the streams of this science, maybe it is pathology or maybe it is biotechnology, everything can be harnessed to deliver the genetic gain. Big examples, Genelytics from Corteva. So they have done multiple editing, 70 plus edits in one plant. I think many colleagues should be from Corteva here. So, and then AI-guided target discovery. So 3 to 4 higher genetic combination rate they have reported. Multi-gene disease resistance. So that also has been reported. So multiplex editing has been reported by Corteva, and they have published it. CIMIT and other Grover partners, that is also a very famous example. So for lethal necrotic virus, they have delivered they have solved the problem of African continent, and they have delivered this really to on the ground. So now, what are the bottlenecks that were solved through these NBTs? So transformation, that was a limiting factor. They resolved it. Now it has become transformation independent. Directly it can be delivered. Polygenetic quality traits, they are very, very slow to improve, but that is also possible through NBTs. And then hidden deleterious alleles that reduce the genetic gain. So for that also, knockout is an answer. So that is one AI generated corn. So why it is important for corn now? Precision genome editing, multiplex gene editing, overcoming transformation bottlenecks, biotic stress resilience, and global food security. These are the key challenges in front of us, for which, especially in today's context, and more so in Indian context, because we are targeting reducing our foreign exchange deficit by importing less and less crude oil. So then it becomes too much important for us. And somebody quoted, I know many of us are non-corn people. I am also one of the non-corn breeder. But we feel or don't feel, one of my colleague who breeds corn, he told me one day, oh, you grow corn and leave everything. You can sell corn and buy anything else. So NBTs for biotic stress tolerance in corn. When we talk specifically about, now see, fall armyworm, and those in the bottom, they are the percentage of losses that happens by these biotic stresses. Fall armyworm causes losses to the tune of 35% on an average. Stem borer, again same example, another 25% of losses. So like that, TLB, so if you talk about it, is almost 15% of losses. So as I was sharing with you that entomological, pathological, weed competition, nematodes, these are all the kind of biotic stresses that cause problem to the yield delivery or yield sustainability or adoption of yield in one state and not in the other state. Say, for example, the corn hybrids that can grow in North Karnataka may not be growing in South Karnataka. So those are the differences. Those barriers can be crossed if we utilize NBTs and try to make changes. What is the value proposition for us? Protecting the genetic gain, it increases the resilience. Biotic stress is a throughput but bottleneck also. As far as I know from the private industry, if you are developing 100 hybrids, every year 30 to 40%, almost 30 to 40% of hybrids go away just because of the one or the other biotic stress issue. And then at the end of the day, you end up just promoting 10% at each level of your promotion. So over the years, if you look at your 10 years data across your breeding pipeline, you will see that your selection at each and every stage is not more than 10 to 12%. It could be approximately, varies between 8 to 12% only. So that is a throughput bottleneck. Value, so ensures market leadership of your company or institute or organization, because you can deliver market leadership. If you have leaves looking bad, if there are leaf diseases on corn, farmer will be very far away, even if it is yielding. So that is a reality of life. Lowers production risk for the farmer. Faster and durable, Downy mildew is one of the example you have seen very recently. So if you have biotic stress resilience, then it lowers the risk for farmer. Faster and durable improvements are possible. So these are the key diseases everybody knows, so I don't have to throw a lot of light. And these colored ones, dark colored ones, are the areas where corn is grown throughout the country. So you can look at very less area is I don't think anything is uncolored out of this graph. There are just few examples how this can be brought in. Very simple way could be you treat with physical mutagens, or you treat it with chemical mutagens. Nowadays anyway gene editing has come in, so you can create mutation through gene editing as well. And then you can just screen and take to M1, M2, M3 generations. And then you can do PCR screening, and then you can do gene tilling. So that's a very easy method to find out, not so easy as I am talking, but yeah, it's a comparatively better method for delivering the new alleles. Now, corn key diseases, published genes, QTLs and markers. Too much busy slide, but why I brought in only for one purpose. Those highlighted ones are only the examples where it has come to deployment. HT genes, and then Corteva has found those QTLs and implemented them. So those are the only examples. Probably in the hall you may be knowing more than me that there may be additional examples of the real-time implementation of these NBTs. But for me, this is what I could highlight when I tried to find out, like, what is the commercial implementation. So from this slide, I just want to show, or it is more like a thought-provoking kind of a question, that let us go ahead and see what additionally can be done and how much is the horizon available for improving the crop and really contributing to the Indian genetic gain. Now, that's the standard, like, I don't know why it is appearing like this on the screen, but can it appear as colored? Okay, no problem. Okay, not appearing. So anyway, I can tell you, CRISPR mostly it looks yellow, like, the genomic selection, mass and QTL. In the colored presentation, it all looks green because, most of scientists have implemented those technologies and they have come on the ground. Marker-assisted selection, it's an old thing, a lot of examples are there. Genomic selection has been implemented, and most of the places now people are trying for additional tools like, utilizing AI in that, utilizing additional new models like, random forest, and utilizing the covariates as other traits as covariates like flowering time, like moisture. So those all things are being utilized as covariates. So additional things are happening, but that looks more of green. Promoter engineering, it has not even come out of concept. It is just inside the laboratory. R-gene parameting and single locus stacks. Somewhere it is yellow and somewhere it is pink only. So that is the status of these new NBTs application throughout the globe. And that I have compiled only for the corn crop, not for any other crop. So you can look at this is a prime crop, it is an industrial crop for the country, and that is the status of, like, NBT's utilization. Now, what could be the considerations and way forward? So one important thing that is of consideration is adding phenomics and process automation tools. So phenomics, what does it mean by? It is not just the drones, but anything. You can have robotics going across the ground. A robotic vehicle that goes across the ground uses a LiDAR camera and recognizes your bar tags and reads through the data. That could be one. Then another could be robotics utilized through, like, your high precision cameras utilized through your drone systems, which almost everybody knows, and then doing the, like, big data analysis through those images that are digitally captured. So, but yes, for really improving the genetic gain, I think this is one technology which our institutes should certainly deploy. What is the way forward for NBTs? Now, that's a very critical question. When you talk about decision-making consideration for the organizations, which includes universities also, not only the private sector. So gene editing system license that, there is still some more clarity to be there. Cas9, that is the most popular system, but has lot of patents around it. Then Cas12, another system, then it has, that is the second popular choice. Now, newly evolving alternate system is TNP nuclease, TNPB. So that has got additional advantage because the protein size itself is very, very small and delivery methods will be easier. Then Open CRISPR is another newly patented platform that is almost open for everyone. So these could be like, obtaining licenses or working with other institutions that could be there. Then research service agreements can also be there. Now, if you look at why research service agreements, because, these technologies are changing day in and day out. Today, university deploys a particular type of, instrumentation and everything. Down the line, after 5 years, technology is going to change. So, are we going to consider having our own genetic engineering labs everywhere, or CRISPR labs everywhere in the universities, or we are going to have a center for excellence in just one place where it is easier to replace the technology as soon as it changes? Then public-private partnership in gene editing projects is another possibility. So we can access the gene-edited products for commercialization through utilizing those MOU framework, and then there could be co-development and various other partnership opportunities can also be there. Priorities for India, how would they stand? Pan-genome variability, that is still a question for corn. We need to focus on that. Somebody has to focus on it, specifically for our corn. focus on biotic stress resilience, then focus on well-defined 3 to 5 gene targets per trait. So, like, rather than going random, we should, for every kind of trait that we want to do, we should have a focus on what we want to do exactly, and what are the gene targets there. Adopt germplasm independent technologies, now they are there. Protocols are there for direct transformation, integrations and partnerships. So, integration and partnership, yes, that is a national question, because mostly every institute has their own de novo, like, platforms doing it. We could have thought of a national platform where this can be done together. I think that is something that has to be brought to the leadership's attention. Probably they may, senior leaders are there, like our chairman, sir. Probably they can take it forward to the people who form the policies. Then field evaluations, how it could be done, and then enabling the adoption. So those are the questions for deployment of NBTs in the corn. That's it. Thank you.
Thank you, Dr. Satish. So, beginning with the statement that we need to touch something like, or increase it to 100 million tons in 2047, Dr. Satish has taken us through the status of the maize ecosystem with reference to the methodologies which can be used to enhance the various components of maize productivity, and with special emphasis on some of those tools to reduce the timing of development of technologies. Like, you need not use certain technologies if they are not recalcitrant to double uplights. Then you can go for the speed breeding facilities and so on and so forth. And then going through some of those latent technologies. I use the word latent technologies because many of them, they don't approach these technologies. Like, for example, the epigenetics. There's a lot to be done in that particular area. So a good summary of the new technologies that needs to be probably applied to take the country to next level in terms of increased productivity. Can I quickly have some questions for him? Yes, sir.
Yes, sir.
What is the status of corn oil? Because we see a lot of corn oil and many others. I have also been talking with CIMAT. They say they have excellent genotypes which can contribute. And we, as it is, we have a tremendous shortage of. Is there a segment which is trying to work on corn oil, or for India it is not a very profitable crop?
No, when we talk about corn oil, it comes in a kind of a ecosystem. As such, alone it is not being utilized. But industries which are working primarily on corn, when we talk about corn ethanol, then certainly all these components are thoroughly important for the industry. Because when they are taking out the ethanol, at that point of time, they take into consideration all these important aspects like corn oil, like protein in the DGS, the protein quality, and then even they go up to the analytics of protein digestibility as well. So that's an important thing.
It's an important thing according to you. I mean, that's one thing. Second one, can you little bit elaborate on maize lethal chlorosis virus and how this issue was addressed for Africa?
So what they have done is that for maize lethal necrosis virus, what has CIMIT has done basically, they have utilized they have gone through a project, and then through that they utilize the technology to confer the resistance for the gene. ICRISAT is also coming up, CG institutes have very coordinated these things. Now ICRISAT is also coming up with a platform where they are thinking about introducing global markers as well as doing the genomic selections effective in crops our SRI and millets also.
What is your opinion on soil health vis-a-vis maize?
So we have when we talk about soil health, yeah, that's a very pertinent question, because some of the thought process is that maize could be taking out a lot of resources from the soil. But then the thing is that we need to come forward with those systems, cropping systems, which can rejuvenate the soil health, as like, you know, like in our farm also we have seen sorghum growing side by side, you know, the till crop. So we need to come up with those crop rotations, or we need to come up with those recommendations which can actually make it possible. But utilizing the crop industrially, that too for the purpose of our exchequer, that is the reality of today. When if we have to become nationally, like, you know, independent, and we have to have a better exchange ratio in the country. So for that process, it will be important. But now the onus again back goes to the scientists only. Like, you know, once upon a time when scientists have solved the question of wheat, lower productivity, bringing it to higher productivity. Same way today there may be a question that India is transforming into a maize crop as a commercial crop for us. Then these are the questions which probably we need to resolve.
I could add a point or two there. When you talked about the new technologies, they have used the CRISPR-Cas method to actually tailor the plant so that it could select the kind of microbiome that it needs in the soil. And that particularly enhances the entire population. So here is a place where you can actually make the plant in order to see that it selectively recruits the kind of microbiome that enhances the soil health and so on and so forth. Probably that could be an answer to his question.
Phosphorus uptake, nitrogen uptake, those are all the genes. They are naturally also available, sir. And they can be further edited. Thank you, sir.
Thank you, Dr. Sunil.
Thank you, sir. As Dr. Parikh is leaving, so we take this opportunity to thank him, and I request our chair to hand over the token of love and affection from University of Agriculture Sciences, Dharwad, to Dr. Parikh, sir. Sir, please. Sir. Thank you, sir. Now we'll have an online presentation from Dr. B.M. Prasanna, sir, Managing Director, BISA, New Delhi. Sir, can you please share your slides, sir?
Yeah, can you hear me? Can you hear me?
Yes, Dr. Prasanna, this is Krishna Raj from Dharwad. Hello, how are you?
Hi, Krishna, how are you?
Yeah, I'm doing fine.
Okay, let me project my presentation. Okay. Okay, I hope you can now see.
Yeah.
Yeah?
Yes, perfect.
Can you see the presentation?
Yes, it's perfect. Thank you.
Okay, should I start my presentation, Krishraj?
Yeah, please go ahead, sir.
Okay, thank you very much once again to Dr. Tikku, Dr. Krishna Kumar, and organizers of this meeting to allow me to talk on a topic that is close to my heart, that is how to strengthen the maize breeding and seed systems in the global south, based on my experience in sub-Saharan Africa, Asia, and Latin America. Okay, as the previous speaker has pointed out, there has been a rapidly growing demand for maize in Asia on diverse fronts, not only with regard to poultry feed or fodder or cattle feed, but also in terms of specialty maize and more recently utilization as biofuel. All this India needs to ramp up the not only production, but also productivity of maize in the country. In 2023, the global maize production was estimated at almost 1.16 billion metric tons across as many as 194 million hectares. And the global average productivity is 5.75 tons per hectare. This is significantly impacting maize utilization not only in the US Corn Belt but also across Africa as well as in America. But what is important is the tropical maize environments, especially in the global south, are inherently stress prone. If you can see the drought impacts in Africa from 1950 to 2021, all those red marked countries were severely impacted by drought stress. And this includes not only a total population that was affected, but also millions of or billions of dollars of crop loss that happened over the years due to abiotic stress. If you come to the South and Southeast Asia, what we are experiencing typically during the Kharif season is increasing temperature, not only daytime temperature, but also nighttime temperature. This was a picture taken between April to May 2024, where you can see several countries in South and Southeast Asia were severely impacted by the heat wave. Not only that, do the abiotic stresses are becoming more prevalent, but increasing incidence of transboundary pests and diseases can be seen in the tropical environments, whether it is the fall armyworm in Africa and Asia since 2016 in Africa, since 2018 in case of Asia, or the maize lethal necrosis in case of Eastern Africa, which happened in 2011 and still persists in Eastern Africa, although we could, through very concerted measures, stop the flow of this devastating disease into Southern Africa and West Africa. Tar spot complex, on the other hand, was earlier confined only to the Central America, but now we can see tar spot complex happening also in the US Corn Belt. Two particular diseases that are becoming increasing concern are the post-flowering stalk rots and the ear rots in case of maize in South and Southeast Asia. So maize-based agri-food systems, whether in Africa, Asia, and Latin America, need to be continuously protected from these devastating transboundary diseases and insect pests. The science behind maize breeding or crop breeding in general has changed significantly over the last 2 decades. Ever since the onset of molecular markers, there are now rapid developments with regard to high-density genotyping platforms and also sparking not only marker-assisted selection, but more recently genomic selection or genomic prediction. And thanks to the capacity to identify and clone genes, or even go deeper into the genome to identify causal variants, gene editing is now possible. Statistical designs and software is a less recognized, but a major advance that has happened in breeding. If you are a modern day breeder, you need to be thoroughly familiar with various statistical designs and software to analyze the data. Breeding data management and breeding analytics have become much more powerful than 15, 20 years back. Similarly, phenotyping advances have happened both on station and on farm, and coupled with rapid generation advancement through double haploidy in case of maize or speed breeding in case of crops other than maize. So all these are game changers in terms of modern day crop breeding. And that's why Mark Twain's quote on continuous improvement is better than delayed perfection really is relevant today, because you cannot wait till all these platforms become much more powerful than they are. You need to continuously adapt to the changes that have been happening on multiple fronts that impact the efficiency and the success of a breeding program. Cimet's breeding for climate resilient maize revolves around well-established breeding schematics. not only in terms of population improvement, but also line derivation, hybrid formation, evaluation through various stages, and finally licensing. Coupled with all this are, I would consider, 5 major factors that are really important if you have to make an impactful breeding program. One, strong germplasm base is extremely important. Second most important aspect is what is your population improvement strategy and what kinds of primary and secondary traits are you selecting for in an efficient manner. The third one is what kind of germplasm testing network an organization has, both on station and on farm in the TPEs. TPE means target population of environments. The fourth important aspect is very well defined breeding schemes with integration of double haploid, marker-assisted selection, genomic selection or prediction, or rapid cycle genomic selection as we call it at CIMIT. And the final most important thing is strong public-private partnerships for product, not only for product development, but also for product delivery or scaling. So the advances that we use, phenotyping is fundamental no matter what new technologies in genomics have come about, but your capacity to unravel the differences in the phenotypes of the germplasm that you are testing, be it for drought stress or heat stress or waterlogging stress or disease resistance or insect resistance, is absolutely fundamental. So you need to have excellent phenotyping specialists together with people who can contribute both on the field-based as well as molecular breeding fronts. Thanks to these advances, even if you look at the CIMIT's germplasm collection, we have around 25,000 landrace accessions in our germplasm bank at Mexico, of which around 15,384 maize landraces have been genotyped completely. And this genotyping enabled us to understand the genetic similarities or dissimilarities amongst the land races that came from multiple countries across the global south, majority of them coming, of course, from Mexico. You can see here, these are all Mexican land races. And these Mexican land races are very different in their composition compared to many other countries across Americas or Asia. But it doesn't mean that not all these land races could be utilizable in breeding strategy. You need to understand what is the functional genetic diversity for a set of characters that you are mainly interested in. That's where we went for land races that came from drought-prone environments of many of these regions, analyzed them intensively, both for their molecular diversity as well as environmental diversity, and shifted this core germplasm panel concept into breeder panels that could be utilized in breeding strategies to diversify the genetic base in our breeding materials, especially for core traits such as drought tolerance or heat tolerance. So fundamental, therefore, in our strategy to utilize plant genetic resources in breeding programs is this capacity to understand or unravel the functional genetic diversity in your germplasm banks. And that's how CIMIT contributed to the creation of what we call molecular atlases for maize as well as for wheat. And these molecular atlas in these two crops provides data, tools, and resources to allow the germplasm bank users, be it breeders or other researchers or extension agents, to identify the diversity of likely value for specific needs of breeding programs from the tens of thousands of germplasm accessions available, be it land races in case of maize or land races and wild relatives in case of wheat. So this is absolutely important. The plant genetic resource management has to change with the availability of powerful molecular tools and technologies that enable us to identify functional and relevant haplotypes for the diversity of traits that we use in our breeding strategies. The second most important thing is technologies to accelerate breeding cycles need to be implemented systematically. That's where the program that I led for almost 15 years at CIMMYT came up with the tropicalized maize haploid inducers. Initially, the first generation haploid inducers were brought out in 2012, and the second generation haploid inducers in 2017. And these haploid inducers, they are quite unique with almost 10 to 13% haploid induction rate. For example, our second generation tropical haploid inducers have been disseminated to almost 50 different research organizations, both public and private, across 18 countries in the global south. And not only we developed and utilized these tropicalized inducers, but we established the Maize double haploid development service to the national partners and small and medium enterprise seed companies by establishing 3 different research platforms, DH platforms. One at Kiboko in Kenya, the second one is Kunigal near Bangalore, and the third is Agua Fria in Mexico. And these DH platforms offer DH development service to national partners as well as SME seed companies. DH can become even more impactful if you combine this with molecular markers. So that's why any mature research organization, be it in maize, wheat, or any other crop, needs to have a well-established molecular genetics team. And this team needs to systematically progress from discovery to validation in independent populations, to validation in breeding populations, development of genotyping assays, and finally deployment of those breeder-ready markers for specific traits. And that's what we do. At each of these stages, we have a stage gate system where the researchers thoroughly thrash out what data is available, and is this data giving us confidence to move from discovery to validation to deployment stage. So this is a robust molecular marker pipeline for prioritized traits. And then once you come to breeder-ready markers, those validated molecular markers can be utilized routinely in forward breeding, marker-assisted forward breeding for allelic enrichment, rather than simply transferring the trait into 10-year-old, 15-year-old varieties. That marker-assisted backcrossing is not as powerful, in my view, compared to marker-assisted forward breeding, where you implement or utilize these markers routinely in your breeding strategies or in your breeding schematic. For those traits which are not amenable, which are not oligogenic, or 1 or 2 major genes or markers linked to them can cause significant phenotypic variance, you have to go through genomic selection or prediction for improvement of such complex traits. This is just a schematic to show how in CIMMYT global maize breeding program, we could save almost 30 to 40% time as well as cost of the breeding pipeline, while increasing the genetic gain almost by double. And this is, for example, Eastern Africa product profile 1, Eastern Africa product profile 2. This is what we call intermediate maturity. This is late maturity. This is highland maize breeding program. This is intermediate to late maturity in Southern Africa. And this is early and extra early maturity in Southern Africa. You can see a combination of traits being utilized for genomic prediction here, right from grain yield, anthesis date, plant height, ear height, striga tolerance, fall armyworm tolerance, GLS resistance, Starsicum leaf blight resistance. You can see a range: grain yield under low nitrogen, grain yield under drought tolerance, grain yield under optimum. So genomic selection or prediction is a powerful tool to utilize a smaller set of markers that cover the genome. Maybe not more than 3,000 to 5,000 markers can be utilized routinely once we have well-established training sets and the data, the breeding values of tester set can be then determined. So genomic selection or prediction can be used for multiple applications, for selecting high-value parents for your next cycle based upon genomic estimated breeding values, to reduce the breeding cycle time significantly, to undertake sparse phenotyping based on genotype relationship matrix, or to predict the crossing process for longer-term genetic gain. All these things are no longer theory. They are actually done in some of our breeding programs, especially maize, for instance, in the global maize breeding program of CIMIT. A series of publications we brought out, right from how to utilize rapid cycling genomic selection in a multi-parent tropical maize population, to combined drought and waterlogging tolerance in tropical maize, or genomic prediction across years in a maize double haploid breeding program to accelerate early stage test cross testing, or maximizing the efficiency of genomic selection in a tropical breeding program. Plenty of publications, but I'm just showing a few of them to show that this has moved much beyond pilot or proof of concept stage to actual mainstreaming in breeding programs. So GRM based prediction, for example, this is how we moved from a test half, predict half strategy to utilization of historical data and training sets to now test a few and predict a lot. That means once your genomic relationship matrix are well established, you need not have to phenotype every test cross. But while you genotype every founder line, every line that has gone into formulating those crosses. So genotyping all, but phenotyping a few, and genomic estimated breeding values based upon well-defined genomic relationship matrices. The second most important thing I want to touch base here is about genetic gain analysis. Genetic gain analysis must become an intrinsic part of our breeding strategy. Annual assessment of genetic gains is a must in maize breeding program, for instance. Besides analyzing historical trends or genetic trends across, this is a classic paper that we published in 2022, where data from almost 4,150 advanced breeding trials conducted at 1,331 locations across 28 countries were analyzed over a period of 11 years, 2010 to 2020 data. This is all on-station data. And this on-station data showed that genetic trends for grain yield have reached up to 138 kgs per hectare per year in USA, 118 in case of South Asia, and 143 in case of Latin America. This is, remember, considering all the complex traits that we were talking about, like drought tolerance, heat tolerance, and disease resistance coupled with optimal grain yield. Not only on-station analysis, but we also analyzed more recently on-farm data. So you can compare then on-farm how much yield gain that you are getting compared to what is the yield gains that you are realizing on-station. So over a period of 2011 to 2023, the on-farm genetic gains were 76, 50, and 34 kgs per hectare per year in some of these breeding pipelines, for example, what I mentioned here, which is not bad at all. If you are getting such 2.5%, 1.6%, 1.1% compared to the base yield or average yield in 2011, that means significant progress has been made. It's more easy to get on-station genetic gains compared to even on-farm genetic gains where farmers utilize diverse management practices or diverse inputs. Even then, our gains were quite significant. So this is an absolute must in modern crop breeding programs. You need to put a strategy in place for on-station and on-farm genetic gain analysis. Someone was asking a question about MLN resistance. Breeding for MLN resistance, we made tremendous progress over the last few years. For example, when the disease first broke, almost 95% of the materials, advanced breeding materials in cement pipeline were highly vulnerable. You can see the mean was almost like in 2012, it's around 8. Come 2016, we have shifted this population mean in terms of resistance to almost 5, to 4, to almost 3.5. Now it's almost 3. So that shows, you can see a highly MLN resistant genotype compared to a susceptible line. So this facility that was established at Naivasha enabled us to systematically screen germplasm, identify resistant sources, analyze the molecular basis for resistance, and introgress molecular markers to transfer resistance from different genetic backgrounds into our breeding materials. So if you look at the new MLN resistant lines, which are Simit Kenya double haploid lines, you can see all these with around, on a 1 to 9 scale, they are all 2, compared to the susceptible checks, which go all the way up to 8 or 7 or 8 or 9 even. So this shows the kind of progress that you can make even for a complex viral threat like MLN using breeding if you have a powerful genetic base and a multidisciplinary way of approaching a problem through dedicated facilities and dedicated strategy. The same way, in case of fall armyworm, when it broke in Africa in 2016, I could establish almost 16 screen houses at Quebeco Farm, each of it around 1/3 of the size of an acre. And these screen houses enabled us to screen thousands of germplasm accessions at our Quebeco Farm over the period 2000. For example, you can look at 2018, 19, 20, 21, 22, 23, 24, over 17,800 lines and over 2,000 advanced hybrids. Overall, about 20,000 germplasm entries have been screened from CIMIT against fall armyworm under artificial infestation in these greenhouses. This is a massive work. No public or private sector institution has undertaken such a massive work on insect pest resistance in maize through dedicated screening facilities. And the result of this, today we have released not only lines that have gone to over 90 institutions globally, but hybrids have been, fall armyworm tolerant hybrids have been released in Kenya, South Sudan, Zambia, Malawi, Ghana, and Zimbabwe. And they are already commercializing these CIMIT-derived fall armyworm tolerant white maize hybrids. Look at under no choice trials in the screen houses. That means entire chamber, almost 40 rows of maize of a single genotype or a single hybrid, infested twice at least artificially, one at the vegetative stage, another at the reproductive stage, and then analyzing both the foliar score as well as the ear scores after harvest. You can see a susceptible commercial check with not only very high foliar damage, but also ears that are damaged compared to a fall armyworm tolerant hybrid from CIMIT with much better harvest, much cleaner harvest, and much better foliar response. And that is the critical thing in the fall armyworm, breeding for fall armyworm resistance. Not just choice trials, but also no choice trials to validate the responses of those hybrids. And similar approach we have undertaken in Asia. Now we have fall armyworm tolerant yellow or orange maize hybrids coming from the CIMIT pipeline, which we are testing right now in several countries across South and Southeast Asia, including India, Lavopedia, Myanmar, Timor-Leste, Nepal, and Bhutan. And the process also has been initiated recently with the Philippines. Again, you can see a significant difference between a tolerant yellow or orange hybrid versus a susceptible hybrid. Gene editing is another powerful tool in the maize geneticist armory right now. This is the gene editing work we undertook in partnership with Corteva. You can see the resistant check, the susceptible check almost finished. This is a score of 9 on a 1 to 9 scale. The gene edited version of a susceptible line, this is the null. That means it has no editing done in that particular line, so highly susceptible compared to editing done. And you can see the drastic difference in the phenotype. So we have two approaches. One, editing a eukaryotic translation initiation factors, which have conferred resistance. The second approach is editing a peptidase gene on chromosome 6. Both these have yielded significantly positive results. The second paper is right now under review. In case of maize, not only a trait like MLN resistance, but you can also do gene editing for multiple traits. The genes underlying these traits like tassel branch number, tassel branch angle, kernel row number, ear prolificacy, leaf angle, or tillering habit, a number of traits have been already, the genes underlying them have been cloned and characterized, and the causal polymorphisms explored, and gene editing possibilities are increasingly now becoming available. China is clearly leading on that front. We need to now jointly evolve viable paths for commercialization of already existing genome-edited crops, besides overcoming IP and commercial bottlenecks to do gene editing on an array of other crops, including maize. Irrespective of the route we take, whether it is field-based breeding or genomics-assisted breeding, extensive on-farm trials are as important as on-station testing. In case of CIMMYT maize program, we conduct on-farm trials in Eastern and Southern Africa on over 2,000 sites in total. 2,000 sites. This is not a small number. In the last 3 years itself, the data has been collected from over 5,000 on-farm sites. What we then further did is to create a farm typology concept, not treating all these smallholder farmers as uniform and homogeneous, but to categorize them on the basis of household characteristics, their maize production characteristics, their input usage, etc., etc. So gaining a much greater understanding of product performance versus trait or product preferences vis-a-vis the farmer's typologies, and factoring this information into stage gate advancement of our hybrids. This is stage 5. The extensive regional on-farm trials is stage 5. Up till stage 4, it is on-station testing. So a selected set of hybrids then go into on-farm testing, and then we consider both on-station and on-farm data to identify well-suited hybrids for release under different target product profiles. All this means that the capacity of institutions to dynamically process large data sets, be it phenotypic, genotypic, pedigree data, environmental data, farmers management data, all these are extremely important. That capacity to process data and to take selection decisions. And that's where the human capacity becomes limited. You do need to utilize machine learning or deep learning models and embed them into your genomic assisted breeding strategy, and provide even more powerful predictive algorithms to analyze your data by combining this multi-layered data. This is what we wrote in an article in 2024 on how to utilize artificial intelligence in plant breeding, especially taking the efforts that we are doing at CIMMYT together with partners. Similarly, we came up with another paper in 2022 on how smart breeding can be driven by big data, artificial intelligence, and integrated genomic environmic prediction. Ultimately, what I strongly believe is the need to democratize predictive breeding. Predictive breeding is at present confined to a few advanced institutions whether it is in CGIAR or in some universities in the global north. But many institutions in the global south have very limited capacity to utilize predictive breeding due to various factors. Not only because of lack of well-defined breeding schemes, but the genotyping, even simple routine genotyping is people are not able to establish painless protocols. Education about when and how to apply genomic prediction, lack of computational expertise, logistical challenges related to genotyping analysis and making decisions within a short time frame required for breeding. So how do we bridge this gap? And how do we make even this guy become as powerful as this guy in terms of predictive breeding? That's the challenge for the years to come. For this, fundamental is to establish institutional data management framework. Breeding data at the present is confined to individuals' laptops or many a times in hard written field books and papers, and they all disappear once the person retires. This is not how breeding should be managed, or breeding data needs to be managed. If data-driven decision making has to happen, you need to have end-to-end software system that can connect haplotypes, markers, ancestors, germplasm, samples, seed packages, plots, trials, sites, phenotypes, environments, and product profiles. This is like a galaxy. How you visualize the stars in a galaxy are connected. That's exactly what needs to happen. And that will enable us, together with powerful analytical platforms, automating the GEBVs, PEBVs, GCA, SCA estimates, rich visualization of data. And then breeders can select what line I should use as a founder parent for what specific trait, what kind of optimal cross combinations could be maintained. How do I use sparse phenotyping or sparse test crossing in order to reduce the cost of my breeding programs? These are fundamentally important in modern crop breeding strategies. Coming to the seed systems, it's not simply adequate to release thousands of crop varieties and almost 90% of them never see the light of the day. What is really important is to have much better data and insights even before you release varieties, improved quality assurance and quality control system once a variety is released and put in the market, better strategies for gender and social inclusion, targeted reach of those varieties, digitalization of our seed roadmaps, and creating seed villages, especially when opportunities for commercialization are limited in certain crops. This means we need to scale up the digital and genomic tools and technologies to transform business processes and operations, and establish powerful public-private producer partnerships. And that's what we have done in case of Maize program by creating innovative product licensing models in the first place. So just look at this data. Over the last few years, 263 partners, of which 209 are private sector, are holding licenses across the world for CIMMYT maize products. We have released 270 hybrids through this process, besides 11 OPVs only for targeted market segments. And the number of countries having licenses or institutions coming from those countries are almost 76. This is the largest geographic footprint of any CGIAR breeding program across the world. And it is based upon geographical limited exclusive licensing models to public as well as private sector partners. The result of all this, when I joined CIMMYT in 2010, drought-tolerant maize occupied not more than 0.5 million hectares. Today, it occupies almost 8.5 million hectares in just 14 years, reaching 9.8 million households and benefiting 60 million people. Certified seed production of our CIMMYT-related varieties. The yield benefit, which has been actually estimated by economists in different countries, ranges from 500 to 750 kgs per hectare for drought-tolerant maize adopters, providing 9 months of food at no additional cost to a family. The annual economic impact of CIMMYT-related improved maize germplasm in Eastern and Southern Africa alone is 1.3 billion US dollars. So that is a powerful impact assessment study. All this is possible because of dedicated seed system specialists, excellent public-private partnerships, innovative product licensing models, and targeted reach of drought-vulnerable populations across sub-Saharan Africa. Asia is still at its infancy with regard to deployment of climate-resilient maize hybrids. But what we have created in the last 10-15 years is a unique set of drought and drought plus heat-tolerant maize hybrids and released across Sri Lanka, India, Bangladesh, Nepal, and Pakistan and Bhutan. So, and these hybrids are now being scaled up. For example, this is a project funded by Ministry of Agriculture and Farmers Welfare, where we have taken specific districts where the Kharif maize productivity is low, identified specific climate resilient maize hybrids from CIMMYT germplasm, and now in collaboration with state departments of agriculture, state agriculture universities, innovative maize farmers, farmers producer organizations, SME seed enterprises, deploying these climate resilient maize hybrids in those particular districts where clusters of districts in several states across India where the Kharif maize productivity is low. So friends, this is in brief what I would like to highlight in my presentation today and would be happy to answer any questions. Thank you.
Thank you, Dr. Prasanna. That was a end-to-end story. A concentrated information based on breeding dynamics, which included genotyping, phenotyping, and then the sheer size of experiments which leads to the development of algorithms to sort out the issues with respect to the maize breeding. You've touched upon several aspects, and I'm sure that the audience here have been treated to a beautiful story on maize on their palate. Thank you, Dr. Prasanna. Also, it's wonderful for you to have summarized the great American humorist, Mark Twain, when he talks about small developments which are useful rather than trying to, be a whiz in that particular area. Thank you very much, Dr. Prasanna. Once again, it was wonderful hearing you. I'm sure that's the message that you should get and you will be getting from all across the audience here. Now I request the audience to have some questions, and I'm seeing the hands of Dr. Tiku.
Please put on the video. I cannot see the video. Colleagues, can you switch on the video if possible? Otherwise, I will hear.
As usual, excellent. I mean, no words can express enough what you have said. And I've been following your work for a long, long time. I have only a request. I don't have a question. Why can't we recreate that public-private partnership in the sense you have done in CIMMYT? We don't have. I'm telling you, I attended my first public-private partnership kind of dialogue when Dr. Chadda had it in Trichur in 1989. This is 2026. I don't see even one project that has reached real public delivery. We are not talking of, this is our hybrid, you license. That is not PPP. For me, PPPs are like you did in MLN. I've been also talking to Suresh very often. So can you help now that you are in Delhi? Thank you.
Absolutely important, sir. I think in crops like maize, where seed companies have to play a powerful role in technology delivery, it's absolute must that we need to have these strong partnerships. Without those partnerships, the the impact and the reach of our hybrids or our varieties will remain very, very limited. That's why it is not easy, even at CIMIT, to champion that innovative licensing models. One had to think through a lot. One had to take everybody into consonance, answer many questions raised by different donor agencies. But today, the story itself is so powerful and successful that they recognize that these models need to be replicated across different CGIAR centers themselves. So I was happy that I could do that, and I'm a powerful advocate of public-private partnerships, especially not just in terms of technology scaling, but also in terms of technology development. We collaborate with so many institutions and make them true partners in executing our breeding funnel, breeding strategy itself. They don't have to come necessarily at the end. They can be systematically embedded in our strategy for product development itself. So there are lots of things that one can do. Yeah. Thank you.
Thank you, Dr. Prasanna. Now I have Dr. Krishna Kumar has a question for you.
To answer Dr. Tiku's question, and also, since you know, Dr. Prasanna approached ICR. I was at that time DDG. And though maize was not under me, the double haploid facility, I had to swim against the tide. And then today it is in Kunigal, a wonderful collaboration open up to anybody who wants to use the double haploid facility, and it is contributing a lot. I mean, just giving an example, just to say that, I think on the day
I still remember that meeting when I came to your office.
Yeah. So,
and thank you so much for all your support.
Yeah, the 2-3 quick questions. First of all, compliments for such a mega scale evaluation of germplasm for the newly introduced borer, that, I mean, the pest that was there. But if the mechanism of resistance is antibiosis, how fit it is for human and animal consumption? That is my first question to you. Because antibiosis by in itself, you can imagine that it may not be very desirable purely for a human or animal consumption, for which a significant proportion is being taken. My second question is with respect to maize lethal chlorosis. It is 2 viruses acting together, one transmitted by thrips and another transmitted by aphid, and together in also some beetles are attributed. In such a scenario, when normally we see that you develop resistance to the disease, the one which is suppressed begins to make an expression in many crops, as we have seen in papaya, as we have seen in okra and many others. The one which is weaker begins to assume a dominant position. I'm not sure how it is in case of maize lethal chlorosis. My small,
Thank you, sir.
My third, my only one small anecdote for the third point is, are we mixing up drought, moisture stress, and heat tolerance into one, whereas there are 2-3 independent events?
Okay, all three are very, very nice questions. The first one about the fall armyworm resistance, it is those compounds that we analyzed, even what makes certain inbred lines of maize fall armyworm tolerant naturally or native genetic resistance compared to the susceptible ones. This work was done in partnership with CIRAD France. And the compounds that are elicited are not ones which interfere with the ear taste or the kernel taste or kernel culinary properties. But these are the compounds that deter the insect, the larvae, from feeding on. They are like jasmonates, for example, or terpenoids, beta-caryophyllin. These are secondary metabolites that are particularly expressed in response to the insect attack. And it is a polygenic resistance, unlike BTKs, where that antibiosis is because of a very powerful but easily defeatable, because it is single gene or oligogenic. That's why I would on any day prefer a falamiworm native genetic resistance compared to even BT. Or if you have to introduce BT, introduce it into a native genetic resistant background. Then you have a much better insect resistance management strategy compared to transferring the BT gene into any susceptible background. So firstly, it's polygenic. Secondly, there are multiple mechanisms associated with native genetic resistance to falamiworm, about which we are beginning to understand what are those metabolites. But these metabolites do not interfere with the taste or the yield or other important attributes of related to the kernels. The second one with regard to MLN viruses, you are right that this is a complex of two viruses. The maize chlorotic mottle virus and the sugarcane mosaic virus, or any of the member of the Potiviridae. But that's the reason why CIMMYT went for simultaneously targeting two specific genes. One is a peptidase gene on chromosome 6, and another is the eukaryotic translation initiation factors on chromosome 3. Instead of simply going for one particular resistance gene, we are targeting two different resistance mechanisms. One is the translation initiation, another is the peptidase mechanism. So these mechanisms do not necessarily suppress only one particular virus here, like MCMV. It has an obvious effect on both MCMV as well as SCMV. And therefore, it doesn't make one virus stronger than the other. It attacks both instead of going for only one. So, by the way, gene editing is only accelerating the process. We were able to even breed without gene editing multiple resistant lines, but it took us about 3 to 4, 5 years to get those resistant lines. But what we started with 1 or 2 now have become more than 60 to 70 resistant lines, MLN resistant lines. So it's again a powerful story of how this resistance could be accomplished through breeding. The third question is about, sir, sorry, I could not remember the third one, the third point.
Are we mixing up heat tolerance?
Oh, okay. That's a very good question. Drought tolerance and heat tolerance are not same. Physiologically, heat tolerance is not just a kind of a lack of water availability, but also a vapor pressure deficit. It's the temperature coupled with relative humidity. That's what creates the VPD. So CIMMYT has established a network of sites for phenotyping for heat stress tolerance. Typically, we do it by planting in February and harvesting during the spring season. And that creates a very strong daytime as well as nighttime temperature stress. So drought tolerance, on the other hand, is related but not similar. So we have drought tolerant lines that are highly susceptible to heat stress. We have heat stress lines which are partially drought tolerant, and we have lines which are tolerant to both drought and heat stress. So you can get all the three combinations from the CIMMYT germplasm because physiologically and genetically they are not completely linked. But you can still mobilize both the traits into a single line or a hybrid.
Yes, sir.
Dr. Prasanna ji.
Patil sir, namaskar.
Namaste, good afternoon. Thank you very much for your excellent talk. We expected you to be offline. So anyway, you have made it in spite of your busy schedule.
Thank you, sir. Thank you.
So this is your second eye-opening talk to all our scientists as well, especially in particular our maize breeder. So you know our AICRP maize popularizing CIMMYT rain-fed hybrids. Yeah, once again I request you to visit UAS Dharwad
Sure,
Yeah,
sir, sure.
yeah, yeah.
Absolutely. It's a great pleasure.
Yeah, thank
And
you.
so many of my students are there, and I'm so happy to interact with you and the team anytime.
Thank you very much for excellent talk. Thank you.
Thank you, sir.
Thank you.
Thank you, sir.
Thank you.
So thank you, Dr. Prasanna. We'll now move on to another colleague of yours from Summit. I think Dr. Zarkar Rashid is online. Dr. Zarkar Rashid, pathologist from Summit.
Yeah.
Yeah, ma'am, you can go ahead. Thank you for coming in, and you can go ahead with your presentation.
Good afternoon, Chairman and Chairman sir, and the co-chairman sir, and my previous boss, Dr. Prasanna sir. This talk is going to be difficult for me after starting with Dr. Prasanna, because his flawless talks don't have any comparison. So let me just share my slide. Are you able to see my slides?
We are able to see the slide.
Okay. I'm Dr. Zarka. I was previously working with CIMMIT. And today I'm going to talk about the scenario of the maize diseases, particularly in Asia, as most of the things have been taken care by Dr. Prasanna, but still I'll give my point of view and just brush up that. As we know, the global food requirement is projected to double by 2050 due to the population and the socio-economic growth. Meeting this demand, particularly in the developing countries, requires doubling the current gain production of major food crops like maize, wheat, and rice. So maize plays a very important role in ensuring the food and nutritional security and the livelihood of the millions of the resource-poor farmers. While maize is a major source of feed and industrial products in the high-income countries, it provides the food, feed, and the national security.
Dr. Zarka, can you put it on the slide mode?
Africa, Asia, and Latin America.
Dr. Zarka, can you put it on the slideshow? Oh yeah, thank you. Thank you very much.
In tropics, the maize is predominantly grown under rainfed conditions, and which are often under the threat of devastating diseases and insect pests. Approximately 73% of the total maize in Asia tropics is grown in stress-vulnerable environments. Maize is exposed to stress around the year and everywhere. For the biotic stresses like drought, heat, and waterlogging are the main stresses. And coming to the biotic stresses, it is the BLSB, TLB, downy mildew, post-flowering stalk rot, and the fall armyworm. The maize, there is an exponential increase in the demand of the maize. The forefronts are the food, feed, fodder, and the starch-based industry. Apart from that, the surge, there is a 20% surge in the prices of the maize because the government promotes it for the ethanol production. Now, the demand trends for the maize in South and Southeast Asia is increasing, mostly for the starch-based industries, which is the highest, and it is around 3 to 4%. For the food, it is least 1.2, but still the demand is increasing in South and Southeast Asia. Coming to the biotic stresses in Asia, we are having plenty of the diseases for maize here, but the major foliar diseases of maize are the Terticum leaf blight, Medes leaf blight, common rust, Polysora rust, sorghum downy mildew, gray leaf spot, and Curvularia leaf spot. And the major soil-borne diseases of maize are the charcoal rot, Fusarium stalk rot, bacterial rot, and the banded leaf and shade blight. As we know that there is a climate change. The climate change is a fact. Because of the climate change, there is a change in the environment, which has a potential to shift the pathogen dynamics in the different agroecologies. With that change, in the environment, it is predicted that the stalk rot and the ear rot will have a significant impact on the yield of the maize. So linking that, what we need to do is linking the agronomic practices together, breeding for the abiotic stress tolerances, selection for the disease resistance. We will be ensure the yield stability and the climate resilience of maize. So, emergence of the new threats. As Dr. Prasanna has already told you that maize lethal necrosis, it was first observed in the 2011 in Kenya, but then it has widely spread to the other regions in Africa and creating the 100% of the yield loss. Similarly, the tar spot complex, which was a native to the Latin American countries, but now this has been observed in USA also. So pathogens are traveling, they don't need passport, they don't need visa, they are going everywhere where they want and creating a very creating a very less yield. In 2024, it is estimated that 6% of the maize yield reduction in USA is due to the diseases, and 1% is translated to 231 million USDs. In Latin America, it's tar spot. In Asia, it's downy mildews, maize stalk rots and ear rots. In Africa, it's maize lethal necrosis. And in USA, it's a constant expanding towards the pre-production area. So the diseases are occurring everywhere. So this we have also observed the change in the virulence of the strains. What we have in 2002, there were several CMLs that were released as a sorghum downy mildew resistant lines. But when we evaluated them in 2012, it was observed that very few of the lines were resistant and most of them were showing the susceptibility. This is because the pathogen is continuously evolving. And what they are doing is they are overcoming the resistance of the host plant. Similarly, what we have observed recently that there was emergence of the stalk rots in Bangladesh. The BWMRI has noted that in their Kharif season in the northern region of Bangladesh, they have observed that there is some disease occurring on the maize. It's wilting at the later stage. They were not knowing what is it. And in 2022 and 23 also, the same kind of symptoms were observed. The NARS in the Bangladesh speculate that it's a Physarum stalk rot, but they were not sure. There was no systematic study undertaken. Similarly, in my previous organizations, what we have in CIMIT, what we have done is we have observed that in Nepal, there is lots of the ear rot problem. So every time in whichever season, whether it is a summer season planting or the rainy season planting, the ear rot incidence is quite high. And then what we did is that we collected the samples from there and try to do the molecular identification. What we observed is the Physarum verticilloides and Physarum graminearium are the main pathogens, along with the species of Aspergillus flavus, Aspergillus species. Now, why do the plants emerge? The pathogens in why do the plant pathogens emerge? The movement of the pathogens in the infected plant material, extreme weather conditions, plant pathogens may shift to the host and gain the ability to infect the new hosts. A change in the pathogenesis or virulence in an endemic strain can occur, and newly evolved plant pathogen species can also occur through interspecific hybridization or mutation within the existing pathogen. So now how these can be prevented? The disease will occur when there is a pathogen, there is a susceptible host, and there is a favorable environment. But these can be prevented by the regulatory approaches, genetic control, cultural control, biological control, and the chemical control. The maize disease can also be done through preventing the maize disease through the operational management, where the global surveillance systems, which can be a network of the diagnostic regional hub sharing the disease and pest-related pathogen data. And also the integrated disease and pest management, where there is a quarantine and phytosanitary regulation, surveillance, early detections, and then biological control, host plant resistance, then can be help in preventing and managing the plant diseases. Coming to India, the most economically important diseases are the post-flowering stalk rots. Currently, it is the most important constraint, which is caused by the multiple pathogens. It's highly coincident with the abiotic stresses like drought and heat. There are many post-flowering stalk rots, but the most common are the fusarium stalk rots and the charcoal rot. The fusarium stalk rot is caused by the fusarium verticillites, and the charcoal rot is caused by Microphomina faeculina. The tersicum leaf blight, which is a foliar disease, it's an important across all the maize production zones in Asia. Fusarium year rot, which is having a higher impact due to the increased insect pest damage, food and feed quality, and highly impacted. Banded leaf sheath blight, it can cause a complete crop loss, and the downy mildews, which is spread in the many maize production areas across Asia. Now, at CIMMYT, what we used to do is for all the diseases which were economically important, we used to do the precision phenotyping, we used to do the artificial inoculation. From the previously infected disease plants, we used to isolate the pathogen, isolate the pathogens, mass multiply it, and then inoculate in our fields and do the screening. For almost all the economically, so we were using the hotspots plus we were artificially inoculating so that there should not be any escapes. As we know that we were also working on the soil-borne diseases. So for the soil-borne diseases, it's the sick plots is the most acceptable approach for disease evaluation. Again, so at ICRISAT campus, we were having approximately of the 0.5 hectare of the land dedicated for the fusarium stalk rot, and the another half hectare was dedicated to the charcoal rot evaluations. So we have been planting on those from last 8 to 10 years, and they are quite sick. The soil is quite sick. So this was. So this is the pictorial severity scale on the basis of 1 to 9. The disease scale, the 1 is the highly resistant and the 9 is the highly susceptible. And the threshold value what we used to take is 5 or lesser than 5. So now the precision phenotyping of the germplasm in CIMMYT, we were just evaluating the hybrids, stage 2, stage 3 hybrids. We were evaluating the parents of the hybrids and plus the CMLs and the other inbred lines. So because of this artificial inoculation method and the properly taking care of the trials, what we have observed that our heritability, the quality of the data has improved from the time when we have started in 2015 to now. Now, if we'll see, our average heritability of the trials is 0.8, which is quite good, because whatever we do, whether it is a genotyping or marker-resistant selections or Jivas or QTL, the phenotypic data is important. So what we have observed, upward trends were observed in the heritability estimates of the disease trials managed by CIMMYT under artificial inoculation conditions. Now, coming to the host plant resistance to diseases, it can be a qualitative where only one gene is responsible, or quantitative where there is a multiple genes are responsible for the resistance. But in maize breeding, the quantitative is mostly breeders prefer the quantitative because of the diverse nature of maize crop. Now, strategies for the host plant resistance. It is a multi-location phenotyping in collaboration with the partners, population improvement through recurrent selection under disease pressure, trait mapping for discovery, validation, and deployment of the trait markers wherever it is possible, and incorporation of the disease resistant in the product profile-based breeding pipelines. So we were trying in Summit, we were trying to do both the conventional breeding as well as the breeding use the molecular markers. At least we used to study. In this case, we were doing the breeding for post-flowering stalk rods, both for the Fusarium stalk rod as well as for charcoal rod. In this case, what we have developed the multiparent synthetic populations using the two heterotic groups and we were just doing the recurrent selection under disease pressure. And we were deriving the donor lines out of that. And we were incorporating these resistant lines in the breeding pipeline for developing the elite multiple stress tolerant hybrids. Apart from that, we were also improving the existing multiparent populations by incorporating the abiotic stresses like heat and drought. So this is the schematic of how we developed the disease donor lines. In this case, what we were taking is approximately 10 lines which were resistant. We crossed them and produced the F1s. The F1s, then we intermated and make the cycle 0. The cycle 0 was again planted, inoculated. The best lines were taken, again intermated and developing the cycle 2, and either we were just doing it in a conventional way or deriving the DH or deriving the lines to DH. So by doing this from last many years, we have developed some donor lines, and we were used to give these donor lines in an IMIC field day. IMIC is an International Maize Improvement Consortium, and these lines were given to the NARS partners as well as the private sector partners. So now coming to the molecular studies of the diseases, we have done the GWAS for downy, important diseases like downy mildew, Tursicum leaf blight, charcoal rot, and Fusarium stalk rot. We have identified the approximately 26 highly significant SNPs for sorghum downy mildew on the chromosome 1, 2, 3, 5, 6, and 8, where the chromosome 6 was most important because we identified most of the SNPs on that. Plus, we did the GWAS also on for the TLB, and the UAS Dharwad was also one of the partners, and we have published the paper on the genome-wide associations for TLB. In this case, what we have done is this was widely studied. We have used 3 panels, CAM panel, DTMA panel, and the IMAS panel. We have evaluated and identified the regions that were associated with the TLB resistance. Apart from that, we have done it for the charcoal rot, where also we identified the good SNPs, and we have validated those SNPs using the two F23 mapping populations. But the results of the Fusarium stalk rots were quite good, because in that what we identified is approximately 6 of the SNPs were linked together. They were closely linked together, and they were highly significant for this trait. And we validated using 2 F23 mapping populations. The QTLs were also identified at that regions only, approximately at that regions. To see this, we further thought whether we can just develop the possibility of developing the breeder-ready markers. We have developed the bicross nils to validate these QTLs, and we have already done that, and it is at the validation stage. And I hope that this work will be continuing, and we can develop the breeder-ready markers for the Fusarium stalk rot. Because for the traits like the Fusarium stalk rot and the charcoal rot, the doing the phenotyping is very difficult. It's taking a lots of time. Like we are having a 0.5 hectare of the land in the ICRIS campus, and then we used to take 2 to 3 weeks to for the harvesting, because we used to cut each of the plant, split it, and then score each and every plant. So it is becoming tedious for the breeder and for the pathologist. If we'll get the breeder-ready markers, that will be quite useful. Now, this was the study, this was the aspect of the resistance, host plant resistance. Now, coming to the post-flowering stalk rot variation, the species, what we have done is we have in Asia, we have done that studies, and in India, we have taken 7 states. Karnataka is also one of the states where we have taken the samples. Approximately 250 disease samples we have collected from the 7 maize growing states in India. We have isolated the pathogens and we have identified using the translation elongation TEF for the identification. What we observed is, as we are knowing that Fusarium verticilloides is the pathogen that is commonly causing the post-flowering disease. That it was a well-known fact. But what we have observed is that there is a one more complex, Fusarium incarnadum equiseti complex is the one which was recently identified in Karnataka itself. It was first time reported in maize to cause the disease in 2020. And while doing the sampling and while doing the analysis, what we have observed is approximately 27% of our samples were having pathogens. And not only in the southern part, they were in the part in Nepal, they were part of in Bihar and Punjab as well. So this is there is a shift. I can say this pathogen is growing a lot. It is spreading, and maybe in the future, it will be much more virulent as compared to the Fusarium verticilloides. We have done a small study, a pilot study, where we observed that incarnatum and the Fusarium incarnatum equiseti complex and the verticilloids are having approximately the similar virulence. But the way it is spreading, I think that it may the virulence may increase. The same studies we have done in Nepal also. In Nepal also, we have identified this. Apart from the other already reported stalk rot pathogens, we have observed this Fusarium incarnate species pathogens from this group. Now, coming to the conclusion is that the effectively encountering the current and the emerging threats in the plant disease requires the holistic approach that includes the global coordination, diagnostic and surveillance system, epidemiological modeling, risk assessment, forecasting, and preparedness for proactive management and containment. Implementation of the context-sensitive, eco-friendly, gender-responsive, and social inclusion, integrating the diseases and pest management approach to reduce the impacts of devastating transboundary and newly emerged pests and disease. Thank you.
Thank you very much, Dr. Zarka. That was an excellent presentation on the complex state of diseases in maize, plus the associated control measures and the breeding approaches and molecular approaches, and the identification on SNPs and QTLs for disease of various diseases. Thank you very much once again, and it's time for a few quick questions. 1 or 2. No questions? Okay, thank you, Zarka, for that presentation, and the audience enjoyed that very much. Thank you very much.
Thank you.
So now we'll have a short tea break for 10 minutes and again reassemble here for the next presentation.
Yes, sir, I'm there.
No question. Thank you very much, and we loved your presentations. Thank you.
Thank you so much, sir. It is already difficult to present after Dr. Prasanna.
No.
It is an additional stress for us.
No, but there was very good insights into the disease resistance breeding. Thank you.
Thank you, sir.
Thank you.
Welcome back for the next presentation. So before we move ahead with the presentation, just for a few minutes, yesterday we have missed felicitating of two sponsors. I request them to please come on the dais and receive the honor from our Honorable Vice Chancellor, sir, for generously supporting in organizing this national conference. One is from, I call upon a representative from Koteva Agri-Science, and next also a representative from Rallis India Limited. I request audience to have a big round of applause for their generous support in organizing this national conference. Koteva Agri-Science Limited. Next we have Shiva Prasad sir from Rallis India Limited. Please. Thank you so much, sir. Anybody from NABARD? Any representative from NABARD in the audience? Okay. Sir. Thank you. Yes. Next. We have a last presentation from a last lead presentation in this session. I call upon Dr. Subi SB Madam, entomologist, IIMR, New Delhi.
Thank you. Honorable Chairman of the session, VC sir, and the honorable organizers, the distinguished speakers, and dear student friends. And we will see how we are redefining the breeding program after invasion of fall armyworm in India. So the major insect pest of maize and their damage you can see in the slide. We have spotted stem borer in the kharif, pink stem borer in the rabi, and fall armyworm. It is in the kharif and rabi maize, and we have the shoot fly. It is a very endemic. It is a local pest. It comes in spring maize in northwest India, and spring maize is very important in northwest India, and the rabi maize in peninsular India, also in the eastern India, and the kharif maize mostly in the central and northwestern India. And we have other minor pests also. But these are the pests which we need a breeding intervention. And you can see the Chylopatelis, its infestation is range from 5.33 to 52%, which we have recorded. And in pink stem borer, but it can cause around 20 to 45% loss, Chylopatelis, because it is a stem borer. The stalk borers kill the plant, but fall armyworm not. And the shoot fly, which it can cause up to 100% infestation when the maize sowing is done after 14th of February in the northwestern India, especially in Punjab, Haryana, and the western UP, and also in some part of Uttarakhand, and it can cause 100% infestation, and the yield loss is 28 to 45%. And the host plant resistance. So actually, the shoot fly comes when the maize is around 2-leaf stage, around in a single 2 days after germination, the shoot fly is coming. So the seed treatment is the best option. We have a very good seed treatment molecule, and the breeding intervention is done. Okay. The voice is coming now. The breeding intervention we required in the case of stem borers and fall armyworm. And the invasion of fall armyworm, which has dramatically reshaped the maize pest landscape. And you can see it is this we have recorded in a conservation agriculture plot in Delhi. And you can see the foliar infestation in 2018 and 2019, because when you go in the field, you will see only the symptom. The pest will be hiding in the wall. It's the same case in the case of fall armyworm, also the stem borers. And it ranges around 26 and 36%. In 2020, the foliar infestation is above 90%, because fall armyworm came in India in 2018, and in northwest India it came in 2019, because fall armyworm is a pest. It cannot hibernate. It won't slow down or it die in extreme winter, especially in the northwestern India. So it will die off. We are not getting any infestation in this late Rabi. Getting, yes, some infestation we are getting. And if we can see the tassel infestation, it won't cause an economic loss. Chylopatulus, unlike, one more thing is that we did not require any breeding intervention earlier in the case of Indian stem borers, Chylopatulus and Sesamea inferens, unlike the Ostrenia nubulalis in the Europe, and the European corn borer, and they have the western corn rootworm, and also African stem borer. They are the BT. They are the very devastating pest, and in the Indian native stem borer, that we have to think why, because the maize originated there, so they evolved there, and it came very late here. So our stem borers in the millets got adapted to maize, so that could be the cause. They were not causing much damage, and after introduction of the single cross hybrids, they hardly they require even one spray. But after the invasion of the fall armyworm, the farmers in the south, they are spraying 2 spray minimum, and in the north, they are spraying at least 4, especially in the eastern India. And when in the Punjab farmers, when they put it in the Punjab, they are into rice. IMR is trying to diversify the cropping system in Punjab. We want, because of the groundwater depletion and other stress on other rare resources. Okay, thank you. So that, is it okay? Voice, volume, okay. So, ear infestation, the stem borers never cause, I have never seen stem borer causing infestation in the ear. But it has been reported in African borers, also in the European corn borer, also in Asian corn borer, Ostrenia farnicalis. But we have traveled across India. It is very rare and never seen. But but we had the ear infestation by Helicoverpa armigera, and that was very meager, and they eat from it is a fruit borer originally, polyphagous pest. They eat only from the top, they enter from the top. But Spodoptera frugiparida, they can enter from anywhere. So it is really so we have to look into breeding strategy, we have to change. So you can see how there are two pests. In the previous slide, we saw Mithimna lorei also. Mithimna lorei is a minor pest in the northwestern India. And the Mithimna, its counterpart, Mithimna separata, is minor in the South India, also in the eastern India. This Methymna lorei come in the wheat in large number, but they don't cause economic damage. It comes in the maize, and sometimes they damage the ear. Mostly they eat the earlet of the corn ear. So when the pest came in 2018, we started monitoring using the pheromone trap, and you can see the Methymna lorei, actually the same pheromone trap is attracting both the pest. It is because of something with their biology, not the problem with the lure. And we are getting two pests in the two peak, one in Kharif and in the early spring and in the late Rabi. You can see the Methymna lorei and the Spodoptera frugiparida. It's in the Kharif. Typically the sowing happens in July. So we get, when around second week of July, we get a large population of fall armyworm flying from east, and the 100% of the plants will be infested. So after the arrival of fall armyworm, this Methymna also came down. And this is the geographical variation in fall armyworm infestation. You can see our AACRP partners. They are the one who recorded this data across the country, and the maximum fall armyworm recorded in Pantnagar, then Delhi, in Udaipur. It's in the northwestern India. But fortunately, we don't have much of the Kharif maize in northwestern India. As soon as we diversify, we go into maize, it can really cause an issue. So it is recorded in our field, 100% of the plants were infested. It is our trial block of the ACRP trials, that every plant and the underside of the leaf of a 1-2-day-old maize plants were laid with fall armyworm eggs. And we know the northwestern India, everything works with the degree days. Your plant grows fast and the insect grows fast, so they eat. They cause very high damage in the northwestern India, which we have seen in the plot. So is the fall armyworm in India is same as in the country of origin? In the country of origin, we have two strains, the corn strain and the rice strain. But in India, it came into Africa in 2016 and India in 2018. And in both the places, it has been established that we have a hybrid strain, but it is adapted to corn. It do not cause any damage in rice, paddy. In the IRA campus, around 40 acres of the maize will be there. Then around the 20 acres may be paddy, then sorghum, millets are there. But they are infesting only maize. So if somebody is saying it is infesting rice, not. It comes in sugarcane a bit, but it is not causing economic damage. And you can see that we have done, when the pest came, the first reaction of the government was to extend the label claim of some of the pesticides, because this pest is known for carrying resistance against the first generation and pesticides, OP and carbamates. So we have, and 3 mutations has been reported earlier, and in the 2 mutations we have reported in India. Also their susceptibility varies, and you can see that the north and the south population, they cluster differently. So what we have in North India is a much hardy than in south. And also we can see that when you check with the 2 markers, COI and TPI, and when you use the COI, 90% show that it is rice strain, but TPI, it is corn strain. And when you go to the heterozygous, when you go to the pattern, we can see that there are heterozygotes, so they are segregating. Maybe the chance of evolving into a rice strain is very less, because we are not seeing any infestation. They are maintaining that Hardy-Weinberg equilibrium. Then why we have to go for the houseplant resistance? Because maize is a part of a larger ecosystem, and when we spray very hard chemicals, and it can affect the other species also. And also it is a most sustainable one. So everyone told the importance of germplasm screening, and the screening is by visual damage rating. The fall armyworm, it is not killing the plant, so we use the visual damage. It is not going into the stem, so we see as per the severity of leaf feeding, defoliation. But if you go into the stem borers, they feed for a week. They feed only for a week in the top wall, they exit the wall and they penetrate through the stem. So it is most, the important to see how the stalk strength is. So we were using only the visual damage screening till, and we tried how we can improve this phenotyping technique. The other, also the foliar resistance, also the stalk resistance. So this can we use the NDVI for measuring, because NDVI will tell how much is the green matter is there in the plant. So we use the NDVI. Yes, it is working. And we usually take 21 days after infestation, we score in the case of stem borer. It is in 2019. One more thing is that I'm doing artificial infestation in the open field in an isolated location in Delhi, and my colleague, she's doing in a net house in Hyderabad, and she's doing in Rabi, and I'm doing it in Kharif. So my crop undergo multiple stresses. We will see how we can evolve better germplasm under multiple stresses. So here it is, we can use the NDVI, and in that, but it may not, it can distinguish the highly susceptible and resistant genotype, but when we put the moderately tolerant, like hybrid, the NDVI may not work because it is not distinguishing the subtle differences between the germplasm. So it is only an augmenting tool. So you can see that how the NDVI and LIR, the leaf damage rating, how it can complement the germplasm screening score. Then how the visual screening of stem borer can be augmented by a digital tool in that, because the stem borer penetrate. Actually, this penetration resistance is originally used for the stalk strength in Americas. So we tried it using a texture analyzer we have measured, and yes, it is correlating. So the stalk strength is important in the case of stem borer resistance. And we have identified 2 genotypes, and we have gone for metabolomics and transcriptomics also in the case of Chylopatulus. So in the coming slides, we will see. So can the optical sensing can improve the fall armyworm screen and the phenotyping, and they are in crosses. We have tried some crosses, and they put it in the field. And you can see that the Kylo LIR, it is a stem borer LIR, is very much in opposite to the NDVI. Yes, we can. And also from this, we can see that the Kylo is a much yield reducing factor than for fall armyworm. But fortunately, fall armyworm comes in 100% of the plants, and Kylo comes only in some 10 to 15% of the plants only. So even though it is a very yield reducing factor, its infestation level is very low. And after the invasion of fall armyworm, it is undetected in the plant. Sometimes it is 1 to 2%. In inbred line, we can get much more discrimination by using optical sensing. But it is a baby step. It is only using a handheld green seeker device, but it shows that it can be used for high throughput phenotyping using the aerial UAVs. And then we used only the vegetative state stress still for stem borers, but fall armyworm, they comes in the reproductive stage. Actually, these are the 2 contrasting phenotypes, 2 contrasting inbred lines. And we have in that, you can see the year of 3 elite inbred lines used in the breeding. They are in almost 50-60% of the hybrids released from IAMR and IRA. It is HK163, HK1105, and HK193 193-2-1, and they have been used for introgressing vitamin E and vitamin A also, genes also by IRA. And if you can see that, when we spray, you will get the complete protection. When we don't spray, HK1105 is not much affected, but the other genotypes are affected. So reproductive stress and reproductive state screening and this leaf damage screening is equally important. But so far, we have been using reproductive, this vegetative state screening only. So how the genotypes perform under multiple stresses. So we are so fortunate, we got some lines from CIMMIT, fall armyworm resistant designated lines, and they are the CML. You can see the CML 60, those CMLs, they are having good, low leaf damage. They are very good at vegetative stage, but when it comes to the seed production point of view, at the end we have to see how the company or the breeders will be accepting your line. Because every year we are releasing lines at 10% yield advantage. Also, I have seen in the Advantage presentation, in the last 11 years, from 2011 to 2023, the North India got 118 kilogram per hectare of increase, yield increase happened. The gain happened. So a breeder need a agronomically superior line. So you can see that those lines, they are not agronomically superior, but under multiple stresses, their grain yield, if you see, they are not good at foliage level, foliage vegetative state stress, but in the reproductive stage stress, how much they can produce the grain. Yes, we have, but they are not on par with the giant Dr. B.M. Prasanna has shown. So on the CIMIT lines, but yes, they are able to tolerate the multiple stresses. But one more thing you can see here is that when it is infested with actually this Kylo-1 control, actually, one more thing for the pathologist, when it is infested by the Chylopatulus, it is causing stalk rot also. It is an inducer of stalk rot because it is causing an injury. It can cause the entry of the pathogen. Like the insect wounding often facilitate the entry of the pathogen. That is for the fall also, stalk rot. That ear rot, it can facilitate the ear rot. So in 2024, screening again, you can see that we got the CML lines. Also, we have a line called E63. This DMR E63 is a line identified by Drs. J.C. Shekhar in 2007 and 8 before we joined. And that line is still not being used by the breeders. Why, we will see in the coming slides. Because it is a very good, it is showing resistance, but it is agronomically inferior. So we have to transfer its traits to other agronomically superior lines. Yeah. So it is in collaboration with NIPGR. It is under publication. So it is a genetic trade-off between yield and defense. So we know that this pathogen resistance, especially this not necrotrophic, the other kind, it work in the defense mechanism is activated by salicylic acid, but in the case of chewing pest, it is activated by jasmonic acid. So when the jasmonic acid pathway, when it started, your downstream genes, so many genes will be there, and in that they will, in maize, the major is benzosuccinoids. And you can see in the slide that it is our BML6 is susceptible for all. It is susceptible to stem borer, it is susceptible to TLB, it is susceptible to fall armyworm also. So, but when we tell to the breeders, yeah, it is a very good campaigner. It will give a good heterosis. So this BML6, all the jasmonic acid and the jasmonyl isoleucine, it is the next step in the jasmonic acid pathway. It is down-regulated. But in the case of CML71 and DMR-E63, it is up-regulated. It is more in the case of DMR-63 because its basal level of jasmonic acid itself is higher. And you can see that benzosuccinoids, you can see, except the hydration product, that the product with the detoxification product, it is OH BOA. Okay. Thank you. So From the picture, the BML6, it do not have any kind of resistance mechanism to the major pest and the TLB. So you can see it is a vigorous plant, and the E63 has so much of thing activated. So when you wield so many shields, your reproductive capacity will be coming low. So that's how the E63 is not used by the breeders. So we are trying to cross it with the CIMMIT lines, and in that our breeder, Dr. K.R. Yadish, he has made the E63 into CML336 crosses. No, actually it is CML33. No, it is actually CML71, and he has taken into DH facility, so he has some DH lines, and it has been screened, and he has identified. We have identified 2 resistant lines from the crosses. And it is a QTL identification through the BSA. So from this E63 and BML6, it is a resistant and susceptible line. You can see that we got 2 putative marker, and in that the breeders know better. The lot score is good only for the BNLG619. It is not enough to use these markers for screening for insect resistance, because the internationally minimum 4 or 5 QTLs are reported. Based on one, we cannot. So we are going ahead with the other crosses. So it is a vegetative stage resistance to Chylopatelus. Yeah, BML6 and BML7. BML6 is very susceptible. BML7 has a very good stock resistance. So it has a ruling hybrid. It is DHM117. So in that, the BML6 and BML7, how the BML6 is it is a susceptible one. When the insect attack it, it goes for the sucrose synthase gene is activated. But the BML7, it is more going into cell wall strengthening, like cellulose synthase, shikimate kinase. And you can see in that linolenic acid metabolism is activated. Linolylate 9S lipoxygenase, and it is before the JA synthesis. Then the terpene synthase is downstream to the JA pathway. The terpene synthase and terpene synthase 4. Actually, there are 2 kinds. And also we can get the terpene synthase 10. And this terpene synthase 4, one is the beta caryophyllin, Dr. B.M. Prasanna told in his presentation. So these 3 terpene synthase, you can see they are upregulated. And this is based on RT-PCR. And also after the transcriptome, we have identified, and that is based on RT-PCR. And we have gone for the metabolome also, including its hybrid. So in the metabolome, we can see that coumarin and GA, you can see that coumarin and coumaric acid. So in the morning presentation, I have seen Dr. Suntaram. He told Dr. Padma Kumari and another colleague, they made a cross between a stem borer resistant and susceptible line, and they identified a QTL. And in that QTL, he has mentioned 4 coumaryl transferase. Actually, it is exactly that gene which is making coumaric acid, P-coumaric acid from coumarin. So that you can see that it is higher in the hybrid, also in the resistant parent. So what is next in the induced defense? Dr. Prasanna told we cannot. These compounds are repelling. They are not doing. Dr. Krishna Kumar, he asked a question whether it is when we antibiosis work, how it can the human consumption, the taste for food and feed can change. Then he told no. They are repelling. So that comes in induced resistance. So we got a patent for an instrument which phenotype, which can collect the volatiles to phenotype. It is now being established in NIPSAM and IERR. NIPSAM has published some papers using the instrument. For doing this, I have again modified it. And now it is a 2025-26 ongoing research. You can see there are some resistant, also some susceptible genotype. And in that, we identified one compound, not all compounds are not there, but based on one compound, how the other compounds are performing. So in that you can see that when in the 0 hour, the volatile profile is entirely different than the 24 and 48 hours. So now why IPM? So this, our breeders want agronomically superior line. They may not be resistant to pest, but we have to look for moderate resistance. So you can see that which is the yield reducing stage. See the number when the plant is at the 6 leaf stage, the number of husk is coming down. Also the number of husk, also the grain filling, that grain yield per plant is coming down. Also the MLB, the grain yield is highly affected by Maize leaf blight, because when we screen in Delhi, at the end we experience high Maize leaf blight. So it is one experiment. You can see the completely protected. These are the elite genotype germplasm of in the maize program, which has been widely utilized. At least 50-60% of the hybrid has any one of them as parent. So they are completely protected state is a T4 and zero protection is a T3. When you see the genotype HK1105, it is almost the same. There is no difference between T3 and the T4. So here comes the role of host plant resistance. But when we stage the vegetative state screening, this HK1105 is coming in moderately susceptible category, but it has a high foliage compensation. So we have to look for those kind of hybrids. And you can see the BML6. BML6 is completely susceptible. In the T4, we are getting some grains, in the other cases not. So it is a combination of insect, fall armyworm, and the MLB. So how we can deploy with IPM? So we have moved ahead with the next generation management. It is again in collaboration with NIPGR. We got the RNAi also. So we got another project with the DBT to scale up the RNAi technology. But it is, I would say, it is more of an academic. We have the botanicals also, and we have a very good endomopathogenic nematode also. And yeah, it is we developed in collaboration with IRA. We got a novel drone delivered formulation also. So in that we will put around, you need only 20 liters to spray in 1 hectare in the drone when it moves around 4 meters per second, and it has, and we need around 700 gram. We'll be putting into 20 liters of water, and it is a very heavy. So there is less spray drift, and it is the latest technology. So we need to increase the growth rate 3 to 4 times to meet the E20 and the E30, and IMR is into that. And so we have to balance between the moderate resistance and with plants with high vigor and grain yield, which the breeders prefer. So that's maybe the reason that the insect resistance breeding has not moved forward, because we have to balance between the vigor, grain yield, also the susceptibility, and also the multiple stresses. So IPM has a huge role. So it is how we can summarize it, pre-breeding. And the stresses we have to look for is both the stalk strength, also the ear vegetative stage resistance, also ear stage resistance. We can use a wild parent. Some of our breeders, they are crossing with the Teosinte also. Then we have to deploy it with the IPM. Thank you. IMR, thank you all. And thank you for the opportunity. I learned so much in this presentation, in this 2 days session. Thank you, sir, for particularly. Thank you.
Thank you, Dr. Subi, for that overall presentation, giving the status of insect damage in the beginning, and then went on to looking at the genotypes with various stress tolerances, and ultimately leading to kind of a designated strategy to develop resistance. Thank you very much. It's open to questions from the audience. Yes, sir.
Good work.
Thank you, sir.
one or two quick suggestions which I would like you to be on the. We would like to catalog the aphid species coming on maize.
Okay.
Because, and also the thrip species coming on maize.
Okay, sir.
God forbid if tomorrow maize lethal chlorosis makes an entry.
Okay.
We should have a mode of action. We cannot be totally dependent on cement lines will perform here or anything.
Yes.
So I would like normally what we do, we should anticipate an invasion and be prepared for it. So the ideal method of an invasive is we call epicenter, place around the epicenter, and then another radial. So you should develop a mechanism, and in your IIMR every year you should indicate through your coordinated cell. Anybody notices these symptoms should immediately get reported, because we all really fumbled with regard to the fall armyworm. Fault of it is also with the scientists. Nobody wanted to, everybody wanted to hide, saying that they should be the first publisher, and the moth happily kept flying 200 kilometers every day. And this is, am I right?
Sir, even though IMR, most of the ACRP part is recorded.
You are right or wrong?
Yes, sir. But
I am right on the wrong side.
Sir, you are right.
Wrong
sir.
on the right side.
Sir,
Anyhow, it's okay.
Right.
Jokes apart. The second species that I want you to do is thrips.
Okay.
Because thrips parvispinus and other species of thrips, they also have reported Frankliniella occidentalis. If you are not able to identify Frankliniella and different species of aphids, please get it yourself trained, so that we should be exactly in a position to tell what exactly is happening.
Okay, sir.
The second one is fall armyworm we have blown out of proportion. I am not agreeing that it is a major limitation, because one is it is only for 40 days.
No, sir.
45 days, 50 days, take it.
Sir,
Okay.
it is in South India, but in North it comes late, and you can see the ear is completely damaged and lost in some of
all
the
things said and
genotypes.
done.
Yes.
All things said and done. The pest can be managed at this particular stage.
Yes, sir.
It's not something which is, if the rainfall is good, humidity is good, you have a very low incidence. Under dry belt, it can become very serious. But we have to give the due weightage.
Yes.
Yesterday, we made a statement that host plant resistance for insects is not ideal. More so if it is a Lepidopterous pest. When the other options are open, like Metarhizium anisopliae, you have also entomopathogenic nematodes. You can use a small oral of BT. All these things are very comfortably can be used to sustainable management of fall armyworm, because the day we discovered carbofuran, one small carbofuran granule going into the oral of the jawar or maize, we had an answer for the sorghum shoot fly.
Yeah.
So the same thing will operate even for the fall armyworm. You have a screening, but host plant resistance for insect, especially borers, has never been successful in the world anywhere. This is what we told yesterday. And you can have a genotype, but use the lesser of the options with regard to the management. But my only thing is, the management of fall armyworm in baby corn and sweet corn and other commercial crops is a different kettle of fish.
Yes.
You have large areas coming very close to between Delhi and Karnal.
Yeah, true.
Large farmers are growing baby corn and sweet corn, very close to Kurukshetra and such, that belt.
True, sir.
And I think you should really interact with those farmers and
yes,
see
sir.
what best can be done with regard to management of fall armyworm on a crop which is economically much, much more important.
True,
Thank you.
Thank you, sir.
Hello. Just a question. It's not a question, actually.
Okay.
You have mentioned the fall armyworm and EPN.
APN.
I am from the private sector, Nirmal Seeds. So we have developed a EMN product, EPN product that is Sniper.
Okay.
And it's doing well on a fall armyworm in our area.
Okay.
That's the submission actually.
Okay. Thank you, sir. I'll contact you. Oh, yeah.
Make a EPN suspension, put it into the oral.
So thank you very much, Dr. Subi.
We take this opportunity to extend our heartfelt thanks to our learned speaker, Dr. Subi Madam. I request P. Krishna sir to do the honors to the speaker. Center. Oral. Now we'll have oral presentations. We have 3 oral presentations. Sir.
Okay, the first presentation is on rapid routes to reproduction, speed breeding for maize improvement, going to be delivered by Sinchana MR.
Good evening, everyone. I'm here to present on rapid routes to reproduction, speed breeding for maize improvement. As we all know that the climatic shifts are bringing in new pests and diseases, and we need to breed for stress-resilient cultivars. And the scientists all over the world, they came up with the idea of speed breeding, as we cannot rely upon the conventional breeding approaches, as it takes longer time for breeding. And hence, the speed breeding, it is manipulating the temperature and light intensity and quality to reduce the time taken for generation of the generation interval. And but there are few disadvantages to the speed breeding. That is, there are few disadvantages for the speed breeding, as we cannot rely upon the speed breeding facilities because it is not affordable by many institutes, as it involves investment of costly speed breeding facilities. Also, for a crop like maize, where it where it has a tall stature and dense canopy, it has limited applicability in the speed breeding chambers. Also, in the plant breeding experiments where we need to maintain large breeding populations, it is difficult and it is tedious process to maintain these in the speed breeding chambers. So to address the gap between the concept and technology of speed breeding and its applicability in the field conditions, we have developed a novel technology, that is natural speed breeding in maize. So in AICRP maize, we have developed this cost-effective and time-saving speed breeding strategy, wherein we can apply this technology in the natural field conditions. So this is the first report on the speed breeding in maize, which can be done in the field conditions, and it involves no additional cost other than maintaining the crop in the field. Also, we are using early harvesting as a technique for the speeding process. So the materials and methods include, we have used 7 inbreds and tested these in 49 treatments for germination percentage and seedling vigor. And we have tested first by harvesting it at 85 days. But since we did not get sufficient germination percentage at this stage, we have started from 90 days up till 120 days with the interval of 5 days. So we have a treatment set of 7 treatments. And since in the conventional breeding approaches, we store the seeds for a brief period of time before planting it in the field, we have simulated the same in the lab experiment, and we have stored the seeds of all the 7 different treatments and for a period of from 0 days to 30 days after harvest. So it makes a total of 49 treatments. So after testing all the 7 inbreds in these 49 treatments, we have crossed these inbreds and produced the hybrids, and the F1 population derived from these inbreds have been tested for the same set of 49 treatments. So the results were as follows. These are the representative photos of one of the inbred line and one hybrid line, wherein we can see that the 90 and 95 days after planting hybrids and inbreds have shown sufficient germination percentage and also seedling vigor, which is on par with that of the control, and it provides a scope for the exploration of this novel technology in the speed breeding process. So when the graph was plotted for the germination percentage of these inbred lines, which was harvested at 90 and 95 days after planting, and it was replanted at different days after harvest, we can see that in the 0 days after harvest treatment in both 90 and 95 days after planting, it has given a reduced germination percentage of about 13%. But when these seeds were stored for 5 to 10 days, it has given a germination percentage of about 50% in case of 5 days after planting, and about 70 to 80% germination percentage in case of 10 days after planting. So it is reliable. And when the seedling vigor of GI 755 was tested, we can see that the root length and the shoot length of the 10 days after harvest treatments has given almost 9 to 11 centimeter root length and shoot length. So coming to the germination percentage of the hybrids tested at the 90 and 95 days after planting treatment and replanting at different days of harvest. So here we can see it has given a better response compared to that of the inbreds. We can see that the 0 days after harvest treatment also has given a sufficient germination percentage of about 70%. So in hybrids, we can harvest the seeds at 90 and 95 days after planting, and we can go for replanting on the same day of harvest, and we can save additional 5 days compared to that of the inbreds. When the seedling vigor of the GH21547 was tested, so it showed a root length and shoot length of about 10 to 12 centimeter, and it was appreciable. Once we have tested for both the inbreds and hybrids, we have conducted the same experiment in the next season again to confirm the results. And we have done this in 3 hybrids, that is GH21547, 150, 125, and DKC 9133. And this has given the same results as that of in the first season. That is, the 90 and 95 days after planting seedlings have given sufficient germination percentage and also seedling vigor, and it was on par with that of the control. So when the germination percentage of the hybrids were harvested, which was harvested at different days after planting and replanted at 10 days after harvest was seen, the germination percentage was about 95 to 100%, and the seedling vigor was about, the shoot length was about 11 centimeter, and the root length was about 8 to 9 centimeter. So this has given us positive results, and we can explore this novel technology for speed breeding in natural conditions. So once we have tested the germination percentage and seedling vigor, we have tested the root architecture of all the 3 hybrids and 2 inbreds. So within the root scan, Windriser Root Scanner 2021, and we have these are the images of the 3 hybrids, and we can see that the 90 and 95 days after planting roots has no significant difference compared to that of the control. So we can say that by using early harvesting as a technique in speed breeding in natural conditions, it has no negative impact on the root architecture of the plants. So the same results was obtained in case of the inbred lines also, and there was no negative impact on the root architecture compared to the control treatment. Since all the lab experiments that we do in the lab will be implemented on the field by the by the breeders, we have tested the same experiment in the field also. The field germination of the hybrids and inbreds, which are harvested at different days after planting and replanted at 10 days after harvest and 0 days after harvest, was seen, and the germination percentage of 0 days after harvest has given about 30%. But when these seeds were stored for a brief period of 10 days after harvest, it has given a germination percentage of 60 to 70%. And also when in the inbreds, it has given a germination percentage of 80%. So it has shown that not only in the field, but in the lab, but also in the field, we have got the positive results. These are the field germination photos of different genotypes harvested at different dates and replanted at 10 days after harvest. So, and it was seen at 8 days, the germination was seen after 8 days after planting, and sufficient germination was seen. So, there may be a fear that will there be any loss in the breeding material due to the reduced germination percentage. There might be a loss of breeding material, a slight loss of breeding material, but we can overcome this loss by maintaining large breeding population compared to the original one. The vigor of the plants at 25 days after sowing can be seen, and it can be said that due to the early harvesting, the vigor of the plants is not affected. And the morphological traits of the hybrids in the field were also tested. The ear height and the plant height were tested, and a ear height of about 45 centimeter was seen in 90 and 95 days after planting treatment, and plant height of about 145 to 146 centimeter was seen in the 90 and 95 days treatment. So it was there was a slight decrease in the compared to the control, but this will not affect in the rapid generation advancement or the ear bearing capacity of the plants in comparison to those plants that are grown in the speed breeding facilities. These are the GH21547 hybrids after 62 days after sowing, and across the 7 treatments, and in comparison with that of the control. These are the GH150-125 hybrids at 62 days after sowing, and these are DKC9133 hybrids. And the morphological traits of inbreds were also tested, and the ear height and the plant height of the inbreds were almost on par with that of the control, and this also has given positive results. So in conclusion, I would like to say that the speed breeding facilities has shown successful results in case of in speed breeding facilities, but it requires additional investment cost. It requires investment cost of about 3 crores, and also maintenance charges of maintaining the speed breeding facilities. But when we use the speed breeding, natural speed breeding technology, there is no additional cost apart from the cost that is required for maintaining these plants in the field. Also, when we harvest the seeds at 90 and 95 days after planting, and when we store the seeds for about 5 to 10 days, we are able to achieve a germination percentage of about 80 to 95%, and we are able to save up to 1 month in one generation, and we are able to achieve 3.6 generations per year. So this provides a scope for the exploration of this novel technology in the speed breeding process for rapid generation advancement of these hybrids and inbreds. So by following this methodology, we can overcome the shortcomings of the conventional breeding approach and also the challenges of the speed breeding facilities. Thank you.
Thank you, Sinchana, for that brief presentation that you have made. Now it's open to questions from the audience. Yes, please.
It was a very nice presentation, actually. There are 2 queries, actually. What was the moisture percentage at the time of harvesting of the grains that you have harvested at 90 days and 95 days? And second one is there any significance of shank color to the early germination in the inbreds you have selected for the experiment?
Sir, we have not tested the moisture percentage, but the 90 and 95 days after harvest, genotypes will, of course, have a moisture percentage greater than that of that 100 days, other than stored seeds. And about the shank, I have not much idea about that, sir.
Okay, thank you.
Anybody else? Okay, if there are no further questions, thank you.
Thank you, sir.
The next presentation is on genetic variability for reaction to fall armyworm among newly developed inbred lines of maize under natural condition. This will be presented by Roshni KB.
A very good evening to everyone present over here. I am Kalandar Bibi Roshni from Department of Genetics and Plant Breeding. I am here to explain our work entitled Genetic Variability for Reaction to Fall Armyworm among Newly Developed Maize Inbred Lines under Natural Conditions. Madhya Pradesh, Karnataka and Maharashtra being a leading producers of maize, the overall total maize production was 35.76 million tons across the India, indicates the economic importance of maize all over the nation, India. The fall armyworm, maize being a primary host for fall armyworm, it was an invasive pest. It causes an immense economic damage. Economic damages such as the leaf damage and cob damage at various stages of the maize, and it has a broad host range around 180 different species including rice, sorghum and also millets. It has a global food security threat, and also it is very difficult to control as it has a rapid migration and reproduction. Coming to the impact of fall armyworm, we can see here 30 to 70% of a leaf damage can cause the plant infestation of 20 to 80%, where we can see the plants affected, and in turn it causes the yield loss of 15 to 50%. Under severe conditions, if we can even find 80% of yield loss. Owing to the mode of infestation of fall armyworm, an adult moth lays eggs on the underneath the maize leaf, which hatches in 2 to 3 days under warm conditions, and which turns into the young larva, which feeds on the surface of the leaf of a maize, and then it causes the various visual symptoms such as frass and window pane, and that mature larva falls into the soil and pupates for 7 to 14 days, and then it turns to the moth, which can migrate and even cause the repeated infestation from generations to generations. When we see the difference between the infestation for Kharif and Rabi season, we can see the Rabi season, the infestation is very high due to there is no heavy rainfall and also the warmer conditions for infestations, and the yield loss is very high during Rabi season. So our main objective of my study was to study the genetic variability and diversity among newly developed maize inbreed lines, which were developed from UAS Dharwad, that is on ASRP on maize, was to study on yield and genetic variability and diversity for yield and amylose content. During Rabi season, we found, offshoot of our experiment, we found that fall armyworm infestation under high pest pressure, we found some promising lines which was showing the various response to the fall armyworm infestation. So we have planned to record the responses during Rabi season under natural conditions with true prophylactic sprays. We can see with 5 prophylactic sprays only we can't control the pest incidence, but we have seen the promising genotypes which can be performed well only with 2 prophylactic sprays. We have used the scale which was developed by Davis and Williams, which was later modified by Simmet, which consists of scores between ranging 1 to 9 and responses of from highly resistant to highly susceptible with damaging symptoms. With the help of entomologist Dr. Shaila ma'am, we have recorded the damaging symptoms, and these were the plant phenotypes which were showing the frass symptoms, and some lines were also performing very well. And then we had 90 inbreed lines with including 5 checks, and we had a susceptible check also, CM202. In the Rabi season, we have evaluated at ASRP on maize under natural conditions. Analysis of variance, we found that the treatment significance for treatment was we found the significant difference among the treatments, indicating the variations present among the genotypes, and we found no significance between the replications, which indicates the uniformity of those lines. And looking into the distribution of pest reaction, score of maize germplasm lines against fall armyworm, we found the bell-shaped normal distribution curve with a mean score of 3.2. We found most of the genotypes showing a moderate reaction, while few lines showed lower and highest response to fall armyworm infestation, which suggests the polygenic control of these traits. Descriptive analysis of this experiment, we found that moderate variance with slightly positive skewness with leptokurtic kurtosis which suggests that few major genes were governed rather than many genes. And we classified these lines based on the scoring developed by Davis and Williams. And then we classified our 90 genotypes ranging from 1 to 9 scores. We found that 47 inbred lines fell into the 2 to 3 damage score with the category of resistance, and we found 41 genotypes which were found to be having 4 to 5 leaf damage scores with partial susceptibility. We didn't found any highly resistant or highly susceptible, but we found lines with partial susceptible and more of resistance. This is the pie chart indicating the distribution of genotypes for fall armyworm. And we found that promising genotypes which were resistant with a score of 2 and meanwhile they are having the grain yield of 40 quintal per hectare. The 3 lines we found GI23308, GI23206 and GI23216. So finally, I conclude that this is initial information among newly developed inbreed lines, which we can further evaluate for fall armyworm resistance. And the lines which were promising, which were identified, could be further evaluated under the artificial epitophytic conditions for further confirmation and use. Thank you.
Okay, thank you for the presentation. It's open to the audience for questions. Okay, there are no questions. Thank you very much. The next presentation is unraveling the prevalence of Java Downy mildew disease of maize in northern Karnataka through morphological and molecular identification by Dr. Prema.
Good afternoon, everyone, respected chairman, seniors, and all the scientific fraternity assembled over here. So I'm going to present on the topic unraveling the prevalence of Java Downy mildew disease of maize in northern Karnataka through morphological and molecular identification. It's not working. So coming to the importance of maize, maize is called as a queen of cereals, and it's a C4 plant which is having wider adaptability. So coming to the major diseases which are prevalent in northern Karnataka, so it is affected by touchscum leaf blight, northern leaf blight, so curvularia leaf spot. Okay, sir. Okay, sir. So coming to different types of downy mildew, we have, so this 8 different types of downy mildew. So that is sorghum downy mildew caused by Pernosclerospora sorghum, Java downy mildew caused by Pernosclerospora Maize, Philippine downy mildew by Pernosclerospora philippiensis, sugarcane downy mildew by Pernosclerospora sakari, brown stripe downy mildew by Scleroptera raceae varieties A, Graminicola downy mildew by Sclerospora graminicola, crazy top by Scleroptera macrospora, and Rajasthan downy mildew. So coming to the morphological differences, here lies a big picture. So in case of Pernosclerospora sorghum, so the sporangia, they will be oval, and in case of Pernosclerospora maedis, wherein the sporangia will be spherical or subspherical in shape, and in case of Pernosclerospora philippiensis, it will be ovoid to cylindrical, and in case of Pernosclerospora saccari, it will be elliptical, and in case of Sclerospora gramineola, so it will be papillate, and in case of Sclerophthora macrospora, it will be lemon shaped, and in Sclerophthora raceae varieties A, it will be oval to cylindrical, and oospores, they will be almost spherical or globose in shape. So these are the certain infection characters wherein either the oospores or sporangia, they help in the as the initial source of inoculum, and most of the downy mildews, they are seed borne, and they propagate or spread through the germ tubes, and lower temperature is more feasible for either the sporangia production or the germination. So this is a crazy top found in few of the germplasm lines caused by Sclerophthora macrospora. So wherein, so the crazy top symptoms are observed, and this is a Rajasthan downy mildew caused by Pernosclerospora heteropogoni, and that one is a weed which harbors Pernosclerospora heteropogoni. And this is sorghum downy mildew, so which is due to Pernosclerospora sorghum. So these are the sporangia as well as the sporangiophores, wherein you can see the sporangia, they are oval shaped, and due to outbreak of epidemic, especially in southern districts of Karnataka like Hassan, Mysore and Mandya, so our team has been formulated to survey the disease, and initially we thought that it is sorghum downy mildew which is due to Pernosclerospora sorghum. With the intention of that, so intensive survey is also taken up in the northern parts of Karnataka, and we found these kind of symptoms like downy growth. Usually downy mildew means downy growth will be there, but on the upper leaf surface also powdery growth is evident, and also half leaf symptoms are observed. Leaves become chlorotic, and stripes or white streaks, so they are also evident. And we can also see half leaf symptom and also the malformed tassel. So when we went for the survey, so an incidence of about 20 to 25% has been witnessed at Hanumanmatti, and these are the symptoms at post-flowering stage, wherein leaves turn like kedige, so that's a fragrant pine like symptoms. So and also Joycerali and Tirumala Devarakoppa, and at Ranebennur, so all these comes under the Haveri district, and at Dharwad also the traces of downy mildew has been witnessed. And when we went for this morphological characteristics assay, we found that the sporangia, they are round or spherical in shape, and based on that, and again you can see the sporangia as well as the sporangiophores, and a Y-shaped branching or dichotomous branching of sporangiophores has been witnessed, and you can also see the sporangia which is embedded within the leaf tissue, and this is the initial formation of the sporangia, and also the zoospores, and the encysted zoospores also. And for proving the pathogenicity, so we have sprayed the sporangial suspension, so by detached leaf assay, wherein the leaves have been sprayed with the sporangial suspension and kept for incubation at 80 to 20 degree. We can see the development of the mycelium as well as the sporangia at 3 days, 5 days, as well as at the 7 days after inoculation. And the reproductive structures like oogonium as well as the anthridium, so which is hiding just behind, and the ruptured sporangia which is releasing the zoospores. And usually these downy mildews, they spread by the germination of sporangia, and that's a oospore which is a resistant spore. And this is a taxonomic position of Pernosclerospora maedis, which belongs to phylum Umycota, class Umyces, order Pernosporales, family Pernosporaceae, and it belongs to species Pernosclerospora, and the species is maedis. And it was first reported by Rasipovsky in the year 1897 as Pernospora maedis, and then it was misidentified as Sclerospora maedis. And in India, it was Butler who identified it. And according to that, mycelium, so it will be straight and sparsely branched, and it will be irregularly branched, and the host for this Java downy mildew are wild sugarcane, loon sorghum, corn, teosinte, tripsacum, euclena, as well as panicidum. And this particular disease is distributed in India, Indonesia, Israel, Japan, Thailand, and in Africa also, South America and Australia. And based on morphology, we want to confirm through molecular means also. So that's why we have gone for ITS primers as well as the LSU primers. So in case of fungi, most commonly we use ITS 1 and 4, which is a highly conserved region. But since it is a oomycete, we went for ITS 6 and 7, and also one of the housekeeping gene like LSU, which will give a clear vision with respect to the evolutionary history. So these are the different accessions taken for the comparative studies with respect to nucleotide sequence identity. And the Hanmanmatti isolate, it showed 10% identity with the Thailand isolates of Pernasclerospora Maize. And Joyceral isolate also, so it showed 10% identity. And Tranabinur isolate also, it showed 10% identity with the Pernasclerospora Maize, gene bank accessions deposited in NCBI. And Tirumala Devarakopa isolate, it showed somewhat less identity of about 98.16. So based on whatever the nuclear type percent identity obtained and the sequence obtained, we constructed a concatenate phylogenetic tree by integrating both ITS as well as the LSU sequences, and we got 4 different clades wherein Scleroptera macrospora, it formed clade 1, Pernasclerospora graminicola, it formed clade 2, and Pernasclerospora saccari philippiensis and sorghum, they fell into clade 3, and all the Pernasclerospora Maize, including the isolates taken from northern parts of Karnataka, they fell into clade 4. So based on the morphological evidences like the spherical shape of sporangia and also the nucleotide based identity and phylogenetic analysis, we confirmed that it is Java downy mildew caused by Pernasclerospora Maize. And I acknowledge university and also our former DR, Dr. B. D. Barada sir, for giving me an opportunity. My students, Malavika and Akshata, Dr. Shalini, and my colleagues from ACRP on Maize. Thank you, sir. One more thing. So these are the last slide. Last. So these are the zoospores, sir, produced by Pernasclerospora Maize. Since students, we asked them to write a pictorial representation in the form of disease cycle, wherein they write zoospores require a thin film of water and they swim for a while, then they will go for encystment. So here you can see the movement of the zoospores in water. Thank you.
Oh, thank you, Dr. Prema, for detailing the Java downy mildew disease causing organism of maize in northern Karnataka. The audience for any questions?
Sir, management in southern parts, especially Hassan, Mysore, Mandya, so there the ecosystem is different, sir. Seed treatment, it is must. There they are using F2 seeds and they are not going for seed treatment. Seed treatment with metalaxyl, that is Epron, and the seed industry. So they are also going for some changes in the polymer. So wherein the chemical sticks to the seed for longer durations so that it will have a better efficacy. And we also thought that maybe metalaxyl resistance has been developed by these oomycetes, but no, sir, it is a different species which has appeared. Sir, we have to go for intercultivation, and usually we recommend FIM. So, but the case of downy mildew is whenever the cattle has been fed by, so this downy mildew affected leaves, when it passes through the gut of the animal, so the oospores will become activated, sir. So that's one thing in increasing the intensity of the disease. And we have to go for crop rotation, deep summer plowing, seed treatment, and chemical management is essential with metalaxyl or with the Ridomil Gold, and profiler is also working well, sir. Sir? Sir, we have other options also. Seed treatment followed by spray gives a better management, sir.
But one recommendation is cattle should not feed.
Yes, sir, because
No, in
that
a
is
lighter vein, don't take it very seriously. But what I'm wanting is, damage appears rather quite severe. And over years, it can also become even little more. And we should have, I would request to the Director of Research, whether this comes under package of practices recommendations in your university for this particular disease. If it is not there, maybe it's time to include it under package of practices. What is the best recommendation for this disease based on some of the initial field trials that they take up in their field. I think that will be a useful suggestion.
Thank you, sir. Thank you very much for the suggestion.
Okay, sir, that will be taken.
Okay. Yeah, thank you very much, Prema. So that brings us to the end of the session on maize. And as you already know that there were 4 lead presentations, one each by Dr. Satish, Dr. Zarkar Subi, and Dr. Prasanna. And there were 3 paper presentations. The 4 lead presentations actually together, they gave us wholesome food on given a wholesome food on the platter to digest and probably develop strategies to enhance the productivity of maize in total. So thanks are due to the organizers for giving me an opportunity to chair the session, and thanks to the rapporteurs, Dr. Prema and Dr. Banu. Okay, I was just thanking both the organizers and the rapporteurs for conducting the session, and thanks to the organizers for giving me the opportunity, and thanks to the rapporteurs for joining me in this particular session. Thanks to the audience for their wonderful cooperation and participation in the whole events. Thank you very much.
Thank you, sir. We sincerely thank our respected chair, sir, for conducting this session very nicely. And we take this opportunity to thank him with a token of love. I request Dr. B.D. Biradar, sir, Director of Education, U.S. Dharwad, to honor our chair, Dr. P. U. Krishna, sir. Thank you, sir. Up next, we have a panel discussion. I request Dr. Surinder Tikku, sir, from ATPBR Pune, to chair this panel discussion. And I request Mr. Shashi Bankar, Testing Manager, Bayer Crop Sciences, Mr. Shambhulinga Shahapur, Lead R&D, Indo-American Hybrid Seeds Private Limited, Dr. Kushailan, Senior Breeder Maize, Rasi Seeds, Mr. Anand Kumar B, Corn Breeder, Koteva, please join as a panelist. And I request rapporteurs of this session to continue to note the proceedings of this panel discussion too. Yes, sir. Yes, sir. The rapporteurs of this maize session are asked to note the proceedings of this panel discussion also. Dr. Prema Jayu and Dr. Harshia Banu.
Good evening. After such a great session, we'll try to do the maximum that we can and as quickly as possible. I welcome my panelists on my... I don't have a paper and the list of the panelists. Can you give me that? So how I'm planning to do this is that we learned a lot during the session. I made some points. So I will kind of throw them at you and your plus anything else you want to add to what happened in the last seven speakers we had. Yeah. Yeah. Okay. Thank you. I just made some notes and I'll go through that. Number one, I want to mention, you see, we have seen so much progress and overview that we got today. We have seen so much of progress in our maize crop, similarly for other crops. Now, I was just looking at the, am I audible? I was looking at the comparative data on yield and acreage of maize compared to China, which is equally populous and equally large area, and USA. USA comparison will always, I mean, they will always be higher. One, they have just one crop a year, and their precision agriculture, we cannot reach that there. So I won't talk about that, but the question is that China, since 1970, they were very close to where we were, but today, their acreage is almost like 33 million metric tons. We are at 11. And their average yield is, now 11, sorry, 8 in the average, and we are at about 3 point something. The main question that I have is that our lab to land still remains lab to land we haven't been able to reach to the last farmer so that they can increase their yield. Our research yields are 11 to 12 tons, and that is similar to what China has. So this would be an area where I would like you to respond. Number one, how do we make the extension reach to the farmers? Are there areas where the farmers should not grow maize? Then the policy should be, how do we give them something else? Because it's not remunerative to them. Number two, you have heard so many areas of work in the biotic and abiotic stress. I would like you to, in your own way, what do you want to add to that? Anything that you think was missed? Then, I didn't hear anybody speak about it today, but there is a lot of work now going on dwarf corn. Dwarfing corn, especially Bayer, I think they have a hybrid, they call it Precision. So what is happening is they have almost like just 4 feet tall maize with smaller corn, but high density. By high density, they are able to get, I think, in my opinion, I think they are quoting at least another 10-15% more yield, right? So this would be, is there scope for us to get into that? At the same time, we are increasing our yields, on research yields are there. What is happening on the nutritional quality of the maize? That would be my other question. The 9 million tons to 11 million tons acreage has happened because it is mostly now because of the biofuel, right? So on that front, what would be your opinions? And lastly, soil health is something that we have had some comments already. Dr. Krishna Kumar was talking about that. Many others said. are we working on, should we work on better fertilizer use efficient genotypes, or should we also work on better fertilizers that have better assimilation rates but can still be called non-toxic? Because there is a lot of biological work going on. So if you will remember that, so I will start with Shashi. Mr. Shashi Banakar, testing manager at Bayer. I think that you should be able, right?
Yeah. Yes, yes. Thanks. Thank you, everyone. Good evening, all. Myself, Shashi Banakar. I am alumni of this UAS Dharwad, and I am representing from Bayer Crop Science Private Limited. Coming to the first question. Sorry, I forgot the question.
Yields.
Yeah, yield levels, if you see year on year, if you see, even we have a contact with the growers. The yield levels are decreasing year on year. This is one is climate change, and the genotypes which we are using, we have to develop more precise genotypes which are adaptable to the periodical, there is a drought occurrence. So which can adapt to that adaptability, that genotypes we have to develop, and we are doing on that. And third, it is almost related to the fourth question. So nutrient as well as water use efficiency, that is not happening, because the right time application of the nutrient as well as the, so that is not happening, the uptake and the root system, that we have to guide the farmers. We should do the work from our side. We are doing whenever we are this one. And farmers, like, we have to, now we see, as you are mentioning China is using more biostimulants. If we recommend to our growers as well as the farmers, that absolutely increase most of your questions. And China is the number one, and they provided. Meanwhile, they stopped providing to the India also. And the fourth question, smart corn, means short corn. We are doing this program since from last 4 years, and we studied most of the, like, to leverage this smart corn. We are about to launch this in the dry area, and because in the south what we are seeing is this geography adaptation to the short corn, we are seeing more of biotic stress. So we are working on that, and we will launch as we need to cope up with the yield levels, China as well as Mexico. Mexico is also giving the good yield levels as compared to China. So we are just working on that. And temperate and tropical germplasm also we are like combining, but it's still we are in the abiotic. Thank you.
Thank you. Good points, and let us. Yeah. Dr. Kuselan, Senior Breeder Maize at Rasi Seeds.
Good evening to all. Myself, Kuselan, Senior Breeder Rasi Seeds. Actually, coming to second question, maize, we have various diseases and pests is there. Major diseases, we have to private company, mostly we have to 4 to 5 diseases only we are working. Especially in TLB, we are going to the some marker yesterday selection also, and hotspot screening, especially in Bangalore and Hassan area, we are going to regularly, whatever we have genotypes, and we have to screen in the hotspot screening in the TLB. In same way, we are screening in the post-flowering stalk rot in Bangalore and Warangal, some of the places in Hassan, like that. More, what are the important diseases? Where is the hotspot screening? Then we have to, multi-location trials is there. Based on that, we have to screen our genotypes, and the tolerance genotypes, we have to take it in the crossing, and further hybrid development program, we are using the screening. Thank you, sir.
I just want to add to that. I think Dr. Prasanna made it clear. And we believe that two things are very important. The strategy of screening and doing your work is very important. Multi-location testing, even at earlier stage in hotspots, so that you get the right genotypes. And then we are doing like 200 lines and all, it will not happen. A lot of these problems will have to screen huge germplasm collections and have the ability. And this is where we are missing PPP. We need to work together to find new sources of germplasm from the huge collections that NBPGR maintains or CEMIT maintains. All this has to come together to find the right germplasm for us. Then we start the breeding and then do the good. Thank you. Now, sorry? Yeah. Dr. Shambhuling Shahpur, lead R&D Indo-American hybrid seeds.
Yeah, good evening to all of you. I'm also alumni of UAS Dharwad. I'm happy to be sharing this panel with all eminent scientists. To start with the first question, why there is a difference, yield gaps between the global maize program and also Indian maize program. The thing is, US and China, yield levels very high because of congenial climatic conditions. They are very long duration crops. The accumulation of GDD is very high.
No. China, half of China is like us.
Yeah, mostly. So mostly it is
Wuhan
like that.
and down. It's all like.
And they are almost advanced precision agriculture is there.
Yeah,
and
that is.
And they are mostly GM. And mostly they are single cross hybrids. When it comes to India, totally different dynamics. Kharif, Rabi, and spring season. The yield levels of, if you compare last decade to, if you see the yield advance what you made is almost 1 ton we added per hectare. Mostly because area expansion and cultivation of maize has grown in Rabi more compared to the Kharif.
Yes.
Kharif yield levels are remaining almost same level as used to be earlier.
Yeah.
The advance and yield gain has happened in Rabi cultivation aspect. And here the challenges are more in Kharif season, erratic rainfall area and very marginal land, marginal farming, small holding. And major challenge, technology transfer is not reached to the marginal farmers.
Yeah, the knowledge transfer is what I am trying to say. It's not happening at the level. Yeah.
Yes, you're right. Because whatever if you recommend a proper population, they will not be able to maintain because one is labor shortage or maybe interference of rains during the cultivation activities. So simple, these technologies, small technology like proper maintenance of population, really bringing down the yield levels in Kharif season. Rabi season, mostly they do proper maintenance, other things, you'll be able to get it. And coming to the diseases, major diseases, as you told, the pest is a major, the fallworm is a major threat. And next is the testicum blight. And the yield gain or area expansion has increased because it has gained a center stage of among all the crops now because of industrialization, ethanol requirement. Government has set a target. By 2030, we have to be reaching 70. We have to produce 72 million tons of maize to cater to the requirement of government set target. So now we have already reached E20, E30 target is there. To reach to that level, government is planning, or IM is planning, all the maize fraternity is planning for the hybrids which have high ethanol, high starch content, amenable for high starch content and also ethanol extraction or ethanol recovery. So I want to caution one thing because ethanol is more, our starch content will be more in soft starch or dent to semi-dent kind of genotypes.
Yes, dent.
And they are more prone for pests and diseases. Actually, they are more hygroscopic in nature. And if you try to cultivate them in Kharif season, which is rainy season, it may attract other diseases like already ear rot is there.
Yeah, that's why dent is not that common in India.
Yes, we want to channelize this.
But dents have more yield.
Yes, dents they should more yield. On introgression of this temperate germplasm into tropical germplasm should be very cautious.
Semi-dense.
Yeah.
Or what is it? Semi-dense is what we are working on.
If you target only yield, temperate germplasm definitely going to yield higher compared to tropical germplasm. There should be optimum balance of tropical and temperate germplasm introgression.
Yes, yes.
So that the sustainable balanced way of yield gain should happen. These are my submissions.
Thank you.
Thank you.
Now we go over to Dr. Anand Kumar, corn breeder at Koteva, right?
Yeah. Thank you very much,
Your
sir, for the opportunity. So I am Ananda Kumar, alumni of this university, and I work for Koteva Agri-Science. So for the first question, China versus India, and also you can say US versus India. So most of the points were covered by my fellow colleagues. But one thing I wanted to add here, the proper utilization of germplasm is very important. So use of like temp-trop mix, or you can say subtropical germplasm will work in most of the countries. We can compare it to India. So India is majorly tropical. So most of the 70% of the maize which is under tropical and rain-fed, and 30% you can say in post-rainy or rabi season crop. Majorly you can say Bihar and parts of Andhra Pradesh, Telangana and Madhya Pradesh. There we can expect, you can see more yields than rainy season. So same kind of environment which is prevailing, favorable environment which is prevailing in China and also in US, and also population is the main thing. So they have more of erect plant stand, and their hybrids and varieties can accommodate more population, and they are getting more yield per area. So if you compare in 3.5 metric tons per hectare is the India, and if you see China it is around 8, and if you see US it is more than 10 tons per hectare. So it's all depends on genetics, and also climate plays a major role in this, as Shambhu sir already narrated about that. So coming to the major diseases and pests, so if you see loss by diseases are more than pests, I can say. So if you see one of the disease, if I take TLB, so up to 40% of the yield loss we can expect from the TLB, and also other diseases like stalk rots, they are also up to 30% of yield losses we are getting from these diseases. So in this front, so we should be ready to handle these disease-resistant breeding through proper balancing of technology and conventional breeding. So we in the past 3-4 hours, we discussed about many technologies, new breeding technologies. So how the proper utilization of those technologies is main important. So the strong blend of phenotyping is very much required. So proper phenotyping, without proper phenotyping, we can't do any genomic selection or anything, because whatever we do in the field, that reflects in the genomic selection also. So and other technology, so double apply technology, as a maize breeders, we are so blessed to have double apply technology, so which is not there in some other crops. So we can able to get better double applied lines. Those lines, double applied lines can be utilized in the genomic selection. With the proper precision phenotyping, we'll definitely able to solve this major diseases resistant breeding. And also pests, when it comes to pests, fall armyworm. So maybe I think 4 years back it has entered in India. So it is really devastating, especially in rainy season rather than post-rainy season. So we have resistant breeding, so but 100% we can't get the resistance from the fall armyworm. Even if you say 90% we can expect in the conventional breeding, but why not GMOs? So in India, GMOs still, it is question mark. But if you see overall globally, approximate, I think 36 countries already GMOs approved, and 30% of the maize is growing under GMOs. So why not in India? That is the policy issues. Those are the questions we have to think and solve. And if not GMOs, we can go for gene editing. We have discussed a lot about gene editing. You can knock out the deleterious genes and insert the desirable gene for the traits. So those are the things for disease and pests. And coming to the bioethanol, so government itself, as my friend said, so we are 30% is the target for fuel bioethanol blend, and 70 MT is the requirement for 2030. So to do this, we have to breed for the bolder kernels to get the more bioethanols. So bolder kernels, which is having more starch, in turns it is giving more bioethanol. So that is the target we have to achieve in this. And also the starch content will be more in dent kernels compared to the flint. So we have to think about semi-dent and dent kernels with bolder kernels, which gives more ethanols. And coming to the short corn, so short corn still is under process. So I have some experience in short corn also, but so there are some deleterious effects also. So still it is underway. So as Tico sir mentioned, so shorter plants accommodate more plant population and yields better, and mostly it is a kind of temperate genetics, not the tropical. So still work is underway. So people, we have to, as a scientists or breeders, we have to think more on short corn and knock out the deleterious effects which are associated with the short corn. And coming to the one point we have not discussed, so mechanization, why not mechanization?
Yes.
So mechanization is the need of the hour because urbanization is increasing. People are moving from rural areas to the urban areas, and we are going to face huge challenge of labors. So mechanization is the most important thing. So we have to breed the hybrids or varieties which are amenable for the mechanization so that farmer can harvest directly from the field, from the combine, and sell it off to the farmer and retailers. So these are the submission from my side. Thank you.
Thank you. Last point was a good addition because we didn't talk about that farm mechanization, this mechanical harvesting.
Yeah.
It is happening in the north in wheat and it's quite common. But I think one of the problems with mechanization is we still have 80% farmers who are 1.5 acre or less than a hectare. So how do we get the machinery that can help them harvest? And for me, this is something that I would like reporters to note. The FPO is a great idea. If the FPO really happens, then this mechanization will happen. And it will happen at cost-effective rate. So something at a policy level we have to do so that there is machinery that can take care of, at the FPO level. But iske liye, I think there is need for government also to address it along with the right stakeholders and come up with a policy on this. This is not only here. I have written an article recently on farm mechanization, this mechanical harvesting for 3 crops. One is cotton, one is tomato for processing, and chilies, red chilies in Guntur. 6-7 months we have to wait, or 8 months to finally harvest the red crops in Guntur. And there are now genotypes available in all these 3 crops where we can harvest mechanically. But I don't see enough happening. I think now we should add maize also to that. Why not? So, but I feel in India we cannot go away from the small farmers. FPO is probably the route that needs to happen. I would like to add one more point on that. Like this yield gap, our research yields are 10 tons, 11 to 12 tons, and our average yields are 3.5 tons. One of the problems is this smallholder farmers. I think the knowledge transfer is not happening. I was talking to many of my private sector colleagues, because in private sector now there is a department called PD, Product Development. And through that, they do on-farm demonstrations. And I asked them, so have you seen the farmers getting more yield because of your intervention? In vegetables, there's a lot of examples. And it certainly increases their yield by 20% with not much, except a little bit more attention to what they need to do, which hybrid or variety to grow, when to do what kind of fertilizer to use. This needs trained boots on the ground, and we had some discussion under the Academy of Indian Horticulture. One of the proposals is that we need livelihood positive extension workers in the country. And my problem the other day I was asking to one professor here. I said, you know, extension division and the extension workers and they are always at the lower rank of the hierarchy. They don't get the same attention that the breeders. I'm a breeder, but we get much more attention because we release a variety. I have actually in my talk in Bagalkot 2 months back, I said, why don't we have an Indian extension service? So they come at that level, and can this be taken as a discussion point at a very high level? This can come from this as a recommendation. Can we discuss this? Why not have an Indian extension service? And I think government should spend on this and make them relevant, give them the right training. We can start the training from the university level, like a finishing school where they can get additional training on practical extension related stuff. So that is all matter. I think before I do some conclusion, can we have some questions to the panelists on this session? Please feel free to anybody. Dr. Krishna Kumar, you raised the point of soil health. Why don't you? Oh, yes, yeah. Please, Dr. Biradar.
Sir, it's a very good thing that in the entire discussion, in the maize discussion, now something came about the ethanol, breeding for ethanol or ethanol, these things. Apart from the board grain and high starch, ultimately it is the proportion of amylose and amylopectin that matters for the ethanol production. So you have to focus on the lines which have high amylopectin for the ethanol production. These are long-term goals we need to devise now only. We have to classify the genotypes with high amylopectin. And in our university, we have developed one very simple technique that is a cut and dip technique where we can identify the lines with high amylopectin. You can just cut the grain and it will indicate, you can classify it is a high amylopectin and low amylopectin.
Okay.
So like that, your germplasm, please classify that as a high amylopectin and focus on those lines. So that definitely they will yielding more than 30% of ethanol. Those lines with the high amylopectin will be yielding nearly more than 30% of ethanol. And that pinpointed breeding objective, each company, those which wants to focus on this, need to be developed. Thank you.
Thank you. I think that's a good suggestion. Sir. Anything you want to respond?
To add to their point, sir, this estimation of amylopectin, the non-destructive approach could be a better option, like NMR, NIR. How we estimate the oil content in oil seeds.
Yeah, that is very good. But we searched. We searched. But it is only a single seed. Entire genotype. We take one seed and classify that. That's all.
Right. Sir.
Only one seed. No problem. Entire genotype. This thing can be just one seed. You can cut and say what is the proportion of amylose and amylopectin.
Thank you.
Yeah,
yeah. The current hybrids now yield around 65% starch, 68%. Yeah. And the approach, our target should be around more than 72 to 75% of starch content. We'll now recover more ethanol from such kind of lines or hybrids.
But as you said,
not the starch, sir. Actually, now whatever the maize lines we have, the amylose has somewhere around 30, 35. Now, you have to identify the low amylose. See, in one of our study, the amylose as low as 5% lines we have defined. Right. So they are very good for the ethanol production. 5 to 10% amylose means very high amylopectin. Definitely, it will 100% whatever that you fed. Starch will be converted into ethanol.
Yeah, maybe 75 to 80% of the amylose should be amylopectin.
Exactly. We should identify the lines with less than 15%, 20% amylose.
Thank you.
Panelist, yes, I'm Dr. Ashok Sajjan, Professor of Seed Technology. I need some information with respect to the particularly corn seed production, seed industry. The quality control or quality management is a very difficult task, particularly field, as well as seed testing laboratory and other. You have any AI-based tool for the seed quality control mechanism in the seed field, as well as even the laboratory, even the transit, sir?
AI-based on for what?
Seed production. During the seed production, any tools?
Actually, You want to answer? Yeah, first.
Private company, please.
AI-based approach is not there, but we have a perfect set of approaches like organizer and farmer contract
That's
farming.
true. That is very time-consuming.
We include this drone aspect-wise management of the crops.
Okay.
That way it is there. We can perfectly track the quality and quantity of the seed received.
No, but
That
there are
approach is
now seed production. There is a company called Crop In. I don't know how many of you have heard. They have a AI-based system on. Like you are, they'll tell you once you tell them that what are the areas you have to plant. Satellite-based imagery of that plot, they will be able to then monitor on a day-to-day basis whether the guy has done the sowing or whether, what is the crop situation. If there is a disease, they'll give you an indication. It's now a global company. One Dr. Krishna Kumar actually, his name is also Krishna Kumar. So he runs that company. That will give you AI-based monitoring system from sowing till harvest. And they will give you a prediction of how much yield you will get from those plots based on the plot's history and the digital, it's basically a phenotyping tool, but satellite-based. So there is one company, another company, Israel Chemical Limited. They are in India. Dr. Biradar is there. I don't know if you know him. He is in Pune. He made a presentation in our one workshop in Aurangabad the other day. They are saying that you give us a geotag and a photograph of your plot. They will give you, and the crop you want to grow, they will give you exact kind of requirement of the various macro and micro elements, and you can then grow a crop to better seed production. So that is my reply to that.
As you said, with respect to the particularly maize, to identify the op types, to identify the setting tassel is a major task. So at such a situation...
Today it is possible with the digital phenotyping. It is possible.
Okay. Similarly, in case of seed testing also. So seed testing is very laborious in the laboratory. So is there any mechanism or is there any AI tools to get the information immediately, which can correlate to the physical laboratory germination and all?
You mean...
seed testing also.
Seed
testing. To take the example like seed germination.
Seed testing. Seed testing, I don't know if they are, but seed test. There are companies now. If you give them a seed with some impurities, they will increase the purity to nearly 90-95% based on the seed weight and the seed. They do some kind of a rolling on the belt, and digitally they will remove the debris and other things so that you get uniform sizes of seeds. These kind of companies are available. I know some company in Holland. I don't know if there are any Indian companies.
Because there are all the very big companies, stalwarts in the car industry, whether this can be days to come to implement those things.
Yeah, it should be nice. Yeah, you want to add to that?
Yeah, even as doctor was saying, like satellite-based imagery. Even we also use drone, where it gives. We use some lenses, which is which gives soil health, based on before planting and during the season, and wherever the patch is there, it gives the data. Based on that, we can take some control measures, and we can have the good yield data. It gives up to the harvesting the UAV. So we use different lens. Red, IR, and IR will give the imaginary data. So that based on that, we can go into the results.
Thank you.
We also use as the for pollination also drone we are using, where we can minimize the human intervention as well as labor is the measure.
Which crop you are using?
Thank you. Sorry?
You are using a drone for corn. Which crop you are using?
Corn.
Corn does not require, it seems, pollination because each tassel contains around 20 lakh population of the pollen grains.
During environmental challenges. So we may, because we don't want to lose, because now this is the biggest challenge, as many of them speaking based on the ethanol. We are not speaking like the acreage is increasing, but the producibility, the COGS, there it's a challenge. the market demand versus the supply is very, very big challenge. We faced in the 2025, we don't know how much we will face in the 2026 because most of the like a major acreage of other crops is pulses as well as oil seed is coming to the corn. So we need to increase COGS as Dr. Prasanna was saying.
Yeah.
Now that is the biggest challenge.
Thank you. Thank you. I think the answer is that systems are available. We need to
implement and modify the to suit our
we will have
requirements.
many
Thank you.
other solutions to them, but we need to contact them.
Because labor is a very costly input.
Aparna, we will have the addresses, I think. We'll send you the...
Please, sir, please.
To the organizing committee and
We'll get it.
reach you.
Oh,
Thank
so
you.
that will work.
Dr.
Thank you.
Krishna Kumar.
Excuse me, sir. I just have an information. Yes. I just have an information. Two BSc Agri students from Kaveri Agriculture University, they have developed certain AI-based systems which are going to be used to test the seeds with respect to the germination and so on and so forth.
Okay.
They are recently being called by the ISTA, which is going to meet in Canada. Full-fledged. There are 4 people who are going from Kaveri Agriculture University to that.
Very nice.
Just information.
Thank you. Thank you. Yes, sir.
I'll be very quick and to the point. We had a meeting on seed production or on rice today morning. Now we are talking on maize. Yesterday we had on legumes. The package of practices for seed production, package of practices for crop production, we are assuming it to be almost identical. I would request, even if necessary, ATPBR or somebody can really look into, but we need to come out with certain specific, very intensive seed yield and seed quality being the prime factors. What should we do? Because it's an extended period and something that needs to be done, suggestion that all of you can think about. That will also apply to others. Second one is, it is in the interest of the maize industry to, see, maize is grown either in Kharif or in Rabi, but we are also seeing continuous cropping of maize in certain places. What is impact on soil?
Yeah.
If as a consortium or if you can give it to somebody, one way or the other, even if you can suggest a cropping system whereby soil health will not get deteriorated, it will help the longevity of the crops and the varieties and other things that you are, and it will also help you to counter certain questions which may come into the future. My third point is the utilization of maize is in various facets. It is going to starch.
Utilization of?
Maize, per se, corn.
Yeah.
It is going as a starch. And starch itself is there is so much of diversity in starch. Yes. There are specialized starches. Some of them are going for medical purposes and many others. Some of them are going for edible purposes. Some of them are going for poultry purposes and various others. Maybe a time has come to identify these traits, like he was telling amylose. It's also what you said is very critical in potato. When we go for potatoes for chips, we are very particular about these particular traits that's coming.
Yes.
Likewise, you can look into what are the traits in the genotype that I'll be looking. Supposing the genotype is not great, but it can serve greatly for poultry feed, let me encourage it for that. Yes. If it has to go for a starch with a specialized kind of this thing, so those kinds of things let us try to bring. The last one is, I don't think we are at a stage where baby corn or sweet corn, there is a gap between the seed requirement and the availability of good quality seed. And one of the factors that is also hindering is that I know Bayer has been a leader for a reasonably long time, but I feel that this is one crop, considering its short duration and also its economic viability, very rapid urbanization that's taking place, this will have a lot of demand. A little bit of shift, a little bit of more thinking with respect to how best we can advance this particular segment, especially in peri-urban areas, that needs to be seriously looked into. My last question, why did the maize price fall all of a sudden? Do you have any answers?
Sorry?
Why did the corn price all of a sudden took a nosedive? Do you have an idea?
Maize prices have come down, sir. From a
It was very sharp. People are protesting. And people are not happy. People are, you saw the news. How it is. Why it happened all of a sudden? 700 down. Why it happened? Do you have any idea?
No, I don't have. I hope somebody is here.
No,
I don't know. Is it because of the
industry should tell.
Is it because of the, yes? Any of you?
Maybe in anticipation of the import of the maize. And also the quality of the Kharif maize, what produced, they also not up to the mark.
See, there are two things I would like to tell. Like Dr. Tikku will appreciate, the elastic and the volatile. What you are seeing is a volatile drop in the prices. It is not an elastic aberrations that we are seeing. So whenever the prices takes a nosedive, as we call in economic terms, we have to understand the reasons behind it. Sometimes it may be simply the reason may be Bangladesh. We don't know. Dumping of material for somewhere outside. But it's worth to look into these things.
I think that's definitely an economics reason must be there. I have not, because I heard the price drop just today. Maybe I was not aware of that. But I will dig into it and maybe try to find out. Is there anybody else? Or Aparna? Yeah, please.
Oh, yeah. So the precision agriculture could be one option, or the mechanization could be one option, or maybe n number of reasons are there that we are lacking this whole bridging process. So what is the solution what you are recommending, which we can take it forward as a prime recommendation from this meeting, that how the knowledge from the lab need to be transferred to the land? For instance, Abhi Krishna sir just mentioned that the student, they have developed AI-based technology. So similarly, is there any option that some app can be developed for knowledge transmission to the farmers, or any other solution to really bring the knowledge to the farmer level?
See, one of the things that Dr. Krishna Kumar also mentioned, our POPs are pretty, I would say, kind of, it's uniform. You go to anywhere, it's the same thing. There has not even sometimes changed. So we need to have them in the right language and with all the latest information. And we need people who can transfer that to clusters of farmers and make it happen. That's my one thing. So we need to work on that. I don't have immediate solution, but we need an important area. My main concern is that we don't have people on the ground who can reach them. See, for me, it's like we humans, we have a problem, we go to the nearby clinic, we have a general practitioner. We have a still more problem, we go to a MD guy. And if we have some more problem, we go to super specialty guy. Same thing happens in the farmers have a problem, where do they go? So maybe there are now companies who are digitally telling people that this disease is there, but whether that is wholesome, available or not, we will have to develop that system. I agree. Anything else you want to add? Sure? Yeah, please.
Yeah, in Bayer we have developed one app that is called Farmrise, and I think almost 10% of the farmers they are using. We have many options. One is environmental, it gives market, how is the market scenario, how is the price, and other thing, disease. They can connect and they can post the pictures and they will get immediate reply for that. So this is one of the and even we have a package of practice for particular, it may be a crop wise.
Take the lead and give this information to other organizations as well, and we can help for the circulation of this information to the wider audience, because now you are working with your own channel, and if multiple organizations will be involved in this whole process, the magnification will enhance immediately.
No, private sector say, because then there will be competition. Meri variety karegi, grow ki hoga. That's a, it will have to be done from the public sector, maybe in collaboration. That's it. Is there any other? I think we'll close. We have had oh yes, Dr. Subi. last question.
Hello, sir. I would like to add some points, and the question was why the maize prices has crashed. And I have attended one brainstorming session in the past week, and it emerged that we have established the where the maize goes. In India, it goes to ethanol. For the past 2 years, it's going into ethanol, especially the past year. And also, it is the maximum consumption is the feed industry, sir. 60% is going into the field. So when you check it in the past 4 years, the area increased from 9 million hectare to 14 million hectare. And in this year, we got the 43 million tons of production. Sir, all these ethanol industries, they have, I think many of you are joined in the IMR ethanol project. This private sector, sir, we are in collaboration, sir. We have increased the area. So much of maize production happened, but they have established so much of ethanol facilities also. The distillery capacity has increased. Their tanks are full. They are unable to, because the ethanol blending target has already happened. Their tanks are full. They are unable to take up more. And another thing is that next demand comes from the poultry sector. And in this, as doctors suggested, that it is the maize they got in the Kharif season is of high of aflatoxin and more moisture, because they have a technology to check the aflatoxin content in 10 minutes. So all the consignments are rejected at the factory gate. So it is one thing. So one solution emerges that in the Kharif maize, when we increase the capacity, and also when we those farmers, FPOs, has to be provided with the dryers. So we are linking with the private company. So from your CSR fund, we are establishing. Sir, another thing is that I would like to add about the extension. Sir, IMR is into extension for the past 3 years. And if you can see the Assam success story, sir, we have captured all the rice fallow land of the Rabi season. Because Assam go for 2 rice, then their Rabi fallow is there. So the productivity of Assam went to 700%. Sir, you can see it's a success story.
So exactly, because we need that extension to happen, which is not happening at the level we need. After all, we have how many? 47 crore people are into farming now, or more probably. I think we will close here. I just want to make one point. This probably gives me the fact that, she says it has gone to 14 million hectares, because the 24 it was 11.5.
Yeah.
If that is the case, then maybe our market is not ready for that extra production. And that's why, that can be the other reason. Yeah. I want to end with one thing is that's coming to my mind is that we need to, like vegetables, do consumer-based segmentation breeding in corn. Like this starch separately, biofuel separately, and the orange, this for poultry separately. So that segmentation will have to be developed, because in vegetables, we have already reached that level that we now create segments for different consumer segments. So I think otherwise I don't want to spend another, we are already 7:30. We will thank you very much, my co-panelists. Thank you it was a good interaction. Thank you to the audience for the nice questions and discussion. Thank you very much.
Thank you so much, sir, for this wonderful discussions. Now is the time to felicitate our panelists. I request chairman of the panel discussion, Dr. Tiku, sir, to felicitate co-panelists. I request Dr. P.L. Patil, sir, Honorable Vice Chancellor, sir, to felicitate chairman of the panel discussion, Dr. Tiku, sir. Sir, correct. Thank you, sir. This conference has witnessed a perfect complementation of sharing of knowledge between private and public sector. To take this forward, we have a special event of interaction of students with the private personnel. So I request a representative from Advanta Seeds, Bayer Crop Sciences, Corteva Agri Sciences, Bioseeds, Rallis India Limited, Mahiko Seeds, Indo-American Hybrid Seeds, Kaveri Seeds, Nath Biogenes, Rasi Seeds, Nuzi Video Seeds, and also I request...
So as he announces, please come and occupy your chair on the dais. Industry representatives, please.
And also if Dr. Sangam sir is there, I request him also to join. And this session will be chaired by our Honorable Vice Chancellor, sir. And yeah, Nirmal Seeds, Krishidhan.
Yeah, please come.
Crystal Crop, Indo-American, I have told.
Yeah, nice.
Kaveri, Nath Biogenes, Rasi Seeds. Any other company representatives, if they are sitting in the audience, please come and join the team.
Don't sit away. First occupy as you come. As you come, you please occupy. This side is also. Yeah, it is. Students can than the students. More takers is always
So in this session, just we want to make sure that what is expected from the companies and what type of research we are doing, and then where we can bring a perfect complementation to take the Indian agriculture towards Viksit Bharat. Now I hand over this session to Dr. S.A. Desai sir to take it forward.
Respected Chairman, sir, already time is around 7:25. It's a great opportunity for our students because they have requested, because we are one side, we are working on research, and every time, whenever we are asking for our fellow colleagues from the private industry, so we need still more training. Many times it happens. We need 3 months' time, 4 months' time. Now the question is, so we are working for all the 3 years, we are doing good work, we are publishing the good thing, but when we are coming to the private industry, what is their need? So I thank for our Vice Chancellor. He has already made available as a co-chairman. I would like to announce openly, co-chairman from the private industry in the PhD group. A small requirement, he must have released some technology is enough. So with this, the students have asked, especially MSc and PhD, totally from our whole PG group, would like to interact with you with a few. And I would like to request our PhDs. They worked on certain aspects, maybe 7 or 8 PhDs, they worked on the certain aspects. With the permission of the chair, they would like to present their work in 2-3 minutes or 5 minutes. And after that, after the completion, you have to interact with this one. After that one, some MSc questions are in general. With this, I would like to hand over the chair to the President of the function. Honorable Vice Chancellor, sir, please.
Thank you, Dr. Desai. First, let me beg your pardon for conducting this unstructured, almost unannounced quiz. I think every one of you must have undergone at your undergraduate level. The present generation has not, no opportunity for them to undergo an unannounced quiz. We have all undergone. So like that, this is unannounced quiz to all our industry partners. So this is just to give capacity building to our young generation, young researchers, what they expect from the industry, what they have done so far as their postgraduate research, either in the MSc or PhD, how it can find place in the industry. Maybe to go further, are they on right path? They want some guidance from the industry. Now it is entirely the business world. So agriculture is not just for doing for food's sake. So since we have accepted it as an industry, so they should have an exposure. So many industry partners are attending this conference. So we thought today in the afternoon or in the morning, why not to have this interaction session with the students and the industry partners. So with this, I request the students one by one, please express your idea or whatever is there in your mind. Totally it is unstructured. Whatever is there in your mind, what you want to ask, what you have done so far as a MSc or PhD student, and out of this, that directly you ask guidance from the industry partners. Please, one by one, please. Come voluntarily, either there or on the stage. Better you come here. The program is being telecasted through YouTube.
Live, sir.
Yeah.
It's live, sir.
Yeah, program is live. Yes, please.
Sir, good evening, dignitaries and the dais. So, myself, Harish, sir. Yes, sir. Yes, sir. Okay. Sir, myself, Harish, a PhD scholars from Department of Genetics and Plant Breedings. So I am working in cotton, sir. My objective is achieving multiple generation in a single year. So I am working as speed breeding in cotton, sir. So I am exploiting embryo rescue technique for advancing the generation, sir. So we are using, we are taking the bolls at a 40 days anthesis duration, sir. Means after 40 days anthesis, we are taking the bolls and we are subjecting to embryo rescue. So after that, we are hardening the plants. So after that, we are not waiting for the seeds to come. Again, we are taking the 40 days anthesis bolls from that generation, sir. So if we are doing like that, means continuing that, we will able to generate 3 to 4 generations in a single year, sir. So my question is that, we are doing embryo rescue, sir. Means we are almost we are reducing 55 to 60 days in one generation, sir. So can we integrate AI, sir? Why we have to go for up to F8, sir? So F2 is a segregating population. So anyhow, there will be some homozygosity will be there, sir. So using AI with enzymatic based biosensors, we can identify homozygous line in the F2 generation itself, so that we can working on that particular populations. So no need to go for up to F8. So is it possible? I have AI based ELISA sensors. We can, because
interact to him.
Enzymatic expression means why we are choosing enzymatic expression means, so it is for identification of homozygous, heterozygous. Sir.
Harish, this thought process what you have shared is excellent.
Yes, sir.
Already people are working on making the lines homozygous in earlier generations itself.
Yes, sir.
So in other crops, we are having DH technology. Unfortunately, it is not there in cotton.
Yes.
So now, as you are mentioning, F2, you have to identify the homozygous.
Yes.
So in the case, if you're going to use more of SNPs for the required traits, if you're going to identify the homo alleles, in the case, there is a possibility for identifying the one which is having maximum homo alleles.
Yes, sir. Sir, otherwise, can we take any vegetative parts like leaves? So we can go for enzymatic, means some protein extraction From that, we can use enzymatic data. So that enzymatic data, based on that data, we can classify homozygous and heterozygous.
So the time period is important, as you're saying.
Yes.
It is going to be for speed breeding.
Yes, sir.
If you're going to go with the approach what you're saying, if it is going to take comparatively more time, in that case it is going to be not useful for us.
Yes.
If you go for SNPs, it is going to be taking lesser time comparatively,
Yes, sir.
and you'll be getting more number of data points. With that one, you are going to identify the homo alleles.
So along with embryo rescue and this AI, if we used AI also, still we can, I think 5 to 6 generations in a year we can achieve, sir.
Sure.
So means like now instead of going genotyping, so I thought that so we can go for some biochemical markers like enzymatic markers like isozymes and all.
I think we can discuss in detail later on.
Yes,
If somebody is having some points, they can share.
See, as a student, commercially you have to think, right?
Yes.
He said 2 points. One is your molecular assay
Yes, sir.
versus the what you want to do. Okay.
Enzymatic.
Imagine you have 10,000 lines.
Yes.
So which one you will do faster? Like you take 10,000 samples,
Yes.
markers, forget about cost first.
Yes,
okay. The way industry works is like you have the time is important. If you are valuing the time, again your breeder standard genetic gain equation below
Yes,
will be
sir.
T into C. It is
Yes, sir.
time
Yes,
and
sir.
cost.
Yes, sir.
So time is important, cost is important.
Yes,
SNP,
sir.
SNP is now works in like 6 cents, 5 cents per SNP marker data point.
Yes,
It's
sir.
very
Yes, sir.
cheap,
Yes,
right?
sir.
100 cents is $1.
Yes.
So that is very easy. You can identify, but again in F2, what you are trying to do is you have a range of variation
Yes,
for
sir.
homozygosity.
Homozygous, yes, sir.
So you are trying to select that end spectrum of homozygosity, which is
Homozygosity.
more towards more than 75%.
Yes, sir.
That is what you are trying to
Yes,
do.
sir.
Right? So you will achieve that homozygosity faster. But if you do this analysis for 10,000 lines,
Yes,
it will be time consuming.
Yes, sir.
And all that by the time you want to make decisions, your plant is already crossed that stage to another round of embryos.
Yes, sir.
So you have to think in those terms, like application, theoretical one part, and practically how you want to integrate into the commercial breeding.
Yes, sir. Sir, and then like in private, sir, like in private industries and all, so are they using AI for phenotyping, sir? Like how means my question is that they're using cameras and sensors, sir. But how that image data, it will convert into numerical data, sir?
So there is for example, we do some technology. For example,
sir.
we flew the drone,
Yes,
take
sir.
images from the field,
yes, sir.
and you take tassel height in corn, for
Yes,
example,
sir.
or in case of rice, panicle height, and try to assess indirectly
Yes,
the
sir.
plant height. Right? First, you have to train the model. AI is all about training. How it's not like something you give and it will give you something back. So you have to train the model, give multiple training data sets. Once you get that your reputation, heritability, indirectly heritability of the data, then you will use it in deployment. That is how it works.
Thank you. I think, students, I think you have got the answer. Okay? Otherwise, you can also discuss off the stage. I think next question should be very brief, and you should take the brief answer. That's the thing. Okay? Next, who is there? Please come. It's not only related to your research work, you can also ask the production side and other sides also. That's very important. Okay, don't restrict yourself to your R&D or MSc work. Okay, in general questions, please, only one question.
Job opportunities, so
I think, sir, if job opportunity, everyone will ask for the job. Okay, that's right.
Good evening, everyone. I am Maulia. I am also working in cotton itself, that is whole genome sequence analysis and genomic studies for earliness in cotton. And I have some bioinformatics work, and I'm working on high density planting system also. Sir, what technical and soft skills you people from company expect from us, and can we expect any internship opportunities so that we'll come to know about what a breeder role that is different from academics and in your company?
I think yesterday when I was speaking to Bayer, any Bayer representative here, I think Dr. Viresh Gowda, who is from the university only, he has already talked to the university people, and I think there are chances you can go and visit their, especially even Corteva. They can also take this one. Students can go there and work. Okay? Corteva, from Corteva, I think please comment on that.
Mic to your closer.
It's not only from the R&D side, even production and testing. That's what he's going to tell. Yeah.
Yeah, we just, it's a progress is going on. I think we will come back because some internal structure in the buyer organization globally. So that is happening. Because of that, everyone is a little bit occupied there. So I think it may take some few more months, and we will come back, and definitely we will connect with the university, and we will have like a whole day, somehow, like we will select few of the internship candidates. Not
But
only
take-home
only
is
breeding,
definitely it can be worked
we'll
on.
have some different
Okay.
department also.
Okay, thank you. Next,
Thank you.
Next one, please. Yeah. Fast, next, please. Next 4 more questions. I think that's enough.
Good evening, everyone. I am Trilok. I am from MSc second year. First of all, thank you all, sir, for sparing your time and interacting with us. And my question is, like, we will study 2 years for MSc and 3 to 4 years and more than 4 years for PhD. But then also, if we enter an interview into a private industry, we will feel like we don't know anything and our mind is blank. Because, like, we will study everything that is related to genetics and breeding. But sometimes we don't know, like, what to study when we...
Okay. What his question is, I think employability. That's what he's telling. Whatever is grooming here, I think that's not sufficient when we go to the private industry. That's the thing. So for the employability, what best thing can be done? I think, anybody from this one? Shambhu, can you? Or Girish? Yes. One point, please. What best can be done?
For employability, actually, you should really focus on what is happening about your project or what work you are doing, how you can commercially, pack the farmer. That is very important. As a private companies, we always expect, whatever the product we try to develop or put into the market should be revenue generation. That kind of. So you have to globally see it, what kind of work is going on your project, actually what you are doing, and internally and in India also how it is going to impact the farmer, and how it's commercially viable kind of a thing should be focus. So bring to the table for discussion about this.
Thank you very much. Corteva, I think just from the Corteva.
I'll just try to connect two things like internship.
Your TNU model, I think, just
yeah.
the best
So
thing.
I mean, what like TNUs are like. For example, myself, I did my external PhD while working there. So we developed some contacts. Then they they are inviting some of us as a professor of practice. So we just go there once in a year, a couple of years, sometime online, have an interaction with students. Sometimes they give some topic or you on your own topic. That gives that that we know people. Sometimes if there is any requirement or something like that, they will give us. For example, yesterday I did my PhD with some, she was asking for an industrial partner for female fellowship. I'm just exploring that possibility. Like that, some connects.
Thank you, Dr. Somagowda. I think the very important thing here is we need to have institute and industry interaction. That's the main thing. I think that has to happen at the... yes, sir? Yeah.
Actually, when we visited Bayers, they said that there are some PG students who have come to the, their facility. Part of their work has been done there. And after that, they have been absorbed in the company. That's what Vireshgowda told. Yes. Like that, I think you may have to help us with the MOU with the UAS, Dharwad, and take some students for further training or maybe further their studies. Yeah. And then if it is possible, they can be absorbed if they are useful to you. That way you should go. And yes. All the companies, you should try to have this thing, especially internship, what you said. That was done, especially Bayers, 4 or 5 students of GK, VK have been absorbed there. In that way, you have to go.
Giri, Ramchandra.
Yeah, I think it's well said, sir. Actually, if you look at any company for internship, nobody says no. Actually, only you should go and approach. So everybody is accepted. I think TNU is the best example. They are coming to even our company. Biostate, they are coming. Corteva, they are going. Everywhere they are going. I think the professors, that's a requirement actually. Basically, for PhD and also MSc, it's a requirement. They should at least spend 15 days in the industry. So that way, if we can make it, I think nobody refuses. So first thing, I just want to add what he was asking on employability. Just I think when you are a student and when you are doing a BSc or MSc, so first job is to learn technical things. Technically, what you can learn, you learn first. That is more important. So afterwards, you start implementing the things what you can do in the research. So that is next one. So second one, you keep open mind for the new things, new technologies. So even if you cannot do practically, at least learn, at least read, learn. So that is the first thing. The attitude of the new things is important.
Thank you, Ram. Thank you, Ram. I think Girish, one point.
I would like to add some more points to whatever Dr. Ramachandra has said. As he rightly said, for internship, no company will say no.
Yeah.
But some other points are coming in my mind.
Okay, the important point is no company says no. I think the interaction has to happen between institute and there is one DR and DA.
Especially,
yes, one point.
Only one point.
Yes, please.
Actually, what is coming in my mind is whenever university is assigning a topic, research topic, it is better to have a collaboration with the industry consortium and academia, so that whatever is required for the industry, that topic, if it is given
that's good
to the students, it is very beneficial for the industry, and absorbing also it will be easy. And second point I want to convey is, like any IT companies, how they are conducting the interviews, campus interviews, internship, or even internship as well as a job purpose. Like I'm requesting to my all industrial colleagues also, we have a consortium, or even if it is not consortium also, if you can go to any campus and have a campus interview, and like that also can start.
I think in Rave you can include some industry visits also. I think that's best.
Yeah.
Okay.
There, it is there.
That's there.
It is there.
Then
Now it is there for the PG and PhD.
Yes. Anything from Indo-American, Advanta? Yeah. Yeah.
Hi, Sachin here from Advanta Seeds. Sir, we are open for internship. Already we have some student in our biotech lab, DH lab in R&D and in pathology also. And we are hiring intern in production also. So need to approach with concerned person or with HR. So you have to approach. And if any vacancy, as they said, no company say no. Yeah. So we are open for internship.
Thank you. I think take home is they are ready. I think you should be ready.
Actually, the major advisor or the head of the department has to, assign the work to the student little bit from the different companies, and then you approach it. That's what GKUK has done. And then you can have a MOU with the university. With that, you can send for some training or maybe part of their research work there. That's how you have to identify. Then if they do well, they may absorb also. That's what has happened in Advanta and also Bayer, I could see. When we visited their lab, there were students working in their lab. And part of work, or maybe for the training, they said, DH or the other things, if you want any training, you can send them. Like that, you have to have the program.
Sir, good evening to all. Myself, Arman. I am first year MSc student from genetics department. My question is that in some private companies, they don't work on some self-pollinated crops as the government sector is doing. Is there any scope or what the private companies are expecting from us to skills and qualifications related to that question?
I think the point here is, that private companies, they work on the commercial angle. That's a very important thing. If you have the varieties with the commercial angle, definitely they are going to work. I think the pigeon pea is the next thing. I think you want to suggest something on this?
Sure.
Yes, please.
Yes.
Nirmal Seeds is here.
I am from Nirmal Seeds.
Pioneers in, I think, improving varieties and pulses. Yes, please comment. Dr. Kulkarni.
We are working on pulses as well as wheat, OP paddy. So it is not the that private sector are not working on OP crops. Actually, there is a wide scope to develop the OP varieties also.
Sir,
so we are open for that.
There is a notion in the students. Yeah. If I work on self-pollinated crops, I have got meager or less chances in the private industry. It's a very clear cut. They don't work.
No, it's not right. It's not right actually.
It is the concept or something that is more important. That has to come from the right persons.
No, it's not that like that. We do work on self-pollinated crops also, and there is a wide scope to develop the straight varieties in open pollinated crops also.
Hello,
Excuse me, sir, I am from
Just to add, sir, just to add actually, it's a wrong notion actually. The breeder is a breeder. He learns the skills, not the crops. See, anywhere it can be fit into. Actually, all of us, we have not worked on commercial crops. So here only, I think Dharwad only commercial crops, cotton is a. Yeah, I'm forestry in tree breeding actually. I am in forestry, PhD in tree breeding. So I am not in the actually rice breeding actually. I did my MSc in rice breeding, but I did my PhD in tree breeding. So, but still I can. So only thing you are learning the skills is not the crop, anything. Nobody looks at the taking the entry level, nobody looks at the crop. Your skills only they look at, and how much you learned and what is you can utilize.
I think it is for saying. Because I have seen a number of companies, and we were also, for interview also, we went to the companies. They looked at the crop, in which crop you have worked, what is your experience, on what aspect you have worked. That's what, for the cotton, for me at least, there were, a lot of competition from the students to come to our farm to take up the, program of PG. So you said, pulses and other, the self-pollinated crops, we say. And some of you may say, and the Nirmal, he said, there are very few companies working on the self-pollinated crop. And how best those people also can be accommodated. That's what you have to tell us.
Actually, I want to add here. Yeah, any student if is coming for the interview in any company, only interviewer is looking for the what skill he knows and how best he can accommodate with the particular position.
Principles of plant breeding.
So you should learn the techniques and principle properly. That is the only one thing we are looking.
So, your crop where you work doesn't matter here. That's what the panel says here. Okay, I think be cheerful. Okay. Last one.
Good evening, sir. I am Manoj, junior MSc. Because I am first year now, before selecting my research or crop, can I work with your particular private company objective on specific crop? Can we collaborate with your crop objectives, and can I achieve through my research with your collaboration?
I think he got his point. Am I right? You want to answer? Please.
Sir, if I am permitted, I'm repeating more.
1 minute, huh? 1 minute, just. Okay.
So what I like, this couple of days back, the same discussion happened in IRI, the same thing I told. So if possible, through MSc, through university, if private industry can be part of like some co-advisor or something, we can be part of the problem. And internship, I am telling you from an MNC perspective, most highest chance is when you are working it together, part of the problem can be used in our lab or work there. Then I know that person like before they come for interview. So most of our absorptions initially happened like that only. Like internship, people came for there someday, or then they go to contract working through contract, then they are absorbed full times. Currently, we have all the youngsters which are there. Most of them come from US Dharwad, most of these people. So they are employed like that only, initially contract or through internship, then they are hired full time.
Okay, there is chance.
Actually, the same thing, I obey with him.
No.
First, he has to come through the university to the company. We can be the co-advisor or some in the members also. We can fix the objectives.
So some modifications need to be made, sir, at the university level. Okay? If they are, yes, sir.
Co-chairman of the advisory committee of the students' problem, and that provision we have made. And that formally MOU and other things we have made, what are the objective, collaboration, all those things. That he will also attend the advisory committee as a co-chairman. There is no provision to become a chairman because chairman has to be full-time. That we have initiated, already implemented this year, particularly in genetics and plant breeding. This information, I think in future we can enhance that.
One thing, it's not only the just advisor kind of thing or the part.
No, it is a problem.
Problem has to be given by the private industry. Yeah,
crop
that's
of
very
your
important.
company and the problem will be assigned to them.
Important.
Yeah, that objective will be that only.
Sir, right now we are having 34 students MSc, around 7 students PhD, 99 or 100%. They would like to work with the private industry. 100% right now. You can claim right now because they wanted to work private. Part of their research could be the private industry setup. So they will be studying in 1 year, they will be studying their coursework.
I think we will conclude. I think
1 minute.
Sir, one thing.
Yeah.
One important thing what we need to follow for the MBA, how the people they do in the MBA. During the final year, what they do, they advertise all the students' information, what they have done and everything. There only the employability starts. I think that methodology should be started here.
Yeah.
They have to, first year only, when the research topic is announced, that has to be there in the website from the department. Then people will be there from this one, they have to send it to across the private institutions. Whoever is interested, I think they can log into that particular student and they can take him. I think that's
I fully agree with you. But one step forward, just before the start of the academic year, let the private sector write to the Vice Chancellor or the Director of Research. These are our burning issues. We would be very happy if you can give these topics to the students. Now let the private sector take the initiative and write it to the Vice Chancellor with a copy to the Director of Research. These are the crops. These are our burning issues. We would like very much you to allot these kind of problems to students. And if allotted, please inform us. We will be seeing whatever field or other facilities that we can be given. So that goes a little, puts a little bit of also on the private sector. Once it is done, the university will share in their website or this. These are the postgraduate students. These are the ones who are working or whatever it is. Okay. But I'll now come to the flip side of it. I go to many companies. I also working for advisor to some companies.
I think
please don't mistake.
Can help, sir.
ATPBR
can also help in this.
ATPBR also will help. No problem. We'll try to. In fact, we have been talking that we can be good middlemen between the academia and the industry to take these things forward. Certainly, we'd be too happy to do ATPBR on behalf of TICU. I can say this, I guess. But what I am seeing, one thing is, after coming to the company, most of them seem to forget even the fundamentals of science, even after many years. I don't want to, at least today morning, emphasis was on data management. Emphasis was how do you harness the data. Emphasis was how do you squeeze the best out of your data so that breeding gets accelerated. But if you are still running around with the mean and think morphological breeding is the way forward, I think we are way behind. My request to the students is update your potential, at least with regard to what data management with respect to this, you will go miles. Thank you.
Thank you very much, sir. If 1 or 2 points from this side so that we can close up the issues. Any, I think. Sir, please.
So it's my suggestion to all students. Actually, everybody is talking about the advances in the molecular breeding tools with respect to disease resistance and tolerance. But in private sector, there are other opportunities also in the sector of seed production, then plant pathology, entomology, bio inputs. So a private sector is open for all these sections for the opportunities for your better career.
Yeah, I think opportunity is there not only for genetics and plant breeding, it is also there for agronomy, seed technology, entomology, all pathology. Okay, that's good. I think private sector is open. Yeah. 1 point, sir. Yeah.
So I am from Indo-American Hybrid Seeds. I am from the University of Agriculture, Bangalore. So in fact, US Bangalore, it has been already initiated this program.
That's good.
So they conducted industry university meet. It's not with the students, in fact.
Okay.
Because students, they will work under the guidance of what the professor says, in fact. What is the limitation they have, what are the structures, what are the facility they have, in fact. So therefore, Dr. Suresh, he was the VC, what he suggested means, we are open to share the material, even profit sharing, patenting, or whatever intellectual properties. Either private is investing, let them they share the investments. So that kind of agreement has been done. So a lot of programs have already initiated. See, if I have biotechnology, if Indo-American is doing something like that, I'm going to tell to the particular department of the University of Science. So they will say, yes, we have also facility. You can share our facility and you can work it. So such kind of things are agreements are going on. So that is the best way they're having a students, because students are helpless in fact, because I am also in the same background. So therefore industry, university meet is always good. So that respective departments which can be able to.
Thank you, sir. Well taken, well taken. I think all the private sector has got the CSR also. I think from there CSR, you can give the topics to the students and you can work there. That model also works very well. Thank you very much. I think now we have the comments from our Honorable Vice Chancellor on this. It is his brainchild, chairman of the present session.
Yeah, thanks to everyone. So our 5 students have expressed their views, especially about the, you told specific, but to generalize, she wanted to have the research facilities for your PG program, one aspect, and you want to have internship at the end of your degree program or during the degree program, so that your research will be continued uninterruptedly, and after the completion of the degree, employability. Then of course, these are the, I think, main issues that you have raised, and also some of the other aspects about, of course, self-pollinated crops and the taking you on board crop-specific and all these things. All the industrial partners have replied to you. You listened their reply. So in my view, through ATPBR, let us have MOU with almost all the industries. I request ATPBR to facilitate the MOU with the UAS, Dharwad and the other industries. Maybe seed industry or input industry, chemical industry, whatever may be. So it will be benefiting to all our the students as well as the graduates. As indicated by our Director of Research, we have already built-in mechanism to make the industry personnel as co-chairman or co-advisor in the Students' Advisory Committee, either MSc or PhD. The only prerequisite is they should be recognized as a PG teacher of UAS Dharwad. I think they are not ordinary people. They are also highly qualified, MSc, PhD in their subject, and continuously they are working on the field, maybe laboratory as well as field. And slightly we have relaxed the requirement for PG recognition for the industry personnel or scientists. Once we have MOU, then the industry can propose their faculty for recognizing as PG teachers in the UAS Dharwad. So then that path is very clear. So we can take them as a co-chairman or co-advisor in the PG research. Then operationalization of that. So what the friend from Indo-American hybrids indicated, university and industry collaboration meeting or the discussion should take place. For crop specific, we have invited in maybe in the cotton or in the maize. That is for business purpose only, not for academics. Now, I think it is very high time we start inviting. Once the PG students are admitted, okay, we initiate this, or immediately we can initiate this for the present, the students who are just admitted few months back, 1 or 2 months back. We can initiate institution and industry interaction meet so that we can have a dialogue, and as indicated, they can indicate the researchable issue, especially orienting the commercialization from the business point of view, or commercialization of your outputs. What are the researchable issues? So because their orientation is only to capture the market. Okay, so with that perspective, what are the researchable issues in different crops, in the different seed industry or the input industry? So they can indicate, and our students can choose the different topics based on what they propose, so that it will be win-win situation for students, institution, as well as industry. And they have indicated without taking the consent of their superiors, there is no dearth for placements or internship. They are open for taking you as interns. Okay, give them big round of applause without consulting their bosses.
Standing ovation, please. Standing ovation.
They have
and
committed.
Big
It's okay.
applause for our training industry.
They have committed. So there is no limitation. They are open. They are open to extend you at least interns, if not the employment. So they are ready to provide, because you will be going for the research as an intern. So, and with respect to employability, so there is a difference of opinion, of course, as Dr. Kadi sir indicated, and they look for the skill in breeding, or whatever the purpose for which they appoint you, they test you for skill. It is immaterial on what aspect you have worked for MSc or PhD. So if they are comfortable that you are good in the skill with respect to a particular purpose for which they are appointing, so they don't look into on what crop you have worked. But when there is a opportunity, when there are many persons on the market, okay, so when there are many PhDs, why they will choose MScs? Like that, when there are many persons working on the available on the crop on which they are interested, they may choose. Okay, always, so that preference will be there. But their main emphasis is they look for your talent, they look for your skill. Okay, that they have clearly indicated, and we request them not to, because there are limitations with respect to our faculty, and all cannot be allotted to cotton, all cannot be allotted to sugarcane, all cannot be allotted to maize. Okay, all the different teachers are working on different crops and different aspects in the different disciplines. So as per the availability of our faculty, the students would be allotted even if they are not interested. So it goes with the discretionary of the head of the department, and they will work for that and they will complete. So kindly look for their skill, not on the crop or aspect they have worked for their MSc or PhD. And so I think, so these are the main aspect the discussions went on. And so I think whatever the discussion has taken place, it is fruitful. We look forward for UAS Dharwad and industry interaction meet soon. So these few words, I thank all the industry partners as well as students and all the learned scientists for participating in this interaction meeting and taken interest. And also, I respect your views also. So thank you very much for this wonderful opportunity to have this interaction meeting. Thank you.
1 minute. Thank you very much for your unannounced quizzes, sir. It was a sudden, but nevertheless, it was excellent. And thank you very much for your brainchild to arrange this one suddenly, making the unannounced quiz to the all private industry group. I am very happy to see this one. I thank on behalf of the National Conference as well on behalf of the Menchikai Foundation. And soon we will write a request letter. What are the researchable issues which you can offer to this one? Yes, sir. Through ATPBR. Through ATPBR, we are sending the letter to individual. Please, I request your kindness. Please respond. Whatever the things, what best you can facilitate, maybe month or maybe 2 months. Not an issue, not an issue. So we'll be more happy. And thank you very much. And with the chairman allowed this one, it was a longest session, rather 1 hour we have taken. Thank you very much, sir. One announcement, please.
So quickly, I request all the students to please come on the dais to join for a group photo. Very quickly. Please. And as you come on the dais, I make an announcement. Tomorrow we have 5 presentations, lead talks, and 3 oral presentations, 1 panel discussion and plenary session. All this we have to do in the pre-lunch session. So that's the reason we have decided to start the session at sharp 9 o'clock in the morning tomorrow. I request all the delegates, students, to be present in this hall after having breakfast sharp at 9 a.m. in this hall.
Only at all plant breeding students there.
ಇದು ಬಂದ್ ಮಾಡ್ರಿ ಹಾಂ ಅಲ್ಲಿ ಬಂದ್ ಮಾಡ್ರಿ ನಾ ಇದು ಗ್ರೂಪ್ ಫೋಟೋ ಹಿಂದೆ ಹೋಗ್ಬೇಕು ತೆಗಿಲಿ